I'd like to welcome everyone here to Rochester and all of our remote visitors. This is our first reiteration of our kidney stone retreat. We had done this for a number of years with the Rare Kidney Stone Consortium and our O'Brien Center, and then COVID put an abrupt halt to that. So we're starting again today, part of our P20 Center. So we're very excited about that, working with Dr. Koo. And then also our Rare Kidney Stone Consortium work has continued and prospered. And we're going to mix some of our common kidney stone related research that's more related to the P20 Center, and then our rare kidney stone work, which is more related to a lot of genetics, really very genetics heavy. So I'm glad everyone can be here. I'd welcome, and then I want to give Dr. Koo a chance to say welcome.
Well, thank you very much, Dr. Lieske, and good morning to all of you. If I haven't had the pleasure of meeting you, I'm Kevin Koo. I'm a consultant in urology here, and my clinical and research focus is kidney stone disease, like many of yours is as well. We welcome our friends from our campus, our guests from afar. Thank you so much for joining us here for the first of what we hope will be an annual tradition, celebrating also our Forward Urology P20 Center as part of the Caribou Network. We're delighted to have you here, to have representatives from adult and pediatric nephrology, urology, pathology, hematology, oncology, and representatives from patient advocacy groups and other people who make up our vibrant community of kidney stone researchers. So we hope that today we'll really cover the diverse swath of kidney stones and the latest of what's happening, how we can continue to foster collaboration and partnership so we can continue advancing the science, both clinical and research science of kidney stone disease. And we hope that this will spark new collaboration and conversation. So thanks for being here with us, whether in person or all of our friends online. And please ask lots of questions, start lots of new conversations. We're happy to be here with you. Thank you so much.
Good morning. Hi, my name's Michael Romero. I'm a professor of physiology and biomedical engineering and nephrology here at Mayo Clinic. I'm one of the crazy people who likes to study kidney stones in fruit flies, and so I get the dubious honor of moderating the novel models sessions. So our first speaker is Navin Gupta, who we're very fortunate to have lassoed here from Brigham and Women's, and he's going to tell us about functional demonstration of kidney organoids. Navin.
It's my pleasure to join the faculty here at Mayo, beginning on Monday. Excellent. Thank you, Dr. Romero. So thank you for the kind introduction. It'll be my pleasure to present functional demonstrations in kidney organoids. So organoids are artificially grown tissue that resemble an organ. If that organ is the kidney, an ideal organoid would contain nephron structures as the basic functional unit. The structural complexity and cellular complexity of a nephron cannot be achieved with co-culture techniques. By simulating nephrogenesis, beginning with pluripotent stem cells, perhaps then we could generate organoids that contain full nephron structures. Human stem cells can be derived from either human embryos or, what we're using more without ethical concerns is induced pluripotent stem cells, in which you can take any terminally differentiated cell of the body, reprogram it with Yamanaka factors to generate embryonic stem-like cells or induced pluripotent stem cells. These cells carry the genome of the original host, so they're good for genetic disease modeling and personalized medicine. Stem cells are characterized by two things, the first being immortality. They have unlimited proliferative capacity, as well as pluripotency. They can differentiate into all different tissue types of the body. And so on the upper left, you can see how human pluripotent stem cells can reconstitute the three germ layers, namely endoderm, mesoderm, and ectoderm. As you go down this branch point to each nodal region, you're lineage restricting to the derivatives below, in which case our goal would be to generate nephron progenitor cells with high efficiency. Those would then differentiate into podocytes, proximal tubules, loop of Henle, distal tubule, and connecting tubule. Now, nephron progenitor cells are marked by the transcription factor 6-2, and this is lineage tracing in the mouse from Akio Kobayashi when he was in Andy McMahon's lab. And so on the right-hand side, you can see multiple different published protocols to generate kidney organoids or with their to generate nephron progenitor cells. Often these protocols involve multiple growth factors over many steps and for weeks duration to generate a paucity of nephron progenitor cells. At Brigham and Women's and Mass General, I've been working with Ruji Morazane, who developed a protocol in 2015 that generates nephron progenitor cells with more than 90% efficiency in a simple 3-step protocol over 8 days. Now, organoids generated from this protocol at day 26, here is a UMAP showing the different cellular clusters, showing that there's significant cellular complexity. Here you can see 3 clusters of podocytes, clusters of independent tubular epithelial cells, a mesenchymal cluster, endothelia, some off-target neuron and muscle tissue as well. I think this sort of highlights 2 concerns with organoid work, one being stromal overgrowth that is derived from the less than 10% of non-6-2 induced cells include stromal progenitor cells, that are FOXD1 positive, as well as FLK1 positive endothelial progenitor cells. So besides stromal overgrowth, there's also very limited vasculature in static organoids. And here you can see, so while you could have all these different cell types, the reason why you couldn't reconstitute a nephron in co-culture is that it wouldn't aggregate in an appropriate way of generating contiguous structures. Here you can see that these organoids are structurally complex. Here are podocalyxin positive podocyte clusters, as well as LTL positive tubular epithelial structures, proximal tubular structures, as well as CDH1 or E-cadherin positive loop of Henle and distal tubular structures. And these are admixed with PDGFR beta positive stromal cells, as well as a small amount of cords of CD31 positive endothelia. So these organoids have been used for certain functional demonstrations, including glomerular vascularization and filtration, proximal and distal tubular absorption and secretion, and some endocrine studies that may be germane to this group. Recently in JCI, we had published that organoids respond to human recombinant parathyroid hormone to activate CYP27B1 or 1-alpha hydroxylase to generate functional vitamin D. With implications on SLC34A1 also. I'm going to very, very briefly touch on graphical abstracts of these bolded studies, noting that I was a part of the ones that are starred. And so for glomerular vascularization in kidney organoids was accomplished by combining kidney organoid with kidney on a chip technology. In the upper left-hand corner, you can see a top-down view of a 3D printed of a chip. It consists of a 3D printed PDMS gasket, which houses organoids embedded partially in an extracellular matrix. They're embedded and adherent to permit superfusive flow through this chamber. In a cross-sectional view, you can see where it's labeled organoids is the flow chamber, and we can model with COMSOL modeling and bead perfusion studies what the flow profile is in this region of organoids. By optimizing organoid day, media, supplements, whether we needed co-culture, which we did not, and extracellular matrix type, we found that high flow corresponding to a fluidic shear stress of 0.2 dynes per centimeter squared induced significant PCAM1, MCAM double positive vascular cords. On closer inspection, we found that this stem cell-derived vasculature infiltrated podocyte clusters consistent with glomerular vascularization that progressed through S-shaped body, single capillary loop, and early corpuscle stages by immunofluorescence and transmission electron microscopy. Here is scanning EM that shows podocyte clusters within a parietal epithelial cell layer. On higher magnification, you can see that they manifest primary and secondary foot processes, forming primitive glomerular basement filtration barriers with capillary loops with open lumens juxtaposed by podocyte foot processes. This is the one study I'll be showing that I was not a part of, but is worth mention. There's glomerular filtration in kidney organoids in vivo by taking organoids and transplanting them under the kidney capsule in the mouse and creating this retroperitoneal optical window with a glass cover slip, which allowed an upright confocal to perform continuous microscopy. They anesthetize the mouse, and via the tail vein, they introduce different sized dextrans with fluorescent tags. And what you could see is while they were looking at the stem cell derived tissue, they found that, oh, my fault. Here it's important that glomeruli are vascularized by infiltrating mouse endothelia and not by, the human vasculature as in the previous study. What you could see is live monitoring of glomerular sieving of different size fluorescent tag dextrans. Large GFP tag dextrans remained in glomerular capillaries, while RFP tag dextrans were visualized in Bowman space in the tubular lumen. Extending to the proximal tubule, we could see that organoids express functional OCTs and OATs. This is just one demonstration where organoids express OCT2 in a similar distribution as human nephrectomy samples, OCT2 being the transporter for cisplatin, which at low dose caused DNA damage in LTL-positive proximal tubules, sparing adjacent podocalyxin positive podocyte clusters, and CDH1 positive tubular structures. Because as a product of Joe Bonventre's lab at Brigham, I immunostained KIM1, and it confirmed an acute tubular injury that served as the basis for a study modeling the AKI to CKD transition in organoids. But that's beyond the scope of this talk. To expand on that prior work, we looked at proximal secretion both from the basolateral aspect and the apical aspect using rhodamine 1,2,3. Organoids, this is an interesting methodology. I'd say that most of these studies are characterized by some interesting methodology, either putting organoids on chip, putting organoids under the kidney capsule and doing continuous live monitoring. Here, the interesting methodology is adhering organoids to an ECM layer in an optical chamber, and then live staining these organoids so that you could identify tubular structures. And that's with live stains for either LTL, a lectin produced by proximal tubules, or wheat germ agglutinin. And then you could take that optical chamber and image it perpetually in a stage top incubator with a confocal microscope. You could imagine in the controls, when rhodamine 1,2,3 is added, you should end up finding it in the luminal space. If MDR1 was inhibited, it would be absent in the lumen and accumulate in the cytoplasm. If OCT2 was inhibited, it will be absent in the lumen and the cytoplasm. Here you can see controls in green, where rhodamine fluorescent intensity is detected in the lumen and absent with MDR1 or OCT2 inhibition. Meanwhile, in the cytoplasm, in orange here is MDR1 inhibition. With that inhibited, it accumulates in the cytoplasm, and with OCT2 inhibited, it's absent from both the cytoplasm like it was in the lumen. We also look at distal nephron function. Specifically, this is collaborative work with Lisa Satlin's group at Mount Sinai and her perfusionist, Rolando Carasoza. So we gave him organoids of varying age. I performed the PCR and immunostaining for piezo-1, which is a mechanosensitive ion channel for calcium flux. So Rolando had microdissected isolated tubules, microperfused them, and looked for calcium flux using the Fura2 reporter, and found that organoids over time have increasing piezo-1 expression and have increasing calcium flux in response to microperfusion. Lastly, I'll just say that germane to this group, that I wanted to do a quick demonstration that demonstrated that kidney organoids express, many of the monogenic causes for nephrolithiasis. Here I just took the Invitae nephrolithiasis panel of 40 genes, mapped it to single nuclear RNA-seq of day 26 organoids, and found, that 34 out of the 40 are expressed at day 26. And they're expressed in a very similar distribution as the KPMP atlas. Many of them include, let's say, the carbonic anhydrase expressed in the proximal tubule or the calcium sensing receptor, CLCN5, CLCNK8, claudins expressed in the loop of Henle. In the proximal tubule, you can see notable genes such as SLC34A1, 34A3, 3A1. And so this is a future potential. I would say of the 6 underexpressed genes, this was at day 26. At later dates, based on prior collaborative work, I do know that uromodulin and AGXT are expressed as organoids mature. And with that, I'd like to acknowledge folks at the Visa Institute, MGH and Brigham, that contributed to the various studies that I showed, Lisa Satlin's group. I'd like to acknowledge Doctors Lieske, Romero, and Garovich for the invitation to present, the introduction, as well as the opportunity at Mayo. Thank you.
Questions? Dr. Sass.
A quick question. The slide that you showed, the impact of no flow, low flow, or high flow, it just wasn't clear to me. So it sounded like you were saying that the high flow induced greater differentiation. Did I hear that right?
In that demonstration, I was showing that the that the CD146 and CD31 double positive vasculature, was I had limited time for many studies. I will say that developed from expansion and differentiation of FLK1 positive endothelial progenitors. So this is all stem cell derived tissue. There's no co-culture. Those vascular progenitors basically proliferated and differentiated into vascular cords in response to shear stress, which is sort of something that's known about vasculature. But additionally, it caused, tubular epithelial maturation, increased slit diaphragm formation, things that you would expect when, let's say, like a podocyte comes into contact with invading endothelial cells.
Thank you.
I did see a quick question in the chat about appearance, I think, of SLC26A6, if I caught that correctly.
The oxalate transfer.
Yes, the chloride oxalate exchanger.
Yeah, I think many of those transporters that I showed, I would just say that SLC26A6 is not part of Invitae's panel for some reason, and so I did this demonstration on that.
Dr. Navin. Okay. Thanks for showing the first picture, the first slide. That's our figure. So you saw that it's one of the kidney sections kind of thing, and most of that. Your organoid contain more proximal tubular cells and everything. Is there anything, the collecting duct cells there, or the interstitial cells, the immune cells also there in the
there aren't interstitial cells. There are connecting tubules. So there's aquaporin 2 positive cells. The interesting thing for kidney organoid work is the adult mammalian kidney forms from the fusion between the metanephric mesenchyme, the metanephros, and the ureteric bud, which is a derivative of the Wolffian duct. And so these two different tissues, develop, differently in space and time. And so they require independent differentiation protocols to generate each one separately and then do co-culture. Most of our work that I had shown, that's why I had focused on an organoid being resembling an organ. And if you want to resemble a kidney, you'd really want nephron structures. And that's why I showed the directed differentiation to metanephros. But there are some separate protocols. There's about 5 well-published towards ureteric bud organoids, which, it really is different patterning along the primitive streak. But I can discuss that at length after this, if you'd like.
All right. The question from Navin, Dr. Navin. Yeah. So then for him, the Hatem then it says the secreting the protein is a soluble protein because the bacteria itself, it's also secreting the vesicles. It's carrying that all the molecules and even protein, RNA and DNA too. So the protein, what you are doing, that is SL1 or something, the soluble protein or is the inside the vesicle?
So they are secreted protein. All of them, they have a secreted peptide signal.
Peptide signal, individual.
They are
So our next speaker is Matias Simons, who comes to us from molecular human genetics at Universität Heidelberg.
My lab is using Drosophila as one of the models to study kidney disease, and therefore I want to start out by just pointing out some advantages of using Drosophila as a model organism. So it's a cheap, relatively cheap model organism that has a short generation time, 10 days, as you can see here. And there's an excellent genome annotation, as there's been fly research going on for more than 100 years. There's low genetic redundancy. This is important for functional studies. And there's, of course, vast genetic and genomic resources. And traditionally, Drosophila is good for screening approaches. So there is a renal system. I know that Michael Romero is studying the Malpighian tubules, which are shown here in blue, but we are more focusing on the nephrocyte that come in two populations. The Garland nephrocytes shown here, the green cells, and then the pericardial nephrocytes. So these cells, they harbor features of podocytes and proximal tubules in one cell. Podocytes because there is a slit diaphragm at the outermost edge of the cell surface that seal these invaginations that you can see here. And inside these invaginations, you also have expression of cubolin and amnionless, which are known transporters, protein uptake transporters or protein uptake receptors from the proximal tubule. With these cells, we are modeling a number of proximal tubular disease mostly, and today I want to share some data we have on the Dent's disease. To this audience, I don't have to say much about Dent's disease. As you know, it's a problem of CLC5 and OCRL1, and for CLC5, it's known that it is a proton chloride exchanger in the endosomes. And there's maybe not so much knowledge about this, but there are some papers that show that there's also some glomerular phenotypes in Dent's disease. And that's what I want to point out for the sake of this talk. So in flies, we have a number of CLC, so chloride proton exchangers, but less so than in humans. We only have 3, A, B, and C. And CLCC is the one that is closest to the CLC5. And indeed, when we knock down both, so all 3 CLCs, we see that the CLCC is the only one that causes reduction of cubolin on the cell surface. And this is really also a hallmark of the Dent's disease, that the protein uptake receptors, megalin and cubolin, are missing, and that's why you have low molecular weight proteinuria. So when you study functionally the uptake of proteins by using albumin trits, you can see that, again, CLCC is the only one that causes the reduction of protein uptake. And in addition, we also have some defects related to the slit diaphragm. So normally you see this straight line on the outer edge of the cell surface for one of the slit diaphragm proteins, which is here Kira, related to NEF1. And in the CLC knockout, you see these that the Kira is actually found in these invaginations, not as in the control. And this is then reflected also by the EM, where you can see that in these invaginations, you also have ectopic slit diaphragms. So it seems that nephrocytes are actually a good model to study Dent's disease 1. We find both proximal tubular phenotypes and glomerular phenotypes. We did not find a phenotype for the Dent's disease 2 gene OCRL. So to study what these phenotypes are about, we turn to the endolysosomal systems. And in nephrocytes, there is a well-patterned endolysosomal system. We have at the outer, so in the periphery, we have the early endosome as a distinct population, and then more to the cell interior, we have the late endosomes. And markers for those organelles are rhabinosin 5 and Rab7. So this is the control, but when you knock down CLCC, you see that the early endosomes are shifted more to the interior, and this is more dramatic for the Rab7, where you really find a dramatic accumulation of Rab7 positive organelles close to the nuclei. Rab11 is a recycling endosomes, and typically that the hypothesis has been that CLC5 is involved in recycling, and indeed the Rab11 marker for the recycling endosomes is kind of reduced in the CLCC knockdown cells, and also the knockdown of Rab11 causes a similar phenotype as the CLCC, which with regard to the Rab7 distribution. So it seems that we have some phenotypic overlap between CLCC and the recycling endosomal Rab11. And EM then also confirms these nuclear or perinuclear accumulations of probably Rab7 dependent or Rab7 associated vesicles. Another phenotype that we saw was that there is actually a fragmented ER in the CLCC knockdown, and you can also get ER stress markers lighting up here, XPP1 GFP. So there's something going on with the ER, and indeed the cubulin seems to be trapped in these fragmented endoplasmic reticular structures. So this could also account for the loss of cubulin at the surface. Rab11 knockdown again seems to share this phenotype. So to conclude, we have knockdown of CLCC causing a broad zone of early endosomes and this perinuclear clustering of late endosomes and lysosomes, and cubulin appears to accumulate in a fragment in a stressed ER, and some of the phenotypes are shared by the Rab11 knockdown. So it seems that CLCC deficiency is some kind of recycling defect. We also found that actin is increased at the cortex of the nephrocyte in the CLCC knockdown. This was a quite strong phenotype, and this reminded us, when you then co-overlay the rhabinosin 5, the early endosomes, then you can see that there are these holes in the early endosomal populations that are filled by the cortical actin. So it seems that this actin belt is somehow pushing the early endosomes to the interior. So this reminded us a little bit about the papers that have been coming out on OCRL1, where actin also seems to be misorganized. And there's an old paper that I would like to also point out, that CLC5 interacts with cofilin, which is an actin depolymerizing factor. And cofilin is regulated by a kinase called Lim kinase. So when Lim kinase phosphorylates cofilin, then the actin depolymerizing function is decreased. So we try to play with the cofilin itself, which is called twin star TSR. There's a constitutive active form, and we can indeed suppress the actin accumulation that you get with the CLCC knockdown. And then when we knock down the Lim kinase, we get the same phenotype. This again would also lead to an activation, a constitutive activation of cofilin. We get a partial rescue of the albumin uptake defect when we have the TSR constitutive active and Lim kinase RNAi co-expressed with the CLCC. So the actin somehow seems to be important for the uptake of proteins. So this is to summarize. Tubulin seems to be lost at the cortex of the nephrocytes that lack the CLCC homologue of CLC5, and tubulin is also accumulating in a fragmented ER. We also get this actin accumulation at the cortex that somehow seems to broaden the early endosomal population. And then we get a very strong late endosomal lysosomal phenotype. They seem to accumulate in the vicinity of the two nuclei. And I didn't have time to show this, but there's also an accumulation of PI3P and cholesterol in these mislocalized late endosomal lysosomal compartments. And there's a similarity with Rab11 knockdown phenotype suggesting that CLCC is really involved in endosomal recycling. So with this, I want to stop and just thank the people involved. Salomon and Indira are the current lab members that work on this project, but there were some others working on this project before, and I'm happy to take further questions. Thank you.
Awesome. Thank you, Matias, for a very enlightening talk about flies. Anybody here or in the chat that has questions for Dr. Simon? Yes.
Yeah, so structurally, the Malpighian tubes and the fruit flies, are they, I don't know a whole lot about fruit fly biology, but are they straight tubules, or do they have that similar kind of like hairpin structure of like the loop of Henle and human nephrons?
So I guess Michael Romero is the person to ask this question. We don't really work on the Malpighian tubules. Nephrocytes is part of the renal system, but it's not linked to the excretory Malpighian tubules.
Yeah, just a quick input. So the Malpighian tubules in flies are blind-ended tubules. They're a single layer of epithelium, and they're sort of a combination of proximal tubule and maybe collecting duct in the way they look. They have very elongated microvilli, but I mean, we can talk offline about some of the other features, but they don't do anything like the loop of Henle.
All right, the reason why I asked that is because I suspect, with stone formation, that hairpin structure is probably pretty important physiologically, because I feel like, like the mineral components or other things that make up the stone matrix, or these for kidney stones, probably get caught up in that hairpin structure. So I think that's probably an important part of the nephron. And so I'm just wondering if it was replicated in the fly as well.
So again, I mean, that's not really what Matias talked about, but the simple answer is yes. Actually, Carmen Reynolds in my lab has done those experiments in the Malpighian tubule, and we can talk offline about that.
Yeah.
Are there other questions here or online? Yes, Jaya.
Yeah, so there's interesting talk. The suppling of the vesicles and the endosomes and go to that endoplasmic reticulum. Is it alter the crystal formation, or is the vesicle has the phosphatidylserine, negatively charged phospholipid? It helps for nucleation of the crystal formation.
So the question
of stones. I mean, I think the nucleation of stones is extracellular. So what I'm showing here is intracellular defects that are probably upstream of the stone formation. I think the Dent disease is caused by a defect in proximal tubular homeostasis, because what you get is this disruption of the endolysosomal system. But secondarily to this, then there is an alteration of ions outside the cell, that calcium, for example, that is controlled by PTH, and PTH receptor is also a receptor that resides on the proximal tubule and is probably affected by the endolysosomal pathway that is disrupted in the CLC5 mutant cells. So I think you have to really distinguish between what happens inside the proximal tubules and the downstream effects related to stone formation.
If I could, let me ask a quick question. Have you, so this is just really awesome work. have you looked at the fluid that's secreted from the flies where you've manipulated CLCC in the nephrocytes to see if there's altered protein content in the fluid that comes out of the flies, or if there's anything resembling crystals of any sort?
No, we haven't. So I think, yeah. I mean, the nephrocytes, they don't really modify the content of the excretory tubules, but indirectly they could modify the hemolymph composition through I mean, they could first modify the hemolymph composition due to the altered uptake function, and this could have a secondary effect on the content of the lumen of the tubules, you know, and this is all I can say about this.
Yeah,
the nephrocytes are not connected to. Yeah, we didn't study this.
All right. Well, thank you very much, Matias. I appreciate your presentation, especially given your computer problems, and have a good rest of your meeting there in Germany.
Thank you very much, and have a nice day at the Stone Conference.
So our next speaker is Dr. Gerard Quintana from the Vallhebron University in Barcelona. He is working in Anna Meseguer's group, and he's going to talk to us about novel systems for studying Dent disease. Welcome, Dr. Quintana.
Thank you a lot. Good morning, everybody. First of all, it's really a pleasure to assist to this meeting, and I would like to thank the organizers for inviting us to present our work in front of this wonderful audience. As he introduced me, I'm Gerard Cantero, principal investigator of the renal physiopathology group here at Vallhebron Research Institute in Barcelona, which is led by Dr. Meseguer. The main interest of our group are to understand and study the molecular and cellular mechanisms underlying the pathophysiology of different renal diseases. And today, what I will present here are our latest results on Dent's disease that was beautifully introduced before by Dr. Simons. I will present the cellular models that we have generated together with the results that we obtained with these cellular models, and if I have enough time, I will try to show you our preliminary data on biomarkers and therapeutic targets. Just to start, I don't have to explain much about this because you are really experts on the kidney, the renal system, and also the nephrons. So let me just take a moment to show what's about this proximal tubule and the renal proximal tubule epithelial cells. As you can see in this image, these cells, as you already know, they are in charge of the reuptake of small molecules, low molecular weight proteins, and also water. This is because these cells have a very complex and well-specialized endolysosomal system, and this allowed them to endocyte all these proteins and molecules. A key element of this system is the CLC5, that you can see here in the drawing, and this CLC5 is, and in the drawing is in yellow, sorry.
Can you share your, can you go into presentation mode, because we're only seeing the overview mode of, on your, there you go.
Yeah, perfect. Now it's, wait a second then, there was a problem here. Because if I do like that, you don't see, wait. A second, sorry. Now it's not working. Okay. Yeah, wait a second. I will share again because I think I'm sharing the secondary screen. Share this one now. Perfect. It's
yeah.
Okay. The second one. Thank you very much. Sorry. So here you have the CLC5. This is the chloride proton antiporter that is localized at the apical endosomes and the plasma membrane. And there, the main function of this antiporter is to regulate the endosomal pH. When there are mutations in this gene that produce loss of function of this protein, there are problems in the endocytosis, obviously on the pH of these endosomes, and this will affect the function of the proximal tubule. Since the proximal tubule is not working correctly, all these proteins that were supposed to be taken by these cells are lost through the urine. This was described, this disease, many years ago by Dr. Dent, and this is the Dent's disease type 1. And the main hallmark of the disease, accordingly, is the low molecular weight proteinuria. There are other characteristics like the hypercalciuria, nephrolithiasis, etc. And what's also interesting, and I will talk more about this later, is that several patients will also develop renal fibrosis, and the patients also progress to chronic kidney disease and kidney failure with age. There is no treatment for this disease, and there is a lot of variability. This paper, we published it last year, that it's on the characteristics of Dent disease in Europe. And here we also show that the clinical and the genetic characteristics of Dent disease in Europe is similar to the rest of the world. What is interesting here, and I want also to present, because was maybe one of the first times that we show that the mutations accumulate in specific domains of the CLC5. And this could correlate somehow to the phenotype of the disease that was later also showed by Dr. Lieske here. So the question that we want to solve in our lab is, what are the cellular mechanisms that link CLC5 with this proximal tubule dysfunction? Because our last goal at the end is to identify reliable progression biomarkers and therapeutic targets for this disease. To achieve this, what we did first, and it's what I will explain you here, and it was published 2 years ago in the Human Molecular Genetics Journal, is that we developed these cellular models of Dent disease. For this, we use these RPTEC cells that are the renal proximal tubule epithelial cell lines. These are very good cells to study the proximal tubule because they maintain many of the characteristics of the renal tubule, proximal tubule, like the polarization and also a good endocytic system. In these cells, what we did first was to knock down the endogenous CLC5 and then overexpress on top of them the wild-type form of CLC5 or different mutated forms, as you can see in the Western blot and the qPCR. Next, what we did was to assess if these proteins that were nicely expressed in these cells were localized where they should be expressed. And what we saw here is that the wild-type form, as expected, reaches the plasma membrane and also the endosomes, but the rest of the mutants are retained in different compartments of the cell. This is a really interesting question for me because although these different mutations are really close in terms of the domain where they are are retained in different compartments. For example, the I524 is retained at the ER and the others can reach to the endosomes, or the one that is the valin seems to be retained at the Golgi. We can talk more about this later because I think it's really an interesting question. Next, after assessing that the wild-type form and also the mutated forms are nicely expressed in these cells, what we needed to test, if we wanted to use these cell lines as models for the disease, is whether they reproduce the phenotype of the disease. And as I told you before, the main hallmark of the disease is this low molecular weight proteinuria. That is because there is a lack and impaired endocytosis. And what we did here was to test how the endocytosis was behaving in these cells. We treat these cells with albumin, and then we measure the number of albumin dots that we had in these cells. As you can see in the image, only the control and the wild-type cells really endocyted the albumin, while this was impaired in the mutants and also in the knockdown cells. After, sorry, I have to move this. after really characterizing the cell lines, what we wanted to do next was to assess which cellular processes were different between our cell line, our knockdown and mutated cell lines, and the control cell lines. For that, what we did was the global gene expression analysis. And as you can see here, we analyzed all the genes. There were several genes that were up or down regulated when we compared the wild type forms or the mutated and knockdown forms. And what was really interesting here is that many of these processes were related to cell differentiation and epithelial to mesenchymal transition. When we studied this in more detail, what we saw is that several of these epithelial markers were downregulated in the mutated or in the knockdown cell lines, and also other parameters, other features related to the differentiation were also increased in our loss of function cell lines, like cell adhesion or cell proliferation. In global, what we concluded from here is that CLC5 loss of function, what it promotes is this transcriptional switch to cell differentiation pathways. After characterizing these cell lines, these cell models, and understanding the cell processes that are altered in these cells, what we wanted to do is to study another process, another feature of this disease, that is the renal fibrosis, not only because it's important in Dent disease, but because it can be also relevant for other renal pathologies. Here, the results that I will present you now are currently posted as a preprint at bioRxives. And what we did here was to study the collagen levels in our cell lines. As you can see here, I'm showing here just the extreme phenotypes, the control versus the knockdown phenotype. And you can already see that these cell lines, they have a massive accumulation of collagens compared to the control cells. This is similar to what happens in some patients that will start developing renal fibrosis. But here the question still remains, what is the relation between CLC5 and this production and release of collagens? Well, spoiler alert, we already demonstrated that there are two processes that are altered in these cells. On one hand, the loss of CLC5 causes an impairment in collagen's degradations. As you can see here in the Western blot, half-life of collagen is really extended in the knockdown cell line. And what we saw, it's really interesting, it was quite surprising, is that collagens in our RPTEC cells are degraded through lysosomes and not proteasome. As you can see in the Western blot, when we use inhibitors of the proteasome, this MG132, there is no accumulation of collagens, but when we treat these cells with lysosomal inhibitors, we have a massive accumulation of collagens. Then what we did was to study how these lysosomes behave in our knockdown cells. As you can see here in the figure, these lysosomes, they have a more basic pH compared to the control cells, and also these lysosomes are larger. So in conclusion, what we have from here is that CLC5 depletion impairs lysosomal acidification, and also all this together produces this enlargement of the lysosome because the collagens cannot be degraded, and this leads to the accumulation that we see in these cells. But this is not enough to explain the massive accumulation that we see in our cells. So what we also saw is that the transcription of collagens is also increased in our cell lines, in the knockdown cell lines. And this is through the beta-catenin pathway. As you can see in the image, in control cells, most of the beta-catenin is retained at the plasma membrane, while in the CLCN5 knockdown cells, this beta-catenin can be translocated to the nucleus, where it can promote the transcription of collagens. A key experiment here is when we used inhibitors of this translocation to the nucleus, that is this FH535, and you can see that after treating cells with this inhibitor, this accumulation of collagens in the knockdown cell line is totally reverted. So CLC5 deletion also promotes beta-catenin translocation, which will enhance collagen 1 and 4 transcription. But how is CLC5 promoting this translocation of beta-catenin to the nucleus? So after reading the literature, we found that many epithelial markers, epithelial proteins that are located at the plasma membrane can interact with the beta-catenin, and especially one that is really interesting for us is MAC1 that can sequester this beta-catenin at the plasma membrane. So what we did next was to study how MAC1 behaves in our cell lines, in the knockdown cell line especially, and what we saw is that the levels of MAC1 are strongly reduced in the knockdown cell line, as you can see in the image and also in the Western blot. And just to finish this part, all that I explained you with the knockdown cell lines, we also saw with the mutant cell lines. So we can say that the CLC5 loss of function can promote accumulation of collagens due to impairment in the degradation and increase in the transcription. And it was also at the end satisfying to reproduce the same results in we have a collaboration with Dr. Baisang Lu, and we use the CLCN5 knockout mice. And here, as you can see in the image, the renal tissue from these mice, they also show an increase in the collagen levels and also present a fibrotic phenotype. Altogether, we have this working model where we postulate that in control cells, we have these epithelial proteins, the MAC1, but also E-cadherin that can sequester beta-catenin at the plasma membrane, and this prevents the translocation. Then the transcription levels of collagens are maintained at a very low level, and this permits that the homeostasis of the ECM is maintained at normal levels. In the case of DD1 cells, there is a defect in the acidification of the endosomes and also of the lysosomes. This affects the MAC1 and E-cadherin location at the plasma membrane, and eventually it will promote their degradation. This releases beta-catenin from the plasma membrane that will translocate to the nucleus, permitting the higher transcription of collagens that at the end they will accumulate because they cannot be degraded and will be also released to the medium, therefore maybe explaining the renal fibrosis phenotype in some DD1 patients. And now, if I have a little bit, maybe 1 minute more, I would like to explain our ongoing research in the D1.
Maybe,
I have one.
If you can maybe wrap this up in about maybe a half a minute, otherwise there's not really going to be time for any questions.
Okay, I will go fast. Then what we have done is to perform an interactome analysis, and we identified new interactors of CLC5. We have also tested in DD1 urine samples the exosomes. We have isolated exosomes from urine samples from our DD1 cohort. And from these exosomes, we are now isolating the miRNAs because we want to obtain putative biomarkers and maybe therapeutic targets. And finally, this is something that we are also doing right now, that we are starting to identify some therapeutic drugs for DD1, because combining the data from our DD1 models and the literature, we have obtained a list of therapeutic targets and predicted compounds. And here, very, very briefly, what we have done is using MAC1 and collagen 1 secreted levels as readouts. Now we are using our DD1 models to assess the effect of different compounds. And we have already some compounds that can reduce the levels of collagen in our DD1 cell models. And with all of this, it's just here the summary of what I have shown you, that then we can go through them. And I would like to finish just thanking all the renal physiopathology group, especially Dr. Meseguer, that is also here, and Dr. Duran, that is the postdoc in charge of the project, all our collaborators, of course, and the funding bodies. Thank you very much for your attention, and I will be happy to answer any question that you have.
Thank you very much, Dr. Knauf. If I could ask just a quick question. So it seems that you see both a increase in transcription of collagens as well as a decrease in the degradation of the collagens. And of course, the degradation is probably more directly related to the Dent disease. I guess my question is really, why do you think there's an activation of the transcription, and how might that be changing the overall renal function?
So what I think it's because at the end, it's true that it's affecting the pH of the endosomes and the lysosomes. This is the main effect. And by affecting this pH, it's affecting the recycling of MAG1. And by affecting this recycling, the MAG1 levels of the plasma membrane are really dependent on this recycling. Then this will lead to the degradation of this MAG1 protein. We don't know if it's which system is the degradation of MAG1 happening. And this is what will produce this release of beta-catenin and this EMT transition. And this at the end related to the differentiation of the epithelial cells and will lead to this fibrotic tissue.
Thank you very much for your enlightening talk. Our next speaker is one of our more recent recruits here to nephrology at Mayo, Dr. Hatim Hassan, and he's going to tell us about understanding and manipulating gut oxalate transport.
Good morning. I would like to thank the organizer for the opportunity to speak about understanding and manipulating gut oxalate. So, works pioneered by Dr. Marguerite Hash at the University of Chicago and Peter Aronson at Yale, shown that the mammalian intestine play a crucial role in oxalate homeostasis, where intestinal oxalate secretion is active through the transcellular pathway. This is a model for the duodenum. Intestinal oxalate secretion is active through the transcellular pathway, and is where this oxalate transporter SLC26A6, I'm going to be calling it A6 during my presentation, play a major role in intestinal oxalate secretion, where oxalate enters the blood from the lumen blood side and then gets secreted from the enteroside into the lumen by this A6 in exchange for intraluminal chloride. And the knockdown, when this mouse, the A6 is knocked down, the mice develop hyperoxalemia, hyperoxaluria, and a high incidence of calcium oxalate kidney stones. And when they did the study in Wissing chamber, there is oxalate secretion in the duodenum and ileum is greatly reduced. The nature of the transporter mediating oxalate influx into the basolateral side remains unknown, probably with A1 SLC26 playing a role in maybe in the ileum and colon. The A1 null mice, Dr. Dawson back in 2010 showed that they develop hyperoxalemia and hyperoxaluria and kidney stone on a mixed background. The Marguerite Hash, when they try these mice in a C57 black mice, they don't have hyperoxaluria or hyperoxalemia. So that the nature of the basolateral transporter still remain unknown. And then while absorption, intestinal oxalate absorption is mainly passive through the paracellular pathway, driven by the prevailing electrochemical gradient, higher luminal oxalate and lumen negativity as a result of sodium reabsorption. And most of the oxalate gets absorbed, gets secreted back through this transporter, therefore maintaining normal plasma and urine oxalate. There is an active component of oxalate absorption mediated by the SLC26A3, and these null mice, the A3 null mice, they have significantly lower urine oxalate by about 70%. This active component in our Hanna and Marguerite Hash is about maybe 20 to 35%, but most of the absorption is passive through the paracellular pathway. So given the important role of this transporter in oxalate homeostasis, so defining the molecular mechanism regulating them is important. So if we find a mechanism to stimulate intestinal oxalate secretion and then get rid of that oxalate in the feces, that can help, or inhibit an absorptive process that can help reduce plasma and urine oxalate and therefore prevent a kidney stone. So with that, my lab has found that the PKC pathway is a major negative regulator of oxalate transport by A6, including the purinergic signaling, where ATP and UTP bind through the P2Y2 receptor, phospholipase CPKC delta, inhibit oxalate transport in vitro in a Keiko cell line by reducing A6 surface expression. Similarly, adenosinergic signaling also does the same through PLC and PKC and reduce A6 surface expression. And also leptin signaling through the leptin receptor also reduce oxalate transport, both active transport and it stimulate passive transport. And these pathway might have potential pathophysiological relevant to the IBD-associated hyperoxaluria. So the IBD-associated hyper is mainly blamed for fat malabsorption, where malabsorbed fatty acid complex with calcium-leaving oxalate. But patients with ulcerative colitis, some of them, they have hyperoxaluria and they don't have fat malabsorption. So patients with IBD, they have significant amount of ATP and UTP in the luminal side. They have a lot of adenosine, and they have significant amount of leptin both in the lumen and blood side. So this regulation might have some relevance to the IBD. We also show that cholinergic signaling also goes through a similar pathway and the muscarinic receptor M3 to inhibit oxalate. And this pathway might have a relevant to obesity-associated hyperoxaluria, where obese patients have increased parasympathetic tone and more acetylcholine. Inflammation also regulates oxalate transport in the gut. We have shown that pro-inflammatory cytokine TNF-alpha, interferon gamma, and IL-6 significantly inhibit active oxalate transport by the Keiko cells. This transport is mainly mediated by the A6 transporter using siRNA studies. And this is by reducing A6 mRNA and protein expression. And also giving TNF-alpha to mice for 2 days, and then get the jejunum and put it in oozing chamber and assess active oxalate absorptive fluxes. This is the JM2S, adding oxalate on the luminal side and sample from the serodal side. That's the absorptive flux. Or adding oxalate on the serodal side and sample from the luminal side, that's JS2M, that's the secretory flux. So there is no effect on active absorption, significant reduction in the active secretion that led to conversion of net oxalate transport from net secretion in control tissue to net absorption into the TNF-treated tissue. So in addition to the effect on active transport, also TNF-alpha and interferon gamma, this is using the loop model. This is the jejunum and ileum, to creating a loop, fill it with C14 and tritium labeled mannitol. Mannitol is a marker of a paracellular pathway. And then I set the permeability coefficient. You can see that there is more than 2 or 3-fold increase of absorption of oxalate. And here there are about the same, the permeability coefficient, indicating that both oxalate and mannitol are going through the same paracellular pathway. So this regulation could have pathophysiological relevance also for the IBD-associated hyperoxaluria, where there is a lot of luminal inflammation, a lot of TNF-alpha interferon gamma, and also the obesity-associated hyperoxaluria. We show that in the OB mice, they have significant luminal and also systemic inflammation. So oxalobacter play a major role in oxalate homeostasis, and the Margaret Hash group, they show that in addition to degrading intraluminal oxalate, this bacteria interact with colon cells, probably as a survival strategy when oxalate is limited, because it totally depends on oxalate. So, but the nature of these bacteria secrete factors when probably there are stress with lower oxalate. The nature of those factors remain unknown in my lab. Became interested in this, and we were able, we grow the bacteria in a liquid culture and then obtain the conditioned medium, which only contain what is secreted by the factors. The liquid culture pass through a filter, no cells. And you can see here, we see like a 2 to 3-fold stimulation with the conditioned medium, while the regular growth medium has no effect. And that's in vitro. In vivo, when we treat mice, a mouse model of primary hyperoxaluria type 1, which has 3-fold hyperoxaluria, after 3 weeks, we see a significant reduction in urine oxalate by more than 32%. And we hypothesize that this is by stimulating colon secretion. We obtain the distal colon at the end, and we assess the absorption and the secretory flux. We see more than a 40% increase secretion, and that led to more than 4-fold net secretion. So these factors, they are doing what the bacteria is doing, given the current difficulty in maintaining colonization on Oxera, failed multiple trials using this bacteria alive probiotics. So these factors, they are doing what exactly the bacteria is doing. Using mass spec, we identify this cell wall-like protein and peptide as the major secreted factors. We tested about 12 out of more than 40. It's just amazing to me why the bacteria need more than 40 protein from a single gene family. They have to be really important for this bacterium. And two of them, like all of them stimulated, but OXB14 and 2 has the maximum stimulation. We created the crystal structure for 6 proteins. And this is a homologous structure for the OXB cell 14. And the whole protein is formed with these repeated units. They call SLR domain, about 35 amino acid. And these SLR domain are important in protein-protein interaction and cell signaling. And we hypothesize these domains are mediating the stimulation. And we probably identified a potential binding site. I don't have time to talk about it. So we tested 5 of these peptides. All of them individually stimulate oxalate transport by 30 to 50%. When we do different combinations, this P8 and P9 give the best stimulation almost close to the conditioned medium, indicating that these are the major factors. And here, these were working with the Keiko cells, which closely resemble the native epithelium, but these are cancer cells. Human organoids emerge as like ex vivo models, which fully mimic the in vivo behavior. And we develop oxalate transport assay in these cells. And these P8 and P9 significantly stimulate by 1.7 to 2-fold distal colon, sigmoid colon, and ileum, confirming that they work in human tissues, and most likely they might work in humans in vivo. So just to summarize this part here, these cell-like protein and peptides, where we bind through a cell surface receptor, we are working on identifying, activate PKA. We found that direct PK activation also stimulates oxalate transport in the Keiko cells through increased expression of A6. And this is through, we knock down A6 and A2, and most of the stimulation are going through them to stimulate oxalate transport. So since these factors are able to retain their biological activity in vivo, they have significant potential, therapeutic potential for hyperoxaluria and hyperoxalemia. So proposing a model that OF, Oxalobacter, potentially serve as an oxalate sensor. It degrades oxalate dietary. If you are not eating enough oxalate, they secrete factors which induce colonic oxalate secretion. So this is a prime example of a host microbe. Host microbe interaction, where the microbe changes its ecosystem to benefit both host and microbe by secreting factors stimulating intestinal oxalate secretion. And we hypothesize this Oxalobacter potentially serve as an oxalate sensor. So given their potential to therapeutic potential, I formed a startup company, and we are working on preclinical studies. The problem with these peptides, they have a lot of cleavage sites for proteolytic enzyme, and we did an LCMS in vitro stability assay and just treat them with trypsin or proteinase kinase or colonic lavage fluid, which has what's secreted by the colon cells and bacteria. They just rapidly degrade them. So we're working on 2 ways, doing optimal structural modification to make them stable. But we are facing with the difficulty. Many of the structural modification are rendering the peptide either completely non-functional or significantly non-functional. So one of them is a retroinverso. So we went through another pathway here, developing like a novel delivery system composed of nanoparticles. And these nanoparticles they are hoping maybe they encase the peptide and protect them from degradation, and also deliver them to the colon mucosa in a localized way, similar to Oxalobacter. In pilot proof of principle, we developed P9 hydrogen nanoparticles, and they significantly reduce urine oxalate in this primary hyperoxaluria mouse. Currently, we're developing a couple of formulations, including chitosan, to try an oral formulation by Gavage. Recently, we're very excited about discovering couple of drugs. I can't talk about the nature of those drugs because of an intellectual profiling, and they stimulate oxalate transport significantly by 70 to more than 2-fold. I'm just showing one of them, but other of them are more active than this, and we're very excited about these drugs. So another way here, I told you the A3 is involved in active oxalate absorption. In a group in California, they found a small molecule inhibitor for this SLC26A3 as a potential therapeutic inhibiting chloride bicarb exchange for treatment of constipation. And they found that it also inhibit chloride oxalate exchange. And when they assess this colonic absorption using a distal loop, closed loop, and they fill it and they introduce like 500 nanomolar of sodium oxalate. And these inhibitors significantly reduce the oxalate transport. They are claiming this active. To me, this is here, oxalate moving from the loop inside the bath, mainly probably maybe there is a passive transport rather than active, but they are showing here and there is significant reduction in urine oxalate. And here they show that they significantly reduce calcium oxalate deposition and also renal injury.
So hello, everyone. I'll be moderating the next session on inflammation and stones. I'm Lama Nazzal. I'm a nephrologist at NYU, and I study the gut microbiome and kidney stones with a focus on oxalate metabolism. So we're going to hear the first talk today from Dr. Muthuvel Jayachandran on macrophages and urinary stone disease in mice and human.
Thanks. Good morning to everyone. There are two types of macrophages there, and all the tissues and also the bloods. That basically macrophages are innate immune cells that's involved in phagocytosis, basically secreting by antimicrobial proteins. Then are also involved in a lot of that dead cells in the body. It's cleared by the macrophages, basically that derived from their monocytes in the residential monocytes and also circulating blood. It's two ways get activated. One is classically activated monocytes by that interbranchioma and the granulocyte monocyte colony stimulating factors. It's converted into M1 macrophages. These are more pro-inflammatory. It's decreased the phagocytic capability and also increased pro-inflammatory cytokines and chemokines. The alternatively activated, another type of macrophages we're calling as M2 macrophages. These are anti-inflammatory. That's increased the phagocytosis and anti-inflammatory cytokines and chemokines. Thereby, it's clearing up that any dead cells and also unwanted foreign material in the body, including crystals, calcium oxalate, calcium phosphate crystals. So another cells in the present in the tissues, then they get renal tissues that we're calling as they originated from their embryonic origin or it comes from the bone marrow. It's also involved in M0 macrophages. Macrophage itself resides in the kidney tissues. It's also get activated. That depends upon the stimulus. It goes to the M1 or M2 macrophages is to the similar function as previously I said. So there are four types of M2 macrophages that depends upon the markers present on the surface and also under their functions. That one is that M2A macrophage and has endocytic activity and cell growth and tissue repair. And M2B macrophage promotes that T helper cell to differentiation and involved in bacterial fungal infections. And M2C macrophage involved in phagocytosis of apoptotic cells. M2D involves mostly pro-angiogenic activity, thereby it's promote the tumor growth progression. So most of this, you can see that all the three types of that M2 macrophages secretes that major cytokines at interleukin-10. These are the anti-inflammatory cytokines. These may be involved sometimes pro and anti-inflammatory capabilities in M2A. So they're based on our previous study. These are the representative slides. We stained with kidney tissues from nephrectomy patients with and without the history of urinary stone disease. We're calling it non-stone formers. They never had any stones. Then stone formers, they had the kidney stone in previous years. So then you can see that these are the M1 macrophage. We used one of the markers for a CD68 staining. That the inserts are the large magnifications in the cortex and medulla. These are the stone formers, non-stone formers for M2 macrophages.
Then we validation of that previous studies, and we found that you can see that number of the macrophage population significantly increase in the medullary region. This is the area for stone formation, stone nucleation. There's no big difference in that M1 pro-inflammatory macrophage in the cortex area. So there was no difference in the cortical and medullary M2 macrophages. The marker we used for CD206 is only combined for, please keep in mind that the 4 types are there. There's the markers involved in only 2 types, and another 2 types we do not know that. In the future studies, we are going to characterize that. So we checked that calcification by Yasu staining. Then you can see that the calcification was significantly greater in the medullary regions. There's no much significant difference in the cortex. So it's correlate very well with increased number of pro-inflammatory macrophages, and also that increase the calcification in the stones. Then based on the previous studies, the recent studies from Kansas City, Dr. Wu's group, they did the experimental animal model. They knocked down the claudin-2 protein, and then the mouses are more hypercalciuric. They found the calcium oxalate, calcium phosphate crystals within the tubules, and also some of in the interstitium. So then we take the advantage of that. So we hypothesize that the claudin-2 protein is the 230 amino acid protein, transmembrane protein. Is basically involved in transport of paracellular transport of sodium and calcium. When you low expression, that knocking down, it's completely blocked the calcium and paracellular transport. Thereby, it's increasing that distal delivery of more calcium. This delivery calcium may lead to that renal block or tactile block formation, thereby it's increasing the urinary stone disease. But we don't know the mechanisms, so how it changes. We are proposing that maybe the calcium and phosphate leaking here, that descending limb and other S3 segments mostly involved on that stone pathogenesis. So then we want to know how that expression changed in the normal, because they did the experimental animal models. We don't know about the human tissues. We use this human nephrectomy kidney tissues. So we found that the expression of the claudin-2 protein, the proximal tubule, and thin descending limb, that the expression was in the median was, even though the data is overlapping, the number, the median was significantly decreased in both cortex and medullary regions. So we got more number of tissue section now, more than 250 sections. So we are going to stain more, that increase the number to validate this data. So then we got that mouse from Kansas City, where founder mouse to make the colonies of that claudin-2 constitute global knockout mouse. So we found that the baseline character, we established the colonies now. We have pretty good number of mouse with the claudin-2 knockout and the wild type mouse. So we did the urinary chemistry, urine chemistries using that. You can see that there's no significant difference in the age and body mass index other than the urine calcium excretion. These are expected. The urine calcium was significantly greater, and the creatinine is a little lower in that claudin-2 knockout mouse. There is a minimal change in the magnesium, but there's no change in other urinary parameters. So then we did that micro-CD, micro-CD using in vivo micro-CD, one of the knockout mouse. The younger mouse, youngest one, 3 months old, we got some mouse, we had the crystals on the kidneys. You can see that clearly crystals by micro-CD. These are in vivo. The same mouse, we take out the kidney, then we did the ex vivo micro-CD. You can see the nice crystal formation in the papillary regions in medulla. Then these are all the founder mouse. Then we got the crystals. You can see by the ex in vivo micro-CD and also the ex vivo micro-CD, one of the kidneys there. Then we did the histology of that aged, histology of the aged mouse. These are lower magnification, is higher magnification. You can see these are the crystals is involved in, these are papillary regions. You can see very nice crystals within the tubules based on that renal pathologist, Dr. Ferrero. So these are Yasu staining. Then you can see there are a lot of crystals inside the tubules and a little bit in the interstitium also, same as what we found that other studies. So these are higher magnification. You can see that crystals there. So right now we have established the model. Now we can work on the more mechanistic studies. So interestingly, we found some studies, even though the mouse are knockout, the urine calcium, the 24 hours calcium correlating the identifying the crystals in the kidney in in vivo micro CT. So some of the mouse then is 6 months old and also 13 months old. We didn't see any of the crystals in this mouse. Then we check that urine calcium for that mouse. These are less than 3 milligram per dl. So significantly nothing. We didn't find any kind of crystals on this mouse, but even the 3 months old, they excrete more calcium. We find the nice crystals in the kidneys papillary regions. So then these are the individual data of that wild type and claudin 2 knockout mouse. These are the 5 of this mouse. We didn't find any crystals on the kidney for ex vivo in vivo micro CT. So and you can see their ages are different. 13 months, 2 of them, others are 6 and 4 and 4 months. These are the mouse who over the high calcium excretion, they have the kidney crystals on the kidneys. So now we have the model that we are trying to do right now. We are studying 3 different group of 2 groups of patient population. One of the experimental animal models, we are using idiopathic hypercalciuric patients with the stones. We are taking out renal papilla biopsy by Dr. Gu, and we are collecting, we are going to collect the blood and urine samples, check that changes in the immunological changes, activation of the monocytes and macrophages. So basically, the macro crystals formation could activate these residential monocytes and macrophages. The either way, as previously I said, classically or alternatively, these changes, activation depends upon how much crystals there. So based on this, the activation, the formation of the population secretes the anti-inflammatory or pro-inflammatory cytokines and molecules, how that propagates and increase the growth of stones. So the future, not only we are going to study that monocytes and macrophages, we are also going to expand our examination of that interstitial cells using the myeloid origin, more in that residential monocytes, and also some of the monocytes infiltrated from the blood. Then similarly, that M1, M2 macrophages, some of that neutrophils, these are phagocytic cells that involve on that, how it changes in the stone formers, non-stone formers, and experimental animals. Some of that in lymphoid origin, these are CD4, CD8 lymphocytes, and mostly the B cells and NK cells. These are the cells surrounded by some of the crystals. This morning, you can see that Grand Rounds, Julie explained that some of the cells are associated with the Randall block, some are not. Then other Randall blocks associated fibroblasts, that fibroblasts may comes from that pericytes, in that peritubular, that pericytes and peritubular fibroblasts came into that. And apart from that, we are also going that undifferentiated cells are increased, that based on their study, that maybe comes from the tubular stem cells, that's increased that undifferentiated cells may be due to increase of senescent cells. So we don't know that which cells get senescence here due to accumulation of calcium crystals. We can characterize by that using senescent marker and cell-specific surface markers by their kidney tissue and also in the urine, collected urine. The vesicles, the extracellular vesicles comes from the specific type of cells, renal and tubular cells. So I'd like to thank most of their funding from that. Right now we have that P20, NIDK, and other previous fundings, and all these works are done without the help of Dr. Lieske, the Rule, and Dr. Herrera, pathologist, Dr. Gu, and J.C. Williams, my colleague in hematology, and Mike Romero. And some of this staining was done by Vega and Elena, some of the technical helps from Seba, Linton, Miles, and Heather. I would like to take any questions. Thank you for it. Yes, we do.
So why some of the mice they have low calcium? I'm assuming all of them are on the same diet.
Yes.
And it's not related to an age of 6 months old eat more.
Yeah.
Did you try to figure out to put them on the same amount of diet and see how much they eat?
They
it seem to be the phenotype is maybe because some of them are eating more than the others.
That's when
urine calcium you see
Yes. So we didn't count exactly how much is eating it kind of thing, but we are feeding normal diet. We are not feeding any high calcium or low calcium diet. But the urine volumes after 24 hours, most of the mice, there's no significant differences. Calciums are different, but the urine volume is not different. So maybe the future we will check how much is eating and kind of thing.
You correct for urine creatinine?
Yeah. We correct the data. The calcium is corrected by creatinine too. One is per DL, then another corrected with creatinine. So,
so the claudin 2 is a tight junction protein, right?
Yes.
And so, like I know there's studies that in kidney cell cultures where oxalate exposure or calcium oxalate crystal exposure reduces the tight junction proteins, and so like in vivo, is it the case that if you have high amounts of calcium oxalate going through there, you're reducing claudin 2 kind of in a similar way that you're seeing here, so that would also increase calcium?
No, because that the crystals forms the downstream, right, down that medullary regions. These are claudin 2 is proximal tubules. It's observed out there. So nothing with that crystals doing that. So maybe that calcium, the expression, or maybe here the descending limb also they have it, that some of expression claudin 2, that maybe changes the expression too, but we don't know. But his expression is both cortex and medulla is decreased. Okay, Peter.
I had a similar question actually about variability in the calcium. I guess you have a similar genetic background on these animals?
Yes. Yeah.
Is there any crosstalk between claudin 2 and claudin 14? Because the claudin 14 is the second
Yes.
inhibit the parathelium and lead to more hypercalciuria.
Yeah.
Is there any crosstalk between claudin 2 and 14?
There's no mouse there, but if we need it, we can create that. So there's no claudin 2 14 and claudin 2 crosstalks. So there's no. Yes, please.
Do the mice with hypercalciuria, do they have polyuria? Do they make more urine?
Nope.
They don't.
Yeah, there's no difference, the urine volume. So it's the data.
But not all of those 16 have hypercalciuria, right? Like, so the question is, does volume?
Yeah, volume. Yeah, I understand that. That's why we corrected creatinine too, right? Here, if we correct the creatinine, it needs to take care of that, right? No?
But because volume may be mediated by something else, right? By calcium sensing receptor or something else in that, right? We can talk about it.
Yeah, it's fine. I understand that, but we didn't check it, but I have all the volume. We can check that one. I can check it that later. Yeah, thanks.
Thank you. Okay, so we'll hear from Dr. Felix Knauf from Germany, talking about the role of anion transporters in oxalate crystal clearance and renal macrophages.
I would like to start by acknowledging actually the person who did most of the work I will be talking today about, which is Anna Nijensen from my group, shown up here on the right. These are my disclosures. Just to be brief, I mean, you've seen that before. We know that oxalate is produced in the liver and also absorbed from the intestine. Dr. Hassan has talked about this already. And enteric hyperoxaluria, we've modeled that in mouse models by feeding mice a high oxalate diet, similar to what we see in patients with short bowel syndrome, where there's an increased absorption of soluble oxalate in the intestine. We can mimic this in mice by feeding high oxalate in the absence of calcium. This is shown again here. So we've established a mouse model that mimics enteric hyperoxaluria. And when we feed these mice low or high oxalate diet, then the high oxalate diet fed mice develop progressive kidney failure, as indicated by rising creatinine over time. Now, we've used this model to get into the mechanisms how oxalate crystals may cause kidney failure. And again, I would like to emphasize that this is a model of oxalate nephropathy, not oxalate kidney stones that we've just heard about. So in this model of oxalate nephropathy, we can see the crystals in the tubular lumen, but also in the interstitium. And one thing we also noted back then is this very prominent infiltration of macrophages here shown by F480 staining surrounding these crystals very prominently, and there's also in some areas granuloma formation. Again, and you've seen just this picture before. Again, our model is more of oxalate crystal nephropathy, and what Dr. Jayachandran has just talked about is kidney stone disease. But in both these, I would say, milder forms of a crystal disease and the more severe form, there seems to be an involvement of inflammatory cells, which different groups of investigators have shown now over the past several years. One question that we've been looking at in the past is, how do crystals activate macrophages and lead to eventually then cytokine secretion and the inflammation and the chronic kidney disease? And we and others have been able to show that this is largely due to activation of the innate immune system and the toll-like receptor activation followed by inflammasome activation. I would like to emphasize that, again, I mean, this model can be easily mimicked in vitro, so you can give any forms of crystals and they will activate macrophages and dendritic cells to then, via TLR4 activation, will result in cytokine release. We have noted that in vivo models are a bit more tricky, so it depends largely on the genetic background used, most likely also on microbiome composition, when we try to dissect now here the role of the inflammasome. So it works easily in the in vitro model, but it's quite tough actually, depending on the background of mouse models used, to then show the role of the inflammasome also in the in vivo model. But I think what can be said is that I think we mostly agree that we think that crystals damage here the tubular lumen. This then leads to damage of the tubular cell, and eventually then leads to monocyte neutrophil activation, and that then leads to the kidney damage and eventually kidney failure. What is less clear, and what I would like now to address over the next 5 to 10 minutes is, what's the resolution look like, and what's how do crystals actually get revolved? And as mentioned, this is work from Anna Niansen, and it originated back in my mentors led Peter Aronson, where we've been jointly now working on this project. And again, the question is, let's say you stop the flooding of the kidney with crystals, how do macrophages then clear these crystals? Because clinically, we've also seen this actually now in patients, for example, even with primary hyperoxaluria, when their oxalate concentrations in plasma and urine are better controlled, that there might be some resolution of nephrocalcinosis. So macrophages may really have the capability of clearing up the crystals in the parenchyma. And this has been shown by other groups in the past, shown here, where again, macrophages are able to phagocytose and then engulf crystals. And then eventually, after quite some time, so 48 to 72 hours, there's clearance of crystals, as shown here in the lower panels. So you've heard about the SLC26A6 transporter, or the short form A6 from Dr. Hassan. We have originally described it in Peter Aronson's group when I was a student there in the proximal tubule, so it can mediate various forms of anion exchange, but we think it's predominantly mediating chloride oxalate transport, but can also mediate chloride formate and to some extent chloride bicarbonate transport. But it's also, as Dr. Hassan has just shown you, expressed in the small intestine, but in the mouse largely in the small intestine. In humans, we think more in the large intestine. So the hypothesis that Anna has tried to then address is, well, if these crystals are being dissolved, there must be a mechanism how oxalate is then being secreted out of the macrophages, so to lower the oxalate concentration within the macrophage, so there can really be dissolution of the crystal or maintenance of dissolution of the crystal. And what she has been hypothesizing, and again, this is joint work with Peter Aronson, is that A6 here in the macrophage mediates the extrusion of oxalate out of the macrophage. So we used wild type and A6 knockout mice shown here, and then we used our established protocol to then derive bone marrow derived macrophages, and we looked at the expression levels of the A6 transporter and performed function, and Anna performed then functional studies. So as shown here, again, we know that the A6 transporter, as previously shown, is highly expressed in the kidney. Here you can see it's not expressed in the knockout mouse, so this serves as a control for the specificity of the antibody. And then Anna has shown here now that also A6 is expressed in macrophages, as shown here in this lane. And again, the negative control here, so to say, is the A6 knockout macrophages where the band is absent. Anna has also looked at the various expression levels of A6, A1, and A2 in macrophages. So A6 seems to be highest expressed in terms of the known oxalate transporters in macrophages. So the idea again is that then here these macrophages can also mediate the actual transport. So is it functional? Is it just expressed or is it functional? And we've performed the inverse transport by creating a chloride gradient and then measuring the radioactive uptake, so which is somewhat against the physiological function, but this is the way we can assess it easier by measuring the radioactive uptake into the macrophage, and that occurs here. You can see when Anna applied a chloride gradient, 14C oxalate is taken into the macrophage, and then this is absent in the knockout, which shows that really A6 is the main oxalate transporter in macrophages. She's also done it in the reverse fashion, that she's incubated wild type and knockout macrophages under physiological buffer conditions. And as you can see, and as would be expected over time, the knockout macrophages that lack the transporter accumulate oxalate intracellularly within the macrophage. So it works in both ways, like a revolving door. You can do a chloride gradient, it goes into the macrophage. You can apply physiologic conditions, and oxalate will accumulate within the macrophage. So again, the hypothesis at this point is oxalate transporters are critical in crystal clearance, and somehow the crystals get into the macrophage, they get dissolved. I think it's still unknown how this really occurs in detail, but the release of the soluble oxalate may in fact really occur here through the A6 transporter that, again, first was described in the kidney and intestine, but also seems to be here expressed in the inflammatory cells. So next, what Anna's been working on is to demonstrate what's the functional relevance. So she's used wild type and again knockout macrophages for A6, incubated them over time with crystals, and then washed the crystals away. And then she has looked at the crystal retention within the macrophages over these time points, because again, studies shown 20 years ago show that after 24 to 48 hours, crystal can be cleared from the macrophages. These are actually extremely difficult experiments, and Anna has invested a lot of time in figuring this out. And you can see now here in the wild type macrophages, the crystals that are in the macrophage, and also you can see them in the knockout. So the initial phase of phagocytosing the crystal seems to be intact in both the wild type and in the knockout. But then she's also looked, how about of the release of the soluble oxalate over time? And as expected, there's less release now of the soluble oxalate from the macrophages. And she's looked over time, again, no difference at 2 hours. But then over time, she sees that there are differences between wild type and knockout that we're currently working still out. But there seems to be a clear change here that now over time, there's more retention of the crystals in the knockouts as compared to the wild types. So to summarize this, and again, this is work in progress, we think that oxalate crystals get here phagocytosed and internalized by macrophages. And at some point here where the soluble oxalate is present, it then gets extruded from the macrophage through the A6 transporter. This is essential. And I think about it just like urine. You don't want to have supersaturation of oxalate within the macrophage. So there's a mechanism in place to get rid of the soluble oxalate. If that is defective, then this mechanism of crystal clearance appears to be impaired. And again, with this, I would like to close. And again, thank you. And I'll be happy to take any questions now or later during the question session.
Thank you, Felix. Let's take the questions now. Dr. Romero.
Very nice presentation, Felix. Thank you. I noticed, excuse me, that your Western blots, when you hit the A6 protein in the macrophages in the wild type, that in fact, not only is the band higher, but it looks like there may even be a glycosylation processing band. Do you think that's an artifact of loading, or do you think that there may actually be a different form of SLC26A6 that is playing a role in macrophages?
Excellent point, Michael, that you spotted that. Yeah, I mean, A6 does exist in the glycosylated, non-glycosylated form. And even when you compare kidney to intestine, there's different sizes then that it runs at. I don't know if we have done the respective experiments yet to look at whether we can get rid of the glycosylation or not. I think Anna, I've spotted her. She might maybe know. I don't. So it's an excellent point. That the band is truly higher. Maybe I think Anna can answer this.
Hello, everybody. So yes, we have some preliminary data doing Western blot with deglycosylation assays previously to the Western blot, and we saw that the band for the macrophages and the band for the kidneys are at the same molecular weight after the deglycosylation. So yeah, we think it's it's a different glycosylation process in the macrophages A6.
A6 is, sorry. Yes, A6 is present in the kidney macrophages too. They're similar to the blood. The macrophage comes from bone marrow. Their activity is different than the residential macrophages.
Right. I mean, we've used, obviously in our model now, the bone marrow derived ones. So we haven't separately studied resident, that's a good point, versus the bone marrow one. So, I mean, we've so far been focusing just on the in vitro studies. We have plans in terms of, doing in vivo experiments with bone marrow chimeras or more specific knockout in the macrophage, but we haven't gotten there yet. So, I hope Anna said this correctly, but I think, yeah, so we haven't really looked at yet resident versus bone marrow ones at this point.
I had a brief thought. This is John Lieske. Our journal club today, they were doing this detailed phenotyping of the kidney tissues. I wonder if there's data that you could get from that study. They had macrophages in there. I don't know if they would. I'm sure it's there in the data. I don't know.
Which one you're saying?
Well, they had the macrophages.
Yeah, macrophages there, but they don't.
I'm sure it's in their data set somewhere.
Yeah, they may have that A6, but they not have that activities. So yeah, it's get lysed by the lysosomes. Basically, the pH is low. That's what crystals cleared themselves after endocytosis.
One question, this is probably a work in progress maybe. Did you look at the impact of this in the A6 node mice or cells? They are loaded with crystals. You look at the functional characterization of the macrophages, are they behaving different when they have more crystals? Or that's something we plan to do?
So if I understood the question correctly, you were saying in the A6 knockout, whether we studied in macrophages there and whether they behave different. The problem of studying it in the whole body knockout is obviously that the feeding model won't work, because then you have the higher intestinal absorption in the A6 knockout. And then when you compare wild type and knockout, then you just by the higher intestinal flux have much more severe kidney damage in the whole body knockout. So that's why we've transitioned to as has Peter Aronson, and we were grateful to get the Flox one in more tissue specific knockout. So I think to really answer this question, we need to have the same load of oxalate, and therefore our model with the oxalate feeding is not is not suitable because the intestinal A6 depletion will overwhelm the kidney with crystals.
Okay. Thank you. So I'll talk to you today about the mechanism that the microbiome influences kidney stone formation. Okay. So as humans, we have around 30 trillion human cells, but we have more than 39 trillion microbial cells. So we are more than 50% microbial at the cellular level. At the gene level, this is even more striking, where we have 20,000 human genes and up to 20 million microbial genes. So we are like 1% human and 99% microbial. So the microbiome is defined as the collective genome of the microbes, either bacteria, bacteriophage, fungi, protozoa, and viruses that live inside and on the human body. Okay, so there has been a lot of studies recognizing the effect of the gut, especially the gut microbiome, on human health, where microbiome has been involved in the development and training of the immune system, in breaking down of food component, and protection against epithelial injury, and many others. So I'm not going to go through a lot of details about those studies on the antibiotics and kidney stones, but the two studies by Tejan here and by Ferrero have found a significant association with antibiotic use and kidney stones. And then the study that was just presented brought up like the possible limitation with those studies. However, as was again mentioned, that we have a possible hypothesis, a working hypothesis, that the microbiome, by affecting the level of different antibiotics, especially in the gut, can modulate how we metabolize our diet and affect our absorption of lethogenic substances. And in particular, I'm interested in oxalate. So oxalate, we know, this group knows, is produced as from the metabolism of an amino acid, but it's also absorbed from the diet in our GI tract, where a number of bacteria that we call the oxalobiome can degrade oxalate. They include Oxalobacter formigenes, Lactobacillus, and several other bacteria. And they are, we think, protective against oxalate toxicities, including kidney stones. So in a study where we had germ-free mice and conventional mice with their own microbiome, we fed them 3 different diets: normal chow, hydroxyprotein as precursor to oxalate, and sodium oxalate. And we measured their 24-hour urine, and we see here on this graph, where blue is conventional mice and red is germ-free. On all diets, there is some reduction in urinary oxalate, which was close to significance just with the high oxalate diet. And note the variability in the 24-hour urine collection. But we also looked at fecal oxalate levels, and here we had significantly lower oxalate in the conventional mice on the 3 diets. And we also measured serum oxalate. Again, there was lower serum oxalate in conventional mice than our germ-free mice on the 3 diets. So we think oxalate oxalate is reduced by the presence of the microbiota. We also looked at the gene expression of oxalate transporter that we heard about, this SLC26A6, important for oxalate excretion. And we looked in the kidney, and we found that germ-free mice had lower expression of those genes. And we looked in the GI tract, in different segments, there was lower expression of the SLC26A6, especially in the earlier, in the small intestine and the proximal colon, but not in the distal colon. So we think, in addition to oxalate degrading capability of the microbiome, it modulates oxalate transporter, as was presented previously by Dr. Hassan. Okay, so we asked the question whether if in humans, if we suppress the microbiome using an antibiotic, that leads to increase in oxalate levels. And for this, we had a population of end-stage kidney disease, so patients who are on dialysis, that we gave them in a randomized single-blinded fashion, either placebo or vancomycin, a low dose of vancomycin once weekly, oral vancomycin, 250 milligrams. And at 3 months, the subjects were crossed to the other study group. And we collected pre-dialysis blood samples at the same time on dialysis, monthly for those participants. And we see here that in the placebo group, there was no significant change in plasma oxalate over time, as we expected. But in the group that was treated with vancomycin, this low level of vancomycin resulted in significant increase in plasma oxalate in those participants over time. And in each one of them, there was an increase in plasma oxalate, but some had much more than others. So we're currently analyzing, we analyzed the microbiome, and we think there is a role of reduction in oxalate degrading bacteria in that group. So going back a step and looking at whether the microbiome is different in kidney stone patients. So there has been several studies that looked at this question. Earlier, a study looked at oxalobacter colonization in recurrent kidney stone formers and non-stone formers, and showed that in stone formers, we have lower colonization with oxalobacter as compared to the non-stone formers. And looking at the microbiome in general, there was a lower abundance of a lot of species. And here, when they did the analysis, they found a differentially lower number of species in the group of stone formers as compared to non-stone formers. So there is a lot of bacteria that are differentially present or abundant in this. And our group here, with funding from the Rare Kidney Stone Consortium, looked at the gut microbiome alterations in hyperoxaluric conditions, where we compared healthy control to calcium oxalate, stone former, primary hyperoxaluria, and enteric hyperoxaluria. And just a quick overview, we see that there is a number, the abundance and the relative abundance of species of different taxa was different between the groups. And when we focused on the bacteria with oxalate degrading genes, and we found the overlap of those bacteria in those conditions, we see that we have 17 species that were shared between the 4 groups, but each one of these conditions has a different combination of oxalate degraders. So we think that might have a role in oxalate, in the stone formation oxalate metabolism. So what other mechanisms can affect kidney stone formation? So we know that the gut produces large amounts of short-chain fatty acids, and those have been shown to modulate passive absorption of oxalate, here by modulating tight junction, and also by modulation of the active transport of oxalate. So that could be another mechanism by which the microbiome can affect your risk for stones. So in the study by the Denver group, where they looked at children with and without kidney stones, and they analyzed their microbiome and their metabolome, and they found actually that here on the left-hand side, they have the stone formers had a reduction in number of butyrate producers in red here, and a number of oxalate degraders in blue here. In the stone versus the controls. And they also found a reduction in stone formers in the butyryl coenzyme dehydrogenase, which is the enzyme that produces the short-chain fatty acid. So there might be a dysregulation of the short-chain fatty acid affecting the stone formation. And also the study by Stern, what they did was to to perform a fecal transplant from conventional rats to mice, germ-free mice. And in addition to looking at oxalate here that was reduced after the fecal transplant, they looked at calcium, ammonium, and pH, and we see calcium was reduced over time, ammonium was reduced, and pH was increased. So it seems like it's not only oxalate that is modulated by the microbiome, but there is an effect on calcium and on acid base. And they looked again at the SLC26A6, and they see a downregulation in the kidney after the fecal transplant, transplant, and an increase in the cecum. So to summarize, the gut microbiome is an important determinant of oxalate homeostasis. We think through oxalate degradation and modulation of active and passive transport. And the gut microbiome, including oxalate-degrading microbiota, are different between stone formers and non-stone formers, and short-chain fatty acid could play an important role in kidney stone disease. And the microbiome, the gut microbiome affects level of urinary calcium, urinary ammonium, and pH. However, there is no studies to date that show that the gut microbiome manipulation affects stone formation. All of this is through urinary parameters change. Okay, so how about the urinary microbiome? So there has been recognition that the stones can have bacteria in them, and in the study where they cultured, where they reviewed studies on the culturing of kidney stones that are non-infectious, they found that you can culture bacteria from non-infectious stones. So where is this bacteria coming from, and is there a role of this bacteria in stone formation? There are some issues with the urine microbiome studies, because first, it's a really low biomass of urine microbiota. So as compared to the gut microbiome, which is 10 to the 13, 10 to the 14 bacteria, in the urine are less than 10 to the 5 CFUs per milliliter. And it's also the proximity to other bacterial niches with higher microbial biomass that can lead to contamination of the urine with this bacteria. And also there could be a lot of contamination from laboratory environment, from reagents and everything. So it's important to add blanks and to control as much as possible for contamination. And there is also the urinary microbiome can be collected from different area, either from the midstream urine, which could have a lot of contamination from the skin, but it's easy, it's non-invasive, or it could be the transurethral intermittent catheterization, which can reduce slightly the contamination, which it's invasive, or it could be the suprapubic aspiration, which is the most difficult and probably the lowest contamination risk. But those are difficult to perform. So here, Aaron and a few other people developed this possible standardization for microbiome studies to come up with a consensus to how to collect the samples. But a lot of studies in the past have not followed all of those guidelines, so it raises the question of whether this is the results are valid. So again, the urine microbiome from healthy and kidney stone formers have been described by Cantero, and they found that there is a number of taxa that were different between calcium oxalate and controls. And then there was other studies that looked at another study that looked at the urine from the bladder of healthy people compared it to kidney stone bladder, and so they saw different bacteria in there. And then the urine from the pelvis had the lowest number of taxa, which is expected, as we discussed, in the presence of contamination. So what's the possible mechanism is it's still the studies are still new in this, but basically there was the study where they looked at E. coli in solution that can selectively aggregate around calcium oxalate crystals, but not other crystals. So there might be an effect of the bacteria in aggregation of crystals. Other speculated mechanism could be, again, the binding of the bacteria that can change the microenvironment and lead to more deposition of crystals. The other possible effect, which I didn't find really like studies on it, which is the bacterial enzyme citrate lyase that can degrade citrate, reducing urinary citrate, but again, no real data for that. And then the bacteria can bind the urothelium and again change the environment around the urothelium, cause some inflammation, and that leads to kidney stone formation. So to conclude, the gut microbiome affects urinary chemistry that likely translates into change in the risk of kidney stones, but that has not been demonstrated. And there is a possible role of the urine microbiota, but a lot more studies are needed to establish the causation. I'd like to thank everyone in my lab and the funding of the Rare Kidney Stone Consortium, OHF, and everyone. Thank you. Can take questions.
Yeah, great talk. My name is Kevin. I'm one of the urologists here. I'd say from a urology standpoint, we struggle a lot with positive stone cultures and what to do with those. We operate on someone, if stone culture comes back positive, then there's a huge variability what people will put patients on after that, whether it's no more antibiotics, 2 months of antibiotics, longer. What do you think? I mean, it's almost like contradictory data. The bacteria is bad, but it's also good. So you could imagine a scenario where prolonged antibiotics increases their stone formation based on your data. What do you think as far as these positive stone cultures? How should we approach them based on what you and others?
Yeah, as you know, the data is not there, but it's like, as a person who is a more stronger believer of the gut microbiome disruption, because you have the much higher biomass, the effect of the effect of antibiotic on the microbiome of the gut has been like very clearly studied. So I would say, like, unless the patient has maybe like signs or symptoms of an infection, I would probably like not put them on antibiotic, but this is just my opinion. I would not like, I don't think we have data to support that. But
a different side than I think we think about a lot, which is antibiotics could actually be harmful. So it's good to see this. Thanks.
Thanks. Yeah, Aaron.
So nice overview. So I just had a quick, I guess, technical question. In your hyperoxaluria gut microbiota study, how did you identify the species that had the oxalate degrading genes?
So we did the FRC gene identification through our own. We have our own pipeline that identifies oxalate. We published on the.
Okay, so it was like a specific targeted analysis.
Yeah. Just to identify the oxalate degradation pathway, the genes.
Yeah. All right.
Okay.
Great. In your decrease in A6 expression at the mRNA level, did you confirm it? Not all mRNA get translated into protein level.
Yeah. So actually, for that study, we didn't have any more tissues, so we didn't do it. But in future studies, we're planning to do confirmation. But I think that Josh has looked at expression has looked at protein level in his analysis, and the protein level was decreased. So I think not in our model, but in Josh, they looked at protein and it was decreased. They don't have oxalobacter, so it's not oxalobacter. It could be others. Yeah.
I see a differential effect because there is an increase in A6 in the intestines, but a reduction in the kidney. You have a reduced A6 in the kidney, you will have reduced active secretion.
Yeah.
That can lead to more higher plasma oxalate theoretically.
For us, actually, they were decreased in the kidney too. But I think Josh had an increase in the kidney. So yeah, we definitely need more. Okay. Thank you, everyone. We're going to move on to the next talk, Antibiotics and Stone by Charat Thongprayoon. Sorry.
I'm Charat Thongprayoon. I'm one of the nephrologists working in Mayo Clinic, but I mainly work in Cantero. Today, I'm going to talk about our study in the antibiotic and the risk of kidney stone, I have nothing to disclose. So, I mean, it was proposed that based on, as a clinician, it was proposed that antibiotics can increase the stone formation by changing the gut microbiome and leading to alteration in metabolism and absorption in the gut, and ultimately the urine excretion of metabolites such as oxalate, citrate, and calcium. However, the association between antibiotic use and kidney stone can be complicated. That's mainly because kidney stone have similar presentation to urinary tract infection and also can co-occur with urinary tract infection. So when patients present with hematuria or dysuria, often that they did not have imaging throughout kidney stone, and they are treated with antibiotics for a presumed diagnosis of kidney stone. And then later on, they were found to have kidney stone based on obstructing stone on imaging or kidney stone passes. So actually, antibiotics were given due to the symptom of unrecognized kidney stone, indicating reverse causality. A prior case control study using large electronic database in UK showed that Bactrim, cephalosporin, fluoroquinolone, and nitrofurantoin were significantly associated with the increased risk of kidney stone. However, this study only used diagnosis code to identify the kidney stone and did not have detailed medical record review, which is needed to clarify the timing of kidney stone and antibiotic use. So I think the result of this study might be subject to the reverse causality, as I mentioned. I just want to point out that Bactrim, fluoroquinolone, and nitrofurantoin, which were shown in this study to be associated with increased risk of kidney stone, were commonly used to treat urinary tract infection. Although the sensitivity analysis excluding UTI show consistent result, I would question whether the study adequately exclude urinary tract infection because nitrofurantoin, which is antibiotic exclusively used for UTI, still remain in the analysis. So to study the association between antibiotic and the risk of kidney stone, it is very important that we need to carefully review medical record to validate symptomatic kidney stone onset and to determine indication for antibiotic use. And in addition, despite medical record review, it is uncertain that we cannot determine whether urinary symptom is due to unrecognized kidney stone or not. So, I mean, honestly, I think that we could not assess the actual risk of kidney stone associated with antibiotic use for urinary symptoms based from the observational retrospective study. So to address some limitation, we conducted population-based mass case control study, including stone formers and mass control in Olmsted County, Minnesota. We included about 1,047 confirmed first-time symptomatic kidney stone formers and 4,024 age and sex match control. We defined the index date as the date of the first symptomatic kidney stone onset. The exposure of interest was the outpatient antibiotic use within 5 years prior. In our study, we initially identified kidney stone using ICD-9 diagnosis code, but we performed manual chart review to validate a first-time symptomatic kidney stone episode and to determine the exact date of symptomatic stone onset. Symptomatic kidney stone former were required to have flank or abdominal pain or gross hematuria and stone obstructing the ureter on imaging or avoided stone. We excluded patients with asymptomatic kidney stone, patients with suspected symptomatic kidney stone, and patients with prior kidney stone episode. This table shows compare clinical characteristics between stone former and mass control. The mean age was 46 years, 56% were male. Most of patients, 90% were white. Stone former were more likely to have obesity, diabetes, hypertension, chronic kidney disease. Stone former also more likely to have prior urinary symptom before the index date compared to mass control. This table shows odds ratio of the first-time symptomatic kidney stone with antibiotic use. The risk of first-time symptomatic kidney stone was only significantly higher within 1 year after antibiotic use. However, after we adjusting for race and comorbidity, the odds ratio was reduced and no longer statistically significant. As I mentioned, because the cause of urinary symptom can be uncertain, to ensure that reverse causality did not affect the risk of kidney stone with antibiotic use, I think we need to consider urinary symptom before the index days as potentially undiagnosed kidney stone and excluded this patient. This figure shows the risk of kidney stone after antibiotic use. The blue one was before we excluded patient with prior urinary symptoms. The red one was after we exclude patient with prior urinary symptoms. As you can see, before we excluded patient with prior urinary symptom, the risk of kidney stone was highest right after antibiotic use, which is kind of against our hypothesis that antibiotic increase stone formation by changing the gut microbiome, because we would expect that the process would take weeks or months before for stone to form, grow before their symptomatic passes. After the exclusion of patient with prior urinary symptom, substantially reduce the risk of kidney stone in early period after antibiotic use. It's kind of like suggests that likely that this might be more about reverse causality. After excluding patient with prior urinary symptom, the antibiotic use within any time period within 5 years prior was not significantly associated with kidney stone in unadjusted and adjusted analysis. We also perform analysis based on antibiotic class. There was no significant association between kidney stone and any use of any specific antibiotic class in any period, and also as well as the number of antibiotic causes within 5 years. So conclusion based on our study, right now what I can make is there was no evidence of increased risk of first-time symptomatic kidney stone with antibiotic use for non-urinary symptoms. Under recognition of kidney stone that initially cause urinary symptoms resulting antibiotic use may explain much of the observed stone risk with antibiotic use. Thank you.
Questions?
So that's a very nice slide. So when you said urinary symptoms, and then you had a slide that showed what that meant, it seemed confined pretty much to UTI-like symptoms.
Including hematuria, UTI, so that's kind of the main thing.
But not like overactive bladder or other lower urinary tract symptoms.
So kind of along those lines too, were you able to differentiate your stone formers into different stone types?
We have, so we don't have, this is a retrospective study. About 50% of our patients have like stone composition. About 75% is calcium oxalate. And then about 20% is calcium phosphate stone.
I think the period of time it takes to get a kidney stone is not completely clear. So what you said about the time between the antibiotic exposure and the potential stone is interesting, but I think that number keeps changing. So like Greg Tazian's data on heat show that the lag between the hottest day and the kidney stone was up to maybe just 3 days. And that always changes my idea about how long it takes. I used to think it took years to make a kidney stone. Then I thought it was months. And now I think it may be just days. So it could be a relatively short period of time. But it certainly makes sense that if a change in microbiome occurred, you'd expect that to take a while to be manifest as a change in urine chemistry and kidney stones. But I think
yeah, and one of the data that we use is our prior study that we study association between pregnancy and the risk of kidney stone. And as you can know, pregnancy increase increase urine calcium excretion, also increase urine PS. And in our study showed that the risk of kidney stone was higher after the second trimester. So that's why we come up with some kind of discussion about weeks to months before we see increased rate of kidney stone. It takes time to form, but to be the have a symptom, basically is the bigger the more likely it causes symptoms.
So just to piggyback off that and add one comment, so yeah, I agree with you. We can all go to a basic science lab and create crystals and aggregating stone crystals in a beaker. and watch it occur before our eyes in minutes. So for me, I've always thought, well, why couldn't it happen, relatively quickly in the human body, right? It's just urine chemistry. But then the other thing about a study like this that I would caution is that we should all acknowledge the disconnect between a symptomatic stone passage and stone formation, right? They are completely unpredictable events, and so we've all seen patients, who if you image them, they've never had a symptomatic stone, and they're full of kidney stones. So we should acknowledge that there is a challenge there to make a connection between the stone formation versus stone passage.
I agree. Thank you.
So we will move on to the next talk by Dr. Christian Hanna on crystals, inflammation, and cysts.
I'm one of the pediatric nephrologists here at Mayo Clinic. Thank you to Dr. Lieske and Dr. Cole for inviting me for this conference. And just so I have one learning objective. I want to discuss proposed mechanism of urinary crystals, induced kidney cyst formation, and progression of ADPKD. Just a brief ADPKD overview for the audience who's mainly working at kidney stones. It's the most common inherited kidney disease in adults. It affects approximately 12.5 million people worldwide. It's related to relentless development of kidney cysts, and it's the fourth leading cause of kidney failure in adult after diabetes, hypertension, and glomerulonephritis. In the adult population, there's advanced studies regarding imaging and biomarker to address the disease progression, mainly the total kidney volume, which is an FDA-approved gold standard biomarker to predict progression and assess treatment response in adults with ADPKD. However, in pediatric, we know that the cysts start early in life, are the main contributor to total cyst volume and kidney size in adult. And cysts in ADPKD only developed in 1 to 3% of the 2 million nephron in both kidneys, so only 1 to 3%. So there's an urgent need exists to identify any biomarker for predicting ADPKD progression in pediatric patients and guide the preemptive treatment strategies for these children. And this is what led to the discussion about urinary crystals and inflammation. When I joined Mayo Clinic in December of 2019 to run the pediatric PKD clinic, I met with Dr. Dawn Milner. I think she's over the Zoom meeting, and to discuss if she have a cohort of patient of PKD, because she's been here for many, many years, decades. And she shared with me a list, but she told me, Christian, I wanted to look at this cohort of patient of CYP24 A1 deficiency and see if I've seen they have kidney cysts. And how about you look into these cohort and we can work together on that. So with the help of Dr. Milner, Dr. Lieske, and the Kidney Stone Consortium and our PKD expert, Dr. Harris, Torres, and Dr. Shabib, we're able to look for a 16 patient with the CYP24A1 mutation. And for the audience, it's a condition that you lack this enzyme, which degrade the 125 vitamin D. So if you have a deficiency in this enzyme, your 125 vitamin D goes up. That will lead to hypercalcemia, hypercalciuria, and development of kidney stones. So out of these 16 patients identified with genetically confirmed CYP24A1 mutation in Dr. Lieske genetic labs and the clinical lab, we found that 100% of these patients had either medullary or corticomedullary kidney cysts. And this is a samples of the cohort. You can see in panel A, you can see a cyst and a stones in the red arrow. Panel B, another cohort in MRI that revealed kidney cyst, and you don't see a kidney stone in MRI. On the ultrasound panel C and D, you can see a combination of kidney cyst and nephrocalcinosis, medullary nephrocalcinosis in these patients. We moved this further to look at the cohort of HHRH, which are the hereditary hypophosphatemic rickets and hypercalcemia. And these are related to the SLC34A3 pathogenic variants. This gene encodes the NAPI2C channel in the kidney tubules, which is the sodium-dependent phosphor channel. And if you have a mutation of this gene, you will have phosphor leak in the urine that lead to elevated 125 vitamin D and hypercalciuria, and that the proposed mechanism for kidney stone. And these patients can also have rickets because of the low phosphorus. So we look at 12 patients with a pathogenic variant in the SLC gene. Some of them had a monoallelic and other have biallelic variants. And we found kidney cysts in about 75% of these patients. And we have 3 children, and all of them have more than 2 cysts, which is enough, which is above the 99th percentile for pediatric to have a kidney cyst. So, and this is another samples. There is a study from 2009 that looked at patients with parahyperthyroidism compared to the normal population. These patients had a preserved kidney function out of 172 versus 210 of age and sex match healthy control. There is increased risk of simple kidney cyst of 34% versus 16.2%. So the similarity in the biomarker with these 3 different cohorts are they all have hypercalciuria, they all have elevated circulating 125 vitamin D level. The PTH level is high in the parahyperthyroidism cohort, but it's low to normal in the CYP24 and the HRH. So this is why the question is hypercalciuria or crystalluria or elevated 125 vitamin D level are contributing factor to cyst development. Around that time in 2019, a PKD group in California with Tom Wims have looked at animal model and suggested that the position of calcium oxalate or calcium phosphate crystal in the renal tubules, as you can see on the right side of the imaging, can trigger rapid tubule dilatations in healthy normal kidney animal or ADPKD animals. And also can activate polycystic kidney disease associated signaling pathway, which is the mTOR and the STAT3. And that led to accelerated cystogenesis in polycystic kidney disease and disease progression. While in a normal animal model, when these crystals passed, the tubule have just an intermittent dilatation, the tubules went back to normal. Macrophage and monocytes, as Dr. Jayachandran discussed the roles in kidney stone development, their roles in ADPKD progression as well. And this study from Kidney International from 2013 revealed that macrophages in the arrows present in autosomal dominant kidney disease epithelial cells. The same author concluded that primary ADPKD cyst epithelial cells, when they incubated in the low fetal bovine serum versus a high serum bovine serum, you can have more proliferation of the PKD cells. But also if they added a naive raw macrophages in a low serum medium as well, and the right panel of the arrow of A and B, you can see as well increased proliferation of ADPKD cells and the NHA cells as a control. Other factors have contributed to promote ADPKD progression, including interleukin 1B2 and TNF and the monocyte MCP1 as well. This is a study from Europe. They looked at 53 ADPKD children versus control and to look at the MCP excretion as an early event in ADPKD patients. And as you can see in panel A and B, urinary MCP1 was elevated in patient with PKD1 pathogenic variant compared to control of patient who have a mild disease of PKD2. Also, they look at ADPKD patient who have a very early onset PKD, which is rare, usually related to biallelic PKD variant. One is truncated, the other one is hypomorphic. and they found they have a higher MCP compared to asymptomatic patients as well. Then Dr. Lee at Mayo Clinic looked at the EVs and they found that it can promote cyst growth in ADPKD as well. With the conclusion he proposed, mentioning here one of the study that he did when he treated the PKD1 null cell-derived mice with EV exosomes. It promotes cyst growth in vivo. You can see here in the panel A, B, and C as well. Going back to crystals and kidney stones, we know that crystal can trigger tubular epithelial cells and accelerate fibrosis for cytokines and EVs. I'm not going to get into these studies in detail, but there's few studies by Dr. Lieske and Jayachandran that published here at Mayo Clinic. We know that ADPKD patients also are at increased risk for urinary stone disease. And the proposed mechanism, it could be intrarenal anatomic obstruction because of the cyst, but also there's few studies that showed low level of urinary inhibitor as potassium, magnesium, and citrate in ADPKD patient compared to ADPKD patient who does not have stones. So in summary, what's the conclusion from all this this hypothesis and the studies that we're talking about? Kidney cyst formation related to hypercalciuria or other urinary crystals, calcium oxalate or phosphor crystal accelerates cystogenesis and disease progression of ADPKD. And there's an unpublished data from this group in California about uric acid crystal, but I don't think it's published yet. We know that macrophages, monocytes, and EVs promote cyst growth in ADPKD. We know about the relationship of crystals, monocyte macrophage, and interstitial fibrosis in kidney stones and inflammation. We know that ADPKD has a higher risk of urinary stone disease as well. So this what led to a proposed mechanism with the help of Dr. Lieske and Jaya to study if there's any correlation of increased urine supersaturation, intratubular cyst formations, attachment to the epithelial activation of EVs and pro-inflammatory cytokines, and progression of cyst formation in ADPKD patient. And this is we're trying to study in the pediatric population. So with the help of a two fund from the Mayo PKD Center Cell Fund and from the NIDDK, we were able to secure money to study a 24-hour urine collection from 50 pediatric patients, 50 control, and to look at the supersaturation profile, cytokines and EVs, and comparing with a healthy control. We're including patients between 15 and 18 because less than 5, maybe they will not be able to collect a 24-hour urine. We need to catheterize them, so we're not going to do that. And we have a match control, either unaffected siblings or family members. I don't have any prelim data yet, so we're still collecting these results. And hopefully in a year or so, we'll be able to share our data with you. So I don't have any acknowledgement slides, but I want to acknowledge Dr. Milner, who gave me the idea about the cyst and crystals, Dr. Peter Harris, the director of the PKD labs, Dr. Lieske and Jaya for their help in running the samples that we're collecting in the research lab, and all the members that working at the Mayo Clinic Kidney Stone Consortium and the PKD Center as well. Question?
Interesting presentation. That's the SLC 34A3. Then you have that PTH1 that increased the parathyroid hormone and vitamin D. What happened there with FCF23, fibroblast growth factor 23? Fibroblast growth factor 23, FCF, that's one is regulating that protein expression and also inside outside the apical membrane to the endoplasmic reticulum and also inhibiting the vitamin D.
I don't know the answer to that. I don't think we checked level on these clinical cohort of these patients. Some of them have sample, but I don't think I looked at it.
Yeah, I've only seen it measured in a few of them, and it's appropriately suppressed in the phosphate wasting syndrome. Yeah. So we haven't looked at it rigorously, but yeah, it's suppressed as you would expect it.
I would expect it to be higher in the CYP24 because they have one higher 125D.
Yeah, I've never looked at it in CYP24. I'm talking in the normally NaPi2A, NaPi2C cohorts.
There is data saying that FGF23 correlates with increased progression. There may be some connection.
It regulates that vitamin D and CYP24, 27 and 24. That's inhibiting and activating that. The same thing is doing SLC34 to NaPi2C prodynamic. That's why it's releasing more fast.
Wondering one maybe the idea here. It's also follow up to the question earlier to Jane. When you have hypercalciuria, that binds to the calcium sensing receptor in the collecting bulk, and that inhibits alcoholing and leads to polyuria. And that makes perfect sense if you have hypercalciuria. You want to prevent a kidney stone and you have a dilute urine. And this phenomenon is prolonged in patients with kidney stone. So I'm wondering if the patient have hypercalciuria and the calcium binding the calcium sensing receptor and inhibiting alcoholing, and the patient is polyuric all the time. Do they have an aberrant vasopressin coming all the time to compensate? And that vasopressin can lead to cyst development, potentially?
That's one of the theory that we talk about, and I did not mention it here, but I don't think that has been studied. And this would lead to if there's any, I'm a clinical physician, I don't have a lab, so if there's any collaboration, anybody look at the lab for kidney stone model to see if they have increased risk of cyst and see if there's any mechanism. I'm happy to collaborate as well. Thank you.
Our next session is on the genetics of both common and rare kidney stone disorders. So we're excited to have our first talk here from Dr. Peter Harris, who's going to talk about monogenic panel screening to discovery.
I don't think I need to go through the details of this to convince you that monogenic kidney stones are an important part of the overall stone population. I say 10% of adults and 20% of children here. It used to say 2% and 10%, so I don't know what exactly the right number is, and probably somewhere between this number and maybe depending on whether it's a fully monogenic disease or we're looking at a single gene that's acting as a strong risk factor. So we have a protocol here for screening patients where we suspect monogenic kidney stones. And this is the recruitment criteria. I won't go through it in detail, but patients under 18, and then patients over 18 with, as well as kidney stones and nephrocalcinosis, another factor here. And you can see the factors that we have, which are, we think, enriched in the monogenic population. So we use a straightforward next generation sequencing with a panel. Our latest panel is 160 genes. So this is like the 40 or so known genes, but other candidate genes that we have on the panel. So because the panel is not very large compared to whole exome sequencing, we can pool samples for capture and pool samples for sequencing, which makes it a little cheaper. The downside is if we don't have the gene on the panel, we don't detect the changes, and all the time new genes are being identified. Then we go through a protocol here to try and determine whether the variants we are finding are significant. Looking for changes that are rare, obviously, in normal populations. We're looking to see if they've been previously described. We use programs to evaluate missense changes and splicing changes. We look for copy number changes, so larger deletions, duplications. We use these American College of Medical Genetics guidelines to try and determine in there significant, so pathogenic and not lightly pathogenic. And then we tried to discuss them together to see if it makes sense with the phenotype. And we're trying to kind of automate this process, especially for analysis of the Mayo Biobank, as Nick will tell you a little bit about in a minute. So in an older study, we looked at patients that were negative for Dent disease or suspected of Dent or PH, but then we didn't find gene variants in the known genes by Sanger analysis. And from this, we resolved another 10% of PH patients with variants in other known stone genes. And for Dent, 27% of patients were resolved with variants in a number of other genes. And you can see quite a lot of different genes were highlighted in these studies. Some of these were found biallelically. For instance, Claudin 16 was the most common in this study, but SLC 34A3 was quite common too, as was CYP24A1. And then for the monoallelic cases, SLC 34A3 was the one with the highest level. So we've gone on now looking at the monogenic cohort. And so our most recent data is that we've collected and screened 453 families with suspected monogenic disease using the panel I showed you. And we have a genetic diagnosis in 146 of these, so 36.2%. I think this is comparative to other cohorts out there, although this is a pretty large cohort now. There's a couple of examples of interesting results from this studies. Here we can see this affected individual with PH1, so AGXT, is compound heterozygous for these two changes, whereas on this part of the family, they're homozygous for one of these variants. So we can see that sometimes we get this type of unusual complexity even in one family, and genetic analysis is helpful to sort that out. Another example of an unusual variant is a intronic large deletion here that we can see. So this is the intronic region. And we think with this reduction in the size of the deletion, the exon here, the intron here that doesn't splice out properly. And in this case, we have a biallelic case here with the two change homozygous for this change, and then the two parents with kidney stones, which are are monoallelic. So we see both patterns here in one family. So looking at the results of the monogenic cohort as it is right now, you can see if we look at biallelic changes, so 114 families, 14 different genes, consistent with our history or our interest in pH. We can see approximately half of the families here have one of the pH genes. But we can see APRT, CYP24A1, Claudin A3, and then cystinuria genes are also common in this cohort. And then in the monoallelic group, again, CYP24A1, A3 is common. And we have the Dent patients here as well, which have a single variant, but of course they're X-linked. And because of our past history here, we also have a pretty large group there. But we can see here we have 13 different genes, and overall now we have 22 different genes identified in the different patients. A couple of other interesting things we're doing, looking at a cohort with a medullary sponge kidney. As you know, it's not really thought to be necessarily a genetic disorder, but maybe considered to be mainly a sporadic disorder. And we've been collecting with the help of David and Facebook, a collection of about 100 patients now. We haven't finished the analysis of this, and I don't think we're going to find genetic changes in all of these patients, but I think there there are variants that are either causative or risk factors in some of them. And one of them, interestingly, is PKHD1. This is the gene associated with a recessive form of polycystic kidney disease. But here we're just looking at single changes, not two changes, as we find in ARPKD patients. And you can see a couple of the patients here with known pathogenic changes in this gene. We're also collecting, we've collected a large cohort of PH patients, but some of them also have stones. And so we've crossovered and analyzed them both with the stone panel as well as the cyst panel. And one of the interesting genes here that we've identified is ALPL here, the alkaline phosphatase gene. As you probably know, there's examples of biallelic and monoallelic disease here, with the monoallelic disease more of an adult disease, whereas the biallelic is more infantile disease. And we can see here, we're seeing the single change here causing disease in the mother here with nephrocalcinosis and nephrocalcinosis, but also quite a lot of cysts here within the child or offspring, not really child. And another interesting thing that came up recently from clinical testing that we've also recruited for our panel is an identification of this gene EHHADH associated with Fanconi renal tubular syndrome. There's a number of other genes that are associated with this, including SLC34A1, the one on HNF-A4, which we've looked at quite a lot on our panel. You can see it's a missense change here, but a known pathogenic change in a 3-year-old individual is thought to be to do with mitochondrial respiration and impaired ATP production. And I highlight this one because it was found from the clinical testing, but we actually don't have it on our panel yet, so we wouldn't have identified it here. So it's an ever increasing issue where we're trying to add genes to the panel, and at some stage maybe doing whole exome analysis and analyzing the genes that we're interested in is going to be an approach that we will need to adopt. So there's a couple of interesting studies out. This is a publication looking at the UK Biobank and the 100,000 genomes UK England project. And you can see here, they're looking at, we can't see very well, but they're looking at rare variants in this gene and then try and find ones that are associated with the stone cohort in these populations. And you can see that the gene that's come up here in both of these is SLC34A3. So I don't think I need to go through this, but I think that it's helpful, I think, for genetic analysis to try and identify monogenic patients, and this can change the management and treatment of these, especially as in PH, where we have now a specific gene treatments becoming available. So thank you. Well, and just thank the people from the Rare Kidney Stone Consortium, Andrea, that's done all the genetic work here in the group. So thank you. If you want to talk to Barb or refer a patient for research testing, here's the contact information.
This question was great. This patient in this EHHS long abbreviation, did you measure the cyst rate? Because as part of that Fanconi syndrome, generalized optimal tubular dysfunction, this patient may excrete more citrate and they have high first citraturia.
I think you better ask David next too. I think he has more information about this patient than I do.
I don't recall the phenotype. I don't remember.
Yeah, I can't remember exactly. I think it was an external patient, wasn't it, that you had it? But I don't know exactly everything that's been measured on that patient.
I don't think they have. You're talking about the HHRH cohort?
Yeah, that's long. Yeah.
I looked at when I did the cyst study, I looked at the 24-hour urine. I did not see those citrate.
No, you're talking about the EHH.
Right.
One patient, not the.
Sorry about that.
That one.
Yeah, that one. Yeah, that one.
Yeah, no, I don't know.
Yeah, because we expect the cyst rate to be higher, like in patient with proximal RTA.
I didn't notice everything that had been done on it. So.
And how about in your genetic workup? Did you see like any mutation in the citrate transporter in the proximal tubule? One of the citrate transporters?
Yeah, we haven't actually run this patient on our panel yet. There's, I think this came from clinical testing, the results that we have so far.
You know, Jay, I don't remember exactly what Alan used data were about claudin 2, but I was kind of struck. I mean, you haven't said no patients with claudin 2 here.
Yeah, we added it to our panel. It wasn't on there initially. We haven't seen any clear patients with claudin 2 changes. A lot of claudin 16 and claudin 19 though.
And 14.
Yeah.
Yeah.
So the claudin 2, they have missense mutation in the Middle East population. The papers, how they published that leads to kidney stone. But we didn't find anything here, I think. Yeah.
I think they're usually biallelic as well. I mean, so most of the cases that we have are from consanguineous families with homozygous for particular changes, and nearly all of the claudin changes that we found, and they're all missense changes, I believe. Yeah.
Yes.
Really nice talk. Just a bit of an observation from, because I've seen this a couple of times. So in the context of both Fanconi syndrome and proximal RTA, it sort of leaves one with the impression that if something happens that some bulk part of proximal tubule absorption is interfered with, you end up with some kind of cystic disease. And, you know, along those same lines of reasoning, I'm wondering if maybe, you know, thinking about some of the different genes, whether they're transporters or even other interacting molecules that are just overtly associated with different types of Fanconi syndrome or things like that, might be worthwhile to include on some of the panels that you look at, because that would seem to be a theme that's tying that bit together.
Yeah, some of them we may already have on the panel. I think, you know, we're going to have more opportunities with the Mayo Biobank and looking at whole exome sequence data as well, which I think will also be a way of trying to find more novel changes.
Our next speaker is going to be Dr. Nicholas Larson. He's going to talk about genomic research opportunities in the biobank era.
Good afternoon. My name is Nick Larson. I'm a statistician, so I've been quietly judging all your p-values while I've been... So I'm a longtime collaborator of Dr. Lieske and Harris, and this presentation may be a slight change of pace. This is going to be more kind of high level, save you all of the gory details of equations and formulas, and really talk about setting the stage of research opportunities, not only globally as we've kind of entered this biobank era of population genetics research, but even locally here at Mayo Clinic with the large-scale sequencing initiatives that we are conducting through collaborations with vendors like Regeneron and Helix. So kind of broad learning objectives here with respect to the outline of this talk. I'm going to be just going over the current landscape of biobanks and genomics with more of, you know, the current GWAS-centric focus, but then really looking forward to some of the things we're really interested in conducting here at Mayo Clinic with the large amount of genomic data we've been collecting through biobanks and other sequencing based cohorts, including rare variant association analyses, Mendelian randomization, and multi-omics and data integration. But obviously, kind of as we move forward into this era of big data, there are also a lot of challenges and pitfalls that we can sort of fall into. And so I wanted to touch on a couple of those as well. So we talked a little bit about the UK Biobank earlier as one of the kind of large-scale genetic data sets that many people are familiar with. But really, we've seen a global explosion with respect to biobanking. And that's exciting for a number of reasons. First and foremost, just the ability to drive power for discovery in both rare variant analyses, but also the development of things like polygenic risk scores, and facilitating even population-based analyses of rare diseases. But also the breakdown of the Eurocentric bias that we've seen to date in population genetics research, being able to integrate more diversity into the research that we're conducting. Genomic data from biobanks and the specimens themselves, serve a variety of functions, gives us an opportunity to estimate allele frequencies in populations. It gives us a sense of haplotype structure. We use that LD information for things like genome-wide imputation or haplotype-based analyses. It also gives us the opportunity to generate our own association summary statistics as we conduct our own phenotype-based associations within biobanks themselves. But we're also seeing this transition with respect to a DNA-first era in research. And this is a term I generally use with respect to saying that these biobanks become sort of our convenient sampling frames for a lot of embedded research. And so we start with in contrast to the earlier era of GWAS, where we would build these case control studies and then do genotyping, we've kind of flipped that paradigm. And now we are operating with samples that have genetic data already available. And so talking about some of the opportunities beyond GWAS, so the first I'm going to refer to is rare variants. I know we were talking about rare variants earlier with respect to kind of monogenic diseases. I know many of you may be familiar with the figure above from Manolio et al., kind of discussing the issue of missing heritability and common complex traits. And one of those sources of potential missing heritability that we were talking about as early as 2009 was rare variants, and how these may collectively contribute to common complex diseases. The issue at the time was that in order to identify rare variants, we did sequencing, and sequencing was extremely expensive. And powered rare variant analysis requires tens of thousands of samples in order to do any kind of discovery. So even with the enthusiasm of rare variants being a plausible source of missing heritability in these complex diseases, most of the early research in rare variants was severely underpowered. However, with the onset of these large-scale biobanks and the reductions in cost with respect to next-generation sequencing, we're seeing the opportunity to actually conduct these types of analyses at a powerful level. And so the figure below is sort of a realization of the ability to do rare variant discovery. Unplanned issue. Definitely not planned. And so the figure in the bottom is a Manhattan plot that it's illustrating rare variant burden analyses in the Penn Biobank looking across all phenotypes. And so we're seeing, this analysis, which was restricted to approximately 10,000 individuals, was able to identify a number of novel gene-based associations with phenotypes. And so an example that we've already started to look into here at the Mayo Clinic with respect to the biobank is SLC34A3 and kidney stone formers. So we kind of briefly mentioned this gene earlier in terms of its association identified in the UK Biobank. So we've conducted our own, case control study through the biobank's available whole exome sequencing data. So we've gone through EHR-based high throughput phenotyping, along with variant annotation and filtering. So very similar steps to the slide that Dr. Harris had with respect to all the bioinformatics. And filtration of those variants to variants of interest. And we see that there is significant enrichment of loss of function and likely pathogenic variants within stone formers compared to controls. In addition to just genetic association analysis itself, there are a variety of other analyses that we can consider once we have all of these data available to us. So one may be less familiar with, but have heard about, is Mendelian randomization, or the so-called randomized clinical trials with genetics. so in contrast to genetics being the focus of our association analyses, rather we can leverage genetics as a surrogate for a variety of different exposures we may not directly have measured. And so this affords us to look into potential causal relationships between a variety of exposures and outcomes of interest, which may be otherwise infeasible or impractical. So an example would be alcohol consumption and disease risk. We wouldn't conduct a trial and allocate, binge drinking to one group and sobriety to another to investigate the causal relationships. And so biobanks in particular, with the rich phenotypic data available, kind of afford us to examine a lot of these types of questions. Multi-omics, I know we've talked about a lot of different molecular modalities already this morning. And so this is obviously an exciting area with respect to biobanks as well, particularly because we have access to all these biospecimens at scale that allow us to layer a variety of different molecular data types. So instead of focusing on one particular area like genomics or transcriptomics, we can have a more holistic systems biology approach to identifying mechanisms of disease and associating those with a rich variety of phenotypes. And in addition to molecular data, we obviously have the EHR linkage that's kind of driving a lot of the research that we're doing. And so that may, in the context of like an academic medical center, include imaging and pathology-based data, environmental exposures, maybe through survey collection, as well as just multivariate phenotyping itself. And so the idea is that we can kind of trace through the molecular information all the way to the phenotypic information to get a better sense of disease processes. So probably jumped up a lot of excitement, so now we could start to come back down to earth. So with biobanks, particularly those that are embedded in hospital systems, this is highly dependent on EHR linkage, and the EHR is not a research-built it's not purpose-built for research. And so there are a variety of caveats that come with that. There's lots of missingness, there's lots of bias, even phenotype, algorithms in terms of classifying individuals as cases or controls, be it through ICD codes or even through more sophisticated methods like natural language processing. They're prone to error, and that phenotype misclassification can be biased and lead to biased results. Biobanks themselves can also be highly heterogeneous. They could be prone to a variety of selection biases. They may have subcohorts in them that are enriched for certain phenotypes. These can distort associations, leading to both false positives and false negatives. And so it's not always immediately clear why data are available that you want to use. And so the figure on the right is sort of epidemiology 101 with respect to like, what is my question? What data are available to me? And will it answer the question I have? And so when we're talking about biobanks, where we're starting with data and then trying to apply questions, it's really important to think about some of these things to make sure that we are leading to valid and unbiased results, or as minimally biased as possible. And then finally, another major challenge, particularly as we are kind of entering into an era where personalized medicine is becoming more important. We have both the issues with respect to privacy, patient privacy. Obviously, with these large-scale biobanks, we're talking about genetic information, we're talking about the electronic health record, and we want to build research infrastructure around these data elements. That means that we have to make sure that we build those in ways that protect patient privacy, particularly in the context of data sharing, because we want to make sure that we leverage all information available through things like collaborative, research organizations and consortia, where the strength of the data are really going to be through things like meta-analyses. And so with that comes a lot of complexities about what the computational infrastructure looks like with respect to that apparatus. So things like federated learning, so being able to distribute analyses across multiple, distinct data sets with different systems, as well as cloud-based computing are kind of becoming more of the norm. And then alongside those things is also the concerns of exacerbating health disparities. And this circles back to some of the things I was talking about previously with respect to issues of diversity in current, genomics research. And so even, here at Mayo Clinic, the Midwest is has a very high white population, and that there are disparities with respect to continuing to drive research that is focused on one particular patient population. And so we want to make sure that everybody equitably benefits from the research findings that we are able to extract from these data and make sure that we're not perpetuating those disparities or exacerbating them in any capacity. So maybe people have heard about polygenic risk scores. That's a primary example that does not immediately translate well to a variety of different genetic backgrounds. And so a lot of additional post hoc work is necessary to make sure that they work well as, in whites as well as they do in blacks. So I think with, the reduction in costs of these technologies and the growth in infrastructure, we've sort of reached this inflection point in population genomics research. We have more power for discovery. We have improvements in sample diversity. And so personalized medicine with respect to, genetics and other, molecular data elements is really being driven by biobanks. However, there's a lot of complexity in place, and so we need new analytical methods and we need new study designs to kind of fully leverage the wealth of information that these biobanks provide, be it through multi-omic data integration, through more longitudinal-based phenotyping, or even artificial intelligence and machine learning strategies, so that we can leverage not only the connectome of all of these different data elements, but also integrating all the prior knowledge that we have about disease and molecular mechanisms to kind of further empower research. So with that, I have some very tiny text of references, which I'm happy to share. And other than that, I'm happy to answer any questions. Yes.
So I think right now, as a point in time where we are, we're very good at connecting monogenic disease to a phenotype. But what I'm wondering about is for those variants that are sort of more hypomorphic, but maybe important in stone disease, for example, the way we do our kind of which variant is important analysis right now, we would miss those, right? The things that might just increase your risk of kidney stones by a little bit.
Sure.
I'm wondering, do you think there's a way now with these like sort of massive way of looking at data that we can get better at finding those things, those smaller risk factors?
Sure, especially in the context of genetics where maybe they're so rare that association signal is just impossible to identify statistically. Yeah, so one of the ways that we would think about that is through what we would call like annotation-based fine mapping. And so we would try to take into context the characteristics of variants themselves in order to take that as prior information and upweight their likelihood that they are causal with respect to interpretation. So that could be done a number of ways. It could be done through more sophisticated analysis of the domain structure of a gene and how it corresponds to the protein. It can be done through in silico tools, like some of the tools that were in Dr. Harris's slide that attempt to, understand the impact on amino acid structure. All of those things can be factored in such that there may be variants that have one particular kind of profile, so to speak, in terms of their impact on the protein function. And then what we want to see is kind of how well does that cluster with otherwise very rare variants that statistically alone we can't identify an association, but they're sort of guilty by association with respect to that kind of similarity in impact.
So yep. The tapestry data is also available for the biobank or only the biobank data? The tapestry.
Oh, yeah. So
get more diversity kind of thing.
Correct. Yeah. So in addition to the Mayo Clinic Biobank, which is part of a larger initiative called Project Generation, which is in collaboration with Regeneron Genomics Center, Tapestry is a different sequencing initiative here at Mayo Clinic, which is sort of direct patient recruitment. And so in contrast to leveraging existing biospecimen repositories for Project Generation, Tapestry is a male kind of saliva sample to the vendor sort of study. And so it similarly has targeted around 100,000 participants through that kind of recruitment mechanism. Similar data, whole exome sequencing. And
you completed, I think.
They just reached enrollment, yes. Yeah, so they reached 100,000 last week, I believe. All right.
That's good. Thank you.
Thank you.
Next speaker is going to be Dr. Goldfarb talking about genetics and other advances in cystinuria.
Thanks a lot. Thanks for Kevin, John, for having me. Really appreciate it. I am going to talk about cystinuria. And my disclosures. Cystine stones, 1% of all stones, and 7% of stones in children. 1 out of 7,000 is the worldwide incidence of the gene. It's higher in some populations. And here is John's data for 1 year at the Mayo Clinics, which is kind of interesting. It could be that perhaps you don't order cystine stones repeatedly in people with cystinuria. People bring me their stones, and sometimes obviously I don't send them. I'm more likely to send it in a patient with a history of calcium stones. But 0.35% of the stones that were analyzed at the Mayo in 2010. So maybe somewhere in the range of 1% or less. And of course, we're talking about this molecule. That's the bad one. That is the dimer, and the poorly soluble one. It is a dimer of 2 molecules of cysteine. We say it with an extra syllable there to make sure that we make clear and make sure that your computer is not autocorrecting you and putting an E in that, because that will then reject your paper. So simply, cysteine is filtered, freely filtered by the glomerulus and then reabsorbed pretty much in S3. S3, the third segment of the proximal tubule. S1 and S2 have a low affinity cysteine transporter, which is not thought to be important. It's not perfectly molecularly characterized either, but S3 has the high affinity transport process for COL or COLA, cysteine, ornithine, arginine, and lysine. The loss of those other amino acids is not thought to have a phenotype. It's only cysteine that's poorly soluble. And there are some data demonstrating that people have a decreased plasma level, both as the result of their decreased intestinal absorption. I'll say a little bit more about that. So the genetic idea here, and Peter alluded to this already, there are 2 genes that code for 2 proteins. Those 2 proteins form a dimer in the apical membrane of the proximal tubule responsible for cysteine reabsorption. This is a neutral amino acid transporter. It's thought that neutral amino acids are exchanged for cysteine, and SLC7A9 codes for the amino acid transporter that actually does the work, and SLC3A1 codes for RBAT, that's the rat basic amino acid transporter. This molecule is not the transporter itself. It's responsible for trafficking of the transporter to the apical membrane. So without this molecule might be normal, but it won't get to the apical membrane. And if this molecule is deficient, of course, then you just have inadequate transport. So these amino acids then wind up in the urine. And it could be that either of these genes is mutated, either of these proteins is defective, and you get cystinuria. So urine normally is supposed to contain 0.4% of filtered cysteine. In cystinuria, it contains at least 100%. Some studies suggest more than the filtered cysteine, and that's not well understood exactly why that would occur. And I will point out that the exit step of the basolateral membrane is also not perfectly understood, but it's not thought. There's no evidence that there's a transporter that is defective in any cases of cystinuria. So why don't people with cystinuria lose that much cysteine? They do have an amino acid transporter of the same configuration in the intestinal membrane. So you might think that as a result of poor intestinal absorption, increased urinary excretion, that they would be cysteine deficient. And there's some evidence that they have maybe reductions of 1/3 of normal plasma cysteine levels. And that data goes back literally to the 60s and hasn't been repeated in a long time. One thought is that people are reabsorbing a variety of peptides that contain cysteine and therefore don't become absolutely cysteine deficient. But the question of whether there's a phenotype of cysteine deficiency or low plasma levels has not been clear. So the genetics of this recessive disease the most clear-cut is SLC3A1 mutations. And in these patients, here are the heterozygote parents, each of whom have one abnormal gene and one normal gene, and they have a normal amount of cysteine in the urine. The affected individual has an increased, this is micromoles, 3,000 micromoles in a 24-hour urine collection. So this, very typical autosomal recessive disease, 25% of the children would theoretically then be affected, and the heterozygote children would not be affected. They'd have normal cysteine in the urine. So this is very different than what is said to be the case of the SLC7A9 mutations. That's the actual transporter. These people, the heterozygotes, do have an increased amount of cysteine in the urine. So in this case, you can see that if normal was this unaffected individual, the heterozygotes do have an increased amount of cysteine in the urine, is not approaching the amount in the affected individual as well. But potentially then these heterozygotes perhaps could have stones. And I'll show you the data, the Mayo Clinic data, because this has always been an interesting question to me. And it's interesting because I don't have, despite seeing lots of people with cystinuria, I don't have any family where there's 2 generations of people affected. And in other words, you'd expect that if I had an individual with cystinuria, that maybe some of their parents would have had a cystine stone, or maybe some of their children would have had a cystine stone. So we know one family, Gail, that claims to have 2 generations of cystine stone formers, but I haven't seen that family personally, and I don't have any patient in my practice. Of course, this is something that we always ask about. So it's certainly conceivable that under the right circumstances, these heterozygotes could have a kidney stone if they ate a gigantic amount of animal protein, therefore acidifying their urine and increasing their methionine intake as a precursor of cysteine formation, if they were deployed in Kuwait for several months, or if they're experiencing global warming, which is happening even here in Minnesota. And so there could be circumstances where it's possible for those heterozygotes to form a stone, but I'd say that it's pretty unusual. And John and Peter and I have some data about that. So I'll show you that. So here is, I don't know if Mohammed is here. Is he here today? No, he's not. So this is an abstract that we presented at ASN last year, looking at a genotype phenotype correlation. There were 79 patients that were analyzed, and 53 individuals were biallelic for SLC3A1 mutations, and 28% biallelic for the SLC7A9 mutation. So this is interesting because there is an American study done about 20 years ago that was about 50-50 with respect to 3A1 and 7A9. There was one individual who was digenic for both of these, and this is controversial because the Spanish literature, this is Manuel Palacien's data, suggests that all of the people who are, and by the way, I should say, the genetic, it used to be type 1, 2, and 3. That's out now. SLC3A1 mutations lead to type A cystinuria, and 7A9 lead to B cystinuria. So that's how the genetics is usually classified. But there are patients that don't fit in perfectly there. So in the Spanish criteria, they actually found that everybody that was thought initially to be digenic and having one mutation in each of the 2 genes was actually AAB or ABB. So they claimed that there was no one who was A and B digenic, and that's interesting. So this one patient here in the Mayo data is interesting. 3 individuals had a single SLC3A1 mutation. Now that's interesting because, of course, I showed you that the heterozygotes are supposed to have a normal amount of cysteine in the urine. So exactly how a single SLC3A1 mutation could cause cystinuria, perhaps I would love to have these individuals, have a 24-hour urine collection and see what they really have. Do they have abnormal amounts of cysteine in the urine? And then this makes more sense. This is the SLC7A9 mutations. These are 2 patients who had that individual, and perhaps under the right circumstances, they would have an increased amount of cysteine in the urine. And as a result, despite having not the high levels associated with having 2 alleles, might have a kidney stone. So I don't know whether, Peter, do you know if there are any families where we have 2 generations of cystinuria?
I don't think so.
Yeah. So I think that's that whole idea about this being an autosomal recessive disease, but perhaps an autosomal dominant disease with incomplete penetrance. is an interesting question that we haven't really shown. Now, I'm not really a geneticist. I just know Peter Harris. And, you know, here it is also interesting that Peter has explained this to me, but I shouldn't present his data. But it is also the case that some patients who were found to have only one gene mutated at a time through other techniques, and particularly using MLPA analysis, were then found to have a variety of other issues. And so there's a couple of cases where Peter said to me that here's a patient who has one mutation, but we'll do additional analysis. And in some of these patients, they had copy number variants disclosed as a result of a later, more sophisticated analysis, right? Peter, I just want to ask you then, with respect to these patients with a single SLC3A1, do we know that those patients have had additional analysis or not? Or
we have done that other analysis, and in the 3 with just the single cases, we've actually done whole genome analysis and not found another.
Yeah. Yeah,
we could still be missing something.
Right. Yeah, we could be, but it would really be interesting, worthwhile to get to look at whether we have 24-hour urine collection data on that. I should say the other thing that I don't think. Yeah, this is from that abstract from last year, and these data are not yet published, but they're in the works. There really wasn't any clear phenotype-genotype correlation, and this has been true of European data. For the most part, it's not clear that there's any difference in severity of 3A1 and 7A9 mutations. So in terms of the amount of cysteine in the urine, age at first episode, numbers of stones, or the degree of CKD, eGFR, these are really not different. And that is pretty much in line with other databases of this sort. So in terms of genetic therapies that are being worked on, Matt Wilson is at Vanderbilt, and Matt has basically been able to knock out SLC3A1 using a target, and what they did very cleverly was also knock out a tyrosinase, which leads the animals to be albino. And the result is that you can tell which animals are going to have the manipulation. They'll be black. If anything goes wrong, they will turn white. And so they do have a genetic albino cystinuria type A mouse. And they're going to use that to try different techniques of genetic therapy. So this is Matt, and here they're using a variety of techniques to try to get gene transfer to the kidney directly to phenotypically correct cystinuria in a mouse model. And the goal is to use this gene transfer processes to express the cysteine transporter in the proximal tubule cell. So right now they have both viral and non-viral mediated vectors to carry the genes to the cells. And of course, because the kidney is so technically, or I should say anatomically complex, how exactly do you get these cells to the proximal tubule? Currently, they're doing this by injecting their vectors in the tail vein of the animal. And none of my patients have a tail vein. And so, Matt, I have expressed my skepticism of this, but Matt is injecting into the tail vein and somehow then expresses in the tubule the genes. So how the gene exactly gets to the right place is not clear. And they are using a variety of viral vectors to try to make it better and get it to the right place. You know, it's more complicated to try to do this through a renal vein, as an example. They've tried a variety of techniques with different viral vectors. Matt gave us a nice talk. We had him for renal grand rounds at NYU last year. He's written a couple of papers on this. Has some grants pending. So this is basically, again, trying nanoparticles, as we've heard about today, as a way of perhaps getting the genes targeted to the proximal tubule. And this is a paper that he had written for Current Opinion. This is something that some of you know Mylene Perot at Synlogic. And what they did was take a methionine degrading, made a bacteria into a methionine degrading gene. And the goal there would be to degrade methionine and therefore wind up with less cysteine in the urine as a result. This is a treatment for the patients who have cystinuria, homocystinuria. Most of those patients have a genetic abnormality in cystathionine beta synthase, and they wind up with an increase in homocysteine because they don't degrade the homocysteine. They get homocystinuria, accelerated atherosclerosis, and death at a relatively early age. So their goal in treating cystinuria was perhaps this would also work as a way of degrading methionine, showing that that was useful as a precursor. If you could knock out methionine, you would develop less cystinuria in people who don't have a mutation in cystathionine beta synthase. So in order to look at this, they first used this in live cells. These are the bacteria that was able to degrade methionine. And you can basically see that with an oral load of methionine, it blunts the appearance of plasma methionine and total homocysteine in non-human primates and in cells in vitro as well. So they had basically announced this in last November, that their data had shown that this reduces plasma methionine by having a bacteria in the GI tract to degrade methionine. And so they are progressing with this as a treatment for homocystinuria. So in order to then test this bacteria, they worked with our friend Amric, who has his own knockout model of cystinuria. And this is really an interesting study that has clinical significance for patients with cystinuria. So they took Sahota's model of cystinuria, and they basically, has nothing to do with the bacteria. They simply put the animals on a low methionine diet. And this, of course, there's long-standing data suggesting that being on a low-protein diet would lead to less cystine stones for two reasons. One, again, less methionine means less production of cystine, and two, eating less animal protein means eating less protons, less acid, leading to a higher urine pH and a need for less alkali in order to keep the urine pH high. So they basically looked at these animals on two different methionine doses and demonstrated here, this is on the y-axis, this is the changes in methionine on different diets. So you can see that on the low, this is the higher methionine in blue. That's the higher methionine diet. So there was, the animals actually ate more and had a greater body weight change. But the important thing was the development of kidney stones. So here on the methionine diet, there's a shift to the left here. This is 0.5%. And this was a shift of stones to the right, more stones, greater stones on the high methionine diet. These are mislabeled. I'm sorry. This is the high methionine diet. There were more stones, bigger stones, and a shift to the right. This is the first study that I'm aware of in an animal model showing that higher animal protein led to more kidney stones, bigger kidney stones as well. So what it suggests, this was kind of a proof of concept study for Synlogic to then show that if you change the methionine diet, you would change the prevalence of kidney stones, the number and size. Therefore, their methionine degrading bacteria would potentially be useful. The last thing that I want to talk about is Paul Goodyear's suggestion of what he called a no-nonsense approach to hereditary kidney disease. 10% of genetic diseases are caused by nonsense mutations, which then, and this was basically that Hildebrandt has estimated that there's over 50 monogenic kidney diseases that where at least some part of those diseases are related to nonsense mutations that lead to nonsense-mediated mRNA decay. And so Paul basically had shown that there is a series of aminoglycoside antibiotics. These are not antibiotics that turn out to be nephrotoxic, as we usually think about aminoglycosides, that bind to an area on the decoding site of the small subunit of mammalian ribosomes. And this leads to the ability to bridge the defect in the nonsense mutation and allow the translational machinery to insert the intended amino acid. So the potential would be to use these aminoglycosides for patients with nonsense mutations. So what this led to was that Paul and I had a protocol where the goal was to take the patients that we had identified as having nonsense mutations, and Peter helped us with this, of the many patients we had, more than 80 or 90 patients with cystinuria genotype, we demonstrated that there were 10 patients with nonsense mutations who lived in the New York area. So the plan was to get those patients to come to New York on a given day, give us a urine sample, a 24-hour urine collection would be preferred, or bigger urine sample that would then Paul and Fatima, we're going to, here's Fatima, Paul's a postdoc at that time in Paul's lab, we're going to take the urine back to Montreal, a 9-hour drive, plate the cells in the urine, potentially a lot of proximal tubule cells, and plate them in order to demonstrate that, one, they had decreased cysteine uptake in vitro, in cell culture, then treat them with aminoglycoside that is particularly known to prevent nonsense mutations being expressed, sort of bridging over them. And then there was this virus that came around, and that prevented our plan to drive back and forth between New York and Montreal. And so we do have a protocol approved to do this, and it's something that Paul and I have talked about, whether we can revive this idea and actually test this in cells from patients with cystinuria. So that's kind of interesting. The last thing I'll talk about is, we have been working since like 2008 particularly with Amaryx sahota and Long Hu on a group of cysteine diamides that are potentially cysteine crystallization inhibitors. These are drugs that are highly soluble, very well tolerated, and appear in the urine. The problem is that the company that has licensed the patents from NYU and Rutgers has basically not been able to raise money for it. So Pharma Christo put this press release out in 2018. That's the last time I heard from them. So what's going to happen with that is not clear. And then the last thing I'll talk about is, of course, this is all of us are asked continually about alpha lipoic acid treatment, preventing cysteine stones in a mouse model of cystinuria, right? So this is a paper that, Matt, you ask me about this later. Okay, so this is alpha lipoic acid was shown to be useful by Tom Chi and Marshall Stoller in a paper. And they have for 5 years now had an R01 to study this in humans. But they were able to do this with Sahota's mice and demonstrate that alpha lipoic acid was significantly able to inhibit stone volume production. But for 5 years now, Marshall and Tom have not presented any data, and I'm not sure why. And when I ask them, they wave their hands around in this interesting way. So I am not able to say whether or not we're going to get from UCSF some data on alpha lipoic acid. Of interest, it was effective in people, it was effective in mice. John Asplund was not able to detect lipoic acid in the urine and was not able to show that it inhibited cysteine crystallization in vitro. So exactly what it was doing, how it was working, whether it's being metabolized to something that actually is capable of inhibiting cysteine crystallization, it's not really clear. And there are no human data published yet. Thanks for your time.
Question: have you seen increased incidence of kidney cysts in cystinuria patients?
Sorry, have it?
Cysts. Have you seen
cysts?
More cysts in cystinuria patients?
That's a good, interesting question. A non-calcium based proximal tubular disease, could it cause cysts? I'm not familiar with that, and it doesn't ring a bell right now, but we could certainly look at that, Christian.
Thank you.
Thank you.
We have a great talk from one of our former T32 PhD postdocs. Carmen Reynolds is going to talk about some interesting things she's done with FGF23, vitamin D, and Dent.
All right. Okay, so this is the third time we've seen this. So it's the background of Dent disease. It's hereditary, X-linked, CLC, N5, OCRL. It's the third time. Hopefully, you guys have caught on already. And so we have some clinical characteristics. Again, this is just a repeat of what other people have talked about before. I highlighted hypercalciuria because it's really the central focus of my talk. Okay, so here's where we go a little bit off the avenue. And so calcium is here in existence in this balance between blood and bone. And it's in this collaboration with phosphorus. And they're maintained by an excretion into the urine. And this is all regulated through this triad of hormones, including fibroblast growth factor 23, parathyroid hormone, and 1,25-dihydroxyvitamin D, or calcitriol. I have it just vitamin D here. So we asked the question, in Dent disease patients, can phosphate supplement promote this FGF23 PTH vitamin D triad and increase calcium reabsorption and minimize the hypercalciuria? So this was a study that was done quite a while ago. And it was a group of Dent disease patients who all happened to have the CLCN5 variant. Of course, they're all males, and we had 10 of them in total. The average age was 27 years old, ranging from 19 to 54. And then we had another group to compare with this, which had history of kidney stones, the stone formers group. Of course, they're all male to compare with the Dent disease participants, and they were only 9. And their age was a bit older at 55 years old and ranging from 29 to 69. So the study was based on this short intervention where they came in for a first visit, gave blood and urine, and for clinical analysis, and then had a 2-week intervention where they had the supplement for a phosphate supplement for 2 weeks, and then came back for another collection of blood and urine. Sorry. Better. Thank you. Okay, so let's start off with some of the data. And right off the beginning, we have this stone group and their serum phosphorus. Of course, they're getting serum intake, so that we'll measure the serum phosphorus first. So in the stone formers, they were all within clinical range, and there was really no difference between visit 1 to visit 2. And then in the Dent disease patients, they were a little bit more variable, and there again was no change. So if we had seen a little bit of an increase in serum phosphorus, there would have led to an increase in FGF23. And what we see is there's a almost increase in FGF23 in the Dent group. And so if this were indeed an increase in FGF23, it would go on to reduce the sodium phosphate transporter in the nephron and then increase urine phosphorus. And indeed, we do see this where there's a significant increase in the urine phosphorus in the Dent group and not so much in the stone group. So then switching paths to the calcium side of the story, we see the stone group, of course, has normal serum calcium. The Dent group has a much larger variation in the serum calciums, but that's pretty well known. So slight decreases in serum calcium lead to an increase in parathyroid hormone. So next we measured parathyroid hormone, and we do see a trending increase in the parathyroid hormone in the Dent group. And increases in parathyroid hormone lead to a decrease in the urine calcium. And indeed, we see a decrease in the urine calcium in both the Dent group and the stone group. And I'm going to point out that the stone group, we hadn't seen any significant changes in the hormones that would impact the urine calcium. So right here we have the effects of this phosphate supplement, including an increased urine phosphate, possibly because of the increase in FGF23, and then a decrease in the urine calcium owing to a small increase in the PTH, right? But what about vitamin D? We still haven't quite come across any of that data yet. So vitamin D status is measured by the 25-hydroxy vitamin D. And in both of the groups, the stone group and the Dent group, they're all in really good normal ranges, and there's not a particular amount of deficient individuals. So one of the fates of 25-hydroxy vitamin D is to have the 1-alpha hydroxylate added to it by CYP27B1. And so this is increased by PTH, and in the counter sense, it's decreased by FGF23. And so our 125 values, we see a decrease in the stone group, but not so much in the Dent group. So 125 increases expression of CYP24A1, and that adds a 24 hydroxylase to 25 hydroxy vitamin D, and this is regulated also by FGF23. And so when we measure that, we see there's a trending increase in the Dent group and no changes in the stone group. But the actual clinical interpretation of 25 dihydroxy vitamin D is to look at it in the ratio with 25, where 25 is the substrate divided by 24-25, which is at much lower concentrations as the product. And this is assessing the activity of CYP24A1, which is regulated by 125 and then FGF, as we mentioned. So in the stone group, we see that they're all falling within the normal range. If things are above the normal range, that's indication as a diagnostic criteria for infantile hypercalcemia, which is that mutations or variants in CYP24A1, be it heterozygous or homozygous. And so here is where things get a little bit different. In Dent disease, we have people who are much higher in this ratio. And so we still have some that are in the normal ratio, about half and half in this group. And so I decided to go through an analysis where I bin those into different parts where it's a normal function of CYP24 and low function of CYP24. And so this is what we come up with. When we go back and look at the actual concentrations of the 24-25D, we see that they're much different between the normal and the low activity groups. And the normal group has a bit of an increase, and there's no change in the low activity group. So now let's go back and look at our data with the binning included between the normal and low. Before we'd seen that the Dent group had a little bit of an increase in FGF23, and when we separate them out, first of all, we see that the low group has lower FGF23 than the normal group in that visit 1, and they have a significant increase with the phosphate supplement. And so that's really driving that statistic that we'd seen before. In urine phosphorus, we had seen that there was a significant increase. And when we separate them out, they have the lower activity ones have a bit of a lower urine phosphorus to begin with. And they had a much larger increase in the urine phosphorus than the normal group. So then parathyroid hormone, we'd seen a trending increase. And we see that becomes significant, and that low group is the one that's driving that statistic right there. 125, you would expect that in that group, you'd expect the 125 to be increasing because parathyroid increases, but we see no change. More importantly, we actually see that the low CYP24 activity group had really high 125 vitamin D levels. And when we look at the urine calcium, we'd seen a decrease, and this decrease was really driven by the low group compared to the normal group. Not only that, but the low group definitely had hypercalciuria. And so this, because we didn't see any sort of increase in 125 dihydroxy vitamin D. In fact, we'd seen a decrease in the stone formers group. So there's really no evidence that 125 would be driving that urine calcium to go down. And so it's suspected that the increase in FGF23 instead of the 125 is what's driving an increase in TRPV5, which is the calcium transporter. So the key findings from this is that we have come up with a definition or defined characteristics for the Dent patients with low 24-25 D activity, where we have lower FGF23, we have lower urine phosphorus, much higher 125 dihydroxyvitamin D, higher urine calcium, and they had that enhanced response to the phosphate supplement. And because of this group, we were able to come up with more of a stronger theme of what's happening because of the phosphate supplement. And that's FGF is increasing. It's increasing the urine phosphate through that sodium phosphate transporter. It's decreasing urine calcium through increased TRPV5. There's a little bit of parathyroid hormone going up. And because of that parathyroid hormone going up, and the counter effects of FGF23 on CYP27B1, we end up with no net change in 125 dihydroxyvitamin D. In fact, there's a decrease in that stone formers group. So that would suggest, again, that we're leaning toward the FGF23 regulating all of this here. So here's my conclusions, is that oral phosphate supplement decreases urine calcium in Dent patients, and this is really primarily through FGF23. Not only that, but we see this elevated 25 to 24-25 ratio, and that's corresponding to high hypercalciuria and really high 125D levels. And not only that, but these people are having enhanced responses to the phosphate supplement. So maybe an assay like this could drive your treatment plan for a Dent disease patient. But what I asked also is what else is going to correlate with these ratios? We see hypercalciuria correlating. Is there nephrocalcinosis, nephrolithiasis, metabolic bone disease? There's a lot of evidence where 24-25 is needed for bone callus formation, bone fracture repair. Are these ratios going to indicate anything with progressive renal failure? And then how is it regulated? Are we able to make it go down a little bit by more phosphate supplement, by a longer term phosphate supplement? So these are all questions that have come up from just this little quick study. With that, I'd like to thank Dr. Lieske and then the others involved in the study, Abir Lasik, Dr. Kumar, study coordinator, Barb Seid, and Zepha Haskik, and then Dr. Romero.
We're pleased to have you. Dr. Barbara Cellini is going to talk about molecular look on AGT variants.
Thank you, John, for the kind invitation. I will give you a quick view on the molecular pathogenesis of primary hyperoxaluria type 1, looking on AGT variants. So primary hyperoxaluria or type 1 or PH1 is the most severe form of PH, and the disease is due to the deficit of a peroxisomal enzyme named alanine glyoxylate aminotransferase or AGT. The deficit of these enzymes leads to glyoxylate accumulation and in the cytosol to its oxidation to oxalate by lactate dehydrogenase, and is this huge accumulation of endogenous oxalate that leads to the formation of calcium oxalate kidney stones. The only pharmacological treatment available for PH1 patients is vitamin B6, which is the precursor of the AGT coenzyme. It is effective only in a minority of patients, and unresponsive patients should rely on transplantation procedures or to new biological drugs based on the silencing RNA technology. This is the structure of AGT1, and the enzyme is a tight homodimer. Please look to the N-terminus that wraps over the neighboring subunit. And the enzyme, as I said before, utilizes vitamin B6 as coenzyme, and in particular, the coenzyme is pyridoxal phosphate or PLP. If we look at the ClinVar database, we can see that more than 300 mutations associated with the disease are listed until now. And the vast majority of them are missense mutations. They are spread over the sequence, as you can see. So that's indicating that a variety of mechanisms can lead to the deficit of the protein. So one of the main aims of the research of our group has been the use of molecular approaches to study and to try to understand the pathogenesis of the disease. So we asked three main questions that I will try to afford in these few minutes. The first one is, what are the molecular effects of disease-associated mutations? So we can basically distinguish the variants, missense variants associated with PH1 in two main categories. The first one is that of mutation causing a catalytic defect. This means that the protein, the mutated protein, is correctly folded, but the transaminase reaction does not occur because the protein has a kinetic problem. So, for example, here you have a list of some of these kind of variants. The variant can have a reduced K-cut, so the transamination reaction, the rate is very low, and this can be also associated sometimes with a reduced affinity for the PLP coenzyme. And most of these mutations involve residues located at the active site or in the surrounding region. But this is not the main problem in PH1, because most of the mutations associated with the disease rather lead to a folding defect. This means that the protein is intrinsically active, but the folding is so inefficient that the level of functional protein in the cell are very low, so you have a deficit. And the downstream effects of folding can be different, including premature degradation, aggregation, or in some specific cases, the mitochondrial mistargeting of the protein. One example of a folding defect leading to PH1 is the mutation of glycine 41. Glycine 41 is located at the N-terminus of the protein, and our group, using in silico approaches, was able to predict that its mutation to the bulky residue arginine would lead to some structural changes of the N-terminus that becomes more unstable, more flexible, let's say. So we confirmed this hypothesis by protein studies, showing that the variant is catalytically active, but its melting temperature is reduced, and the variant is prone to aggregate, thus indicating a structural defect. And these data were also confirmed in cellular studies, where we found a strongly reduced levels of active protein in the cells. And in these studies, we also found, very interestingly, that the levels of active protein increase upon the administration of pyridoxine or PN. Please consider that PN is the form of vitamin B6 used in clinics. So we predicted at molecular level that patients could be responsive to vitamin B6. And this was actually confirmed by a study from the John Lieske group later on, who confirmed that these patients were indeed responsive to B6. Well, but in order to understand PH1 pathogenesis, looking to these two categories of mutations is not enough, because the pathogenesis is more complicated due to the presence of polymorphic mutations. It is known that the AGXT gene encoding AGT is present in humans in two polymorphic forms they named major allele and minor allele. The minor allele differs from the major one for the mutation of proline 11 to leucine and isoleucine 340 to methionine. These two mutations are not pathogenic. This means that people expressing the minor allele do not display the disease. However, if we look to the protein, we can see, and this is the structure of the minor allele that our group solved a few in recent last year, actually. And when we solved the structure, we found that some regions of AGT in the minor allele are more flexible. They are indicated in red here. And this is responsible for the lower stability of the protein, which is we can be seen both in at protein level and in cellular systems. So the minor allele influence the effects of pathogenic mutation? Well, the answer is yes, and we were able to demonstrate this synergism. In particular for the mutation of isoleucine 56 in collaboration with our colleague at the University of Torino. So the mutation of isoleucine 56 to an asparagine residue was known to be pathogenic on both allergic background. However, the disease seemed to be more severe when associated with the minor allele. And since isoleucine 56 and proline 11 are spatially interconnected in AGT, we wonder if there is any crosstalk between these two mutations that could explain the different behavior on the two allelic background. So we studied these 4 forms of the protein at different levels, but today I just show you the data in cells. Here on the left, you can see protein levels. On the right, you can see the specific activity. You can see that the mutation of isoleucine to asparagine strongly reduces protein level and specific activity, but the effects are stronger when the mutation is on the background of the minor allele. So the protein is almost undetectable and the activity is even lower. But when we treated, we were even more surprised when we treated cells with vitamin B6, because we found that when the mutation is on the major allele background, the variant is completely responsive to vitamin B6, as shown by the recovery of the protein and of the activity. On the other hand, when the mutation is on the minor allele background, no responsiveness can be seen, and at both protein and specific activity levels. So the last question that we try to answer very recently is the possibility to use a molecular approaches or also to inform on the possible pathogenicity of newly identified mutations. And we were contacted by Dr. Takayama in Japan because Dr. Takayama had a patient who had the clinical features of a PH1 patient. However, when they performed the genetic analysis, they found a pathogenic mutation in one allele and a variant of unknown significance on the other allele, the glycine 365 to cysteine mutation. For that reason, this patient was diagnosed as unclassifiable PH, and for that reason was excluded by a clinical trial. So we try to understand using our molecular approaches which are the effect of this mutation and if this mutation could be pathogenic or not. Glycine 365 is located in the region surrounding the active site. And again, by combining protein and cellular studies, we could show that its mutation to cysteine not only strongly impairs the catalysis, so transaminase activity is very low, but also the affinity for the coenzyme is strongly decreased. And when we express these mutants in a cellular disease model, we found that the activity is almost undetectable because the level were similar to the basic level of the cells. So let's say to the specific levels of transaminase. And the protein level were very low, and most of the protein was recovered on the insoluble fraction. So using our approach, we were able to confirm the pathogenicity of the glycine 365 to cysteine mutation. So to conclude, what can we do using a molecular approach to study PH1 pathogenesis? We can better define the effects of pathogenic mutation at enzyme level, and this could also prompt not only to predict the responsiveness to treatments available, but also to develop new treatments, but it is not the focus of my presentation today. Second, we can understand if there is any synergism between pathogenic and polymorphic mutation. And finally, we can help in defining the pathogenicity of newly identified mutations. So finally, let me thank all the members of the group in Perugia who participated to these studies. And if you want to follow our activities, follow us on Twitter. This is our account. And also thanks to the collaborators who participate to the studies I showed you, and also to our financial supporters. And thank you for your attention.
Thanks. I think we have time for a question or two if anybody has any. So what is your capacity for doing that kind of work? So if we had a VUS, how long does it take to get to the final answer like you just showed us?
Yeah, that's a tricky question, John, because it depends. It is difficult to predict. So the platform is set up using different approaches, but sometimes some mutations are very tricky. So it is with proteins, it is always difficult to make predictions, but so if you have the platform ongoing a few months, and you can test a few mutations in parallel. Sometimes we have a study using a similar approach as I showed you today, using different mutations in parallel. But please consider that we can only provide suggestions that obviously should be paired with clinical data, and so.
Sure. No, that's helpful. Anything else? All right. Well, thanks very much. So we're very pleased to have Dr. Stephen Scheinman here. He's going to give a very, I would say, first-person account in a lot of ways about Dent disease and Oliver Wrong, and I think it'll be very exciting. So take it away.
Thanks very much, John. And first of all, I want to acknowledge two people who are not part of the consortium who are on the Zoom call, and I'm honored that they are. One is Anthony Norden, who was Oliver Wrong's colleague for for all of the work that he did on Dent disease and much else. Anthony was responsible for the measurements, for pioneering the measurements of low molecular weight proteins in proximal tubular disorders, and was a very close collaborator with Oliver. And Michaela Wrong, who is Oliver Wrong's daughter, who is an internationally renowned journalist and author, foreign correspondent for the Financial Times, and someone of whom I know from direct information Oliver was very proud of. So I'm really pleased that they're here with us. You all know all about Dent disease and know about Oliver Wrong's contributions to Dent disease. And so but I'm not sure that I can assume that you are familiar with his many other contributions to nephrology. Of the 135 papers that he published during his lifetime, only about a dozen were on Dent disease. His contributions are far-ranging and reflect, I think, the impressive nature of his intellect, his curiosity, his relationship to his patients, and his character. Okay. Oliver Wrong was born in 1925 at Magdalen College, Oxford, which is an institution with which his family had a long relationship, and he spent much of his childhood living there. He died in 2012 at the age of 87 at University College Hospital, London, where he had spent many of the productive years of his career. Magdalen College is, and some of you who've been to Oxford may recognize where he's standing in this picture. He's standing right near the Radcliffe camera. Magdalen College is a beautiful, huge part of Oxford. It is ancient, and it is associated with many Nobel Prize winners in medicine. Those include Sir Howard Florey, the discoverer of penicillin, Sir Peter Medawar for his work on immune tolerance and transplantation, and a person that some of you may know personally, Sir Peter Radcliffe, who just a few years ago won the Nobel for his work on cellular oxygen sensing. Oliver himself grew up on the grounds of Magdalen, which overlooks this huge and beautiful deer park, and past which one of the two great rivers of Oxford, the Char, passes with its punters. His death at University College Hospital was the hospital was a much larger institution than it was when he first started working there. So there is a wholeness to his life, I think, represented by his pedigree. Oliver Wrong descended from a distinguished intellectual family that included, among many other accomplished relatives, 2 Nobel Prize winners in his family. One is Dorothy Hodgkin, structural biologist who won the Nobel in 1964 for her work on X-ray crystallography and the structures of cholesterol, penicillin, vitamin B12, and later insulin. And Alan Hodgkin, the physiologist and biophysicist who won the Nobel in 1963 for his work on giant squid axons that led to the elucidation of the mechanism of the action potential. So these are some pretty important fundamental discoveries in his family. This is the wedding photo of Oliver Wrong's parents. Both his parents, obviously standing in the center, both of them were historians. His father, Murray Wrong, was Canadian. He taught modern history at Magdalen and in fact, was vice president of Magdalen College. His mother, Rosalind Grace Smith, also taught history at Oxford, was a member of a very prominent academic family herself. Her father, A.L. Smith, who was the master of Balliol College at Oxford. They were married in 1915, and so you can see the World War I era garb. Murray's father, so Oliver Wrong's father was Murray on the right, and his father, George Wrong, was himself a distinguished historian. He was from a very prominent Canadian family that included Edward Blake, the Premier of Ontario, who was Murray's maternal grandfather, so Oliver's great-grandfather, and a family friend of the Wrong family in Toronto was Sir William Osler, who would vacation with them. And when Murray Wrong first moved from Canada to Oxford to be an undergraduate, he lived with the Oslers until he found other digs. So they were close. He was also Osler was Murray Wrong's physician. And Murray Wrong, in fact, suffered from rheumatic heart disease and died at the age of 38. At that time, that he left 6 children to his widow, and the family, the children were split up. 3 of them were sent to Canada to live with their grandfather and his family. So Oliver, from the age of 6 to the age of 13, lived in Canada with George Wrong, the distinguished historian. Came back to England at the age of 13 for school, and then went on to study at Magdalen College, where he got his medical degree and clinical training at the Radcliffe Infirmary in Oxford. And then in 1948, he did his military service in the Royal Army Medical Corps in Singapore and Malaysia, which was an experience that informed one of the most significant threads of his research interests. He then did a residency at Toronto General Hospital and a research fellowship with Fuller Albright and Alex Leaf at the Mass General, which is where he really developed his lifelong interest in salt and water metabolism. His first academic faculty position in 1954 was at the Manchester Royal Infirmary, and 3 years later he moved to the University College Hospital. He shortly thereafter became senior lecturer in nephrology at the Royal Postgraduate Medical School at the Hammersmith Hospital. And in 1969, had a brief experience, a 3-year experience as a department chair. He became chair of medicine at the Dundee Hospital in Scotland. And 3 years was more than enough time to convince Professor Wrong that his love was not academic administration, but rather was patients and research. And so he came back in 1972 when the opportunity was presented to him to succeed his colleague Charles Dent as professor of medicine at the Royal Postgraduate Medical School at the Hammersmith. And so that's where he remained for the remainder of his active career, I put that in quotes because he is officially retired in 1990, 22 years before he died. But he spent his time in retirement working basically without remuneration at the University College Hospital, where he had spent so many of his productive years. In retirement, he was in many ways more productive than he had even been during his working years. So I'm going to talk about several themes of his work, starting with one of his most significant contributions, which was at the very beginning of his career, his monumental work on the excretion of acid in renal disease. It was published in the Quarterly Journal of Medicine in 1959. It's really a seminal paper because it defined the short acid load, which we still use today as the classic test for urinary acidification. Not only did he provide what Robert Owen has called a detailed and descriptive tour de force of acid handling by the kidney using this test, but he defined the criterion for renal tubular acidosis that we still use today, which is a urinary pH of 5.3, inability to acidify below that. This was a 54-page article, and he wrote, excretion of acid is impaired in renal disease and acidosis frequently results, but there is little information as to how the chief variables of acid excretion are affected in different forms of renal disease. And he answered that pretty definitively in this first paper. First of all, he defined the normal ranges for urinary pH, ammonium excretion, and titratable acid excretion across the range of urinary pHs. And so we see on the left here the normal ranges that this study defines, and the well-established observation that we're all familiar with, that ammonium excretion increases as the urine becomes more acid. In chronic renal failure, he taught us that the problem is not the ability to acidify the urine. The nephrons that remain in chronic renal failure may become fewer and fewer, but each one is still able to achieve a low urinary pH. The problem is that the kidneys have lost sufficient mass that they cannot excrete a sufficient amount of buffer. And so ammonium excretion impairment is the major reason for acidosis in chronic renal failure, as contrasted with renal tubular acidosis. And in fact, the data in this paper gave us the information that we need to define what we now know. These terms weren't used in the 50s as distal renal tubular acidosis or classic renal tubular acidosis. All of his patients in this study met those criteria, and as we see here, none of them were able to acidify below a pH of 5.3. This paper is where that criterion comes from, but they are still able to excrete ammonium in adequate amounts because renal mass nephron number is not impaired. And so this explained to Oliver, it's another insight that emerges for the first time from this paper, why some people can have an impaired ability to acidify their urine and not develop systemic acidosis. It's because they do have the ability to raise their ammonium excretion. So seminal paper. Another interest, one might even say the word interest is not quite strong enough, is his interest in the role of the intestine in acid-base metabolism and electrolyte metabolism. And so he wrote this paper in 1965 when he was back in London on the electrolyte content of feces. And he was disappointed in the medical establishment at large that they had a distaste for handling feces and would not study it. And so he wrote famously, stool is the Cinderella of electrolyte studies. It's a fascinating material. It contains 70% or more water. It's also known to contain many electrolytes, which are usually identified after ashing in a muffled furnace, a procedure which destroys all organic material and tells us nothing about the physical state of the inorganic constituents. And so in this study, in this and subsequent studies, Oliver used a technique that he perfected over the course of 5,000 of these cellulose tubes. So in the middle of this slide, well, on the right-hand side, you see a clear cellulose tube that could be rolled up, put into a capsule, and swallowed. And he was apparently relentless in getting his colleagues and his friends and his family to swallow these capsules, although nobody swallowed as many capsules as Oliver himself did. In fact, Michaela, his daughter, who is on this call, and she can correct me on this or anything else, she and Anthony are my conscience in this talk, will say that she grew up with the impression that this was what normal families did at breakfast. You swallow a capsule, and then when it emerges later in the day, it is sent off for study. So with all of this exertion, what was learned? Well, actually quite a bit. First of all, he defined the normal range for electrolyte composition of the final stool. And here you just have a table, I'm not going to go through it, of the normal electrolyte composition. And with that, you can study abnormal circumstances. So for example, primary aldosteronism. Here's a patient, pre and post resection of a tumor with primary aldosteronism. So pre-op, this patient has excessive urinary potassium and very low urinary sodium, consistent with, obviously, a vanishingly low sodium-potassium ratio, consistent with the known effects of aldosterone. And then post-operatively, you can see that these numbers correct to the normal range. The sodium-potassium ratio is completely normal. So confirming the role of aldosterone in colonic electrolyte transport. Another study using this method looked at the effect of lactulose on fecal pH and ammonia concentration, and the normal level, which he had defined in a previous study, is shown by the dashed line. And you can see that when people take ammonia, two things happen. When people take lactulose, two things happen. One is that their ammonium excretion in the stool is lower than normal. And the second is that the relationship between pH and stool ammonia disappears, overwhelmed by the osmotic effect of the lactulose. And so insights like this are made possible by Oliver's scientific approach to studying stool electrolytes. And then in 1990, and I'm obviously breezing through this because time is limited, in 1990, he retires. He retired by moving, he and his wife moved to central London so that he could walk to the Middlesex Hospital and to the libraries in central London, and spend more time on his work. And so we're all familiar with his work on Dent disease. And he did us the great service of naming this disease after Charles Dent. Now, I think we all recognize that Charles Dent's original 1964 description did not include that these patients had a family history. In fact, Charles Dent believed himself that this was nephrotoxicity and not familial. He didn't recognize a family history. He didn't follow them long enough to determine whether they developed renal failure, and kidney stones were not part of his description. So I think most people would not have named this disease for Charles Dent. I think it would have been much more appropriate to name this disease for Oliver Wrong, whose paper, when it did come out 30 years later, he's retired and he finally can put everything together, and has been following Charles Dent's patients and other patients for 30 years and more. When that paper finally came out in the Quarterly Journal of Medicine, and as is typical of Oliver's papers and Quarterly Journal papers in general, it was very thorough. And the title of the disease provides a full phenotypic description of Dent disease, a familial proximal renal tubular syndrome with low molecular proteinuria, hypercalciuria, nephrocalcinosis, metabolic bone disease, progressive renal failure, and a marked male predominance. So there are a couple of reasons why he wanted to call it Dent disease. Anthony Norden has said that it's not correct to call Charles Dent his mentor. They were colleagues, they worked together. He succeeded him in the professorship, but I think that Oliver did want to honor him, A. B, it's a nice pithy name, Dent disease. And C, he was self-effacing and concerned that if it became named for wrong, if it was wrong disease, you can imagine the jokes on the wards. And he was conscious of this. And so he selflessly encouraged everybody to call it Dent disease, which is what we call it today, although at the very least, we might call it Dent wrong disease. And there have been some papers with that title. And so Charles Dent's two patients, we now know the first one had mutation in CLC5, but the second one did not. And when that puzzle bothered Oliver, and he was convinced there was another gene, and every time we would talk about it, he would point out that this child was, in his phrase, mentally backward, had an IQ of 65. I'm sure, Oliver would say, I'm sure that's a clue to what is going on. And in fact, we now know that case number 2 had mutation in OCRL1, which of course, he's lucky he's only mentally backward and not much more severely compromised. And so one important reason why Oliver was so good at collecting specimens and collecting data and collecting family histories from his patients is that he had a deep and strong relationship with them. They could call him at any hour of the day or night. He could ask them anything, and he'd say, "Could you do a 24-hour urine, and I'll swing by in 2 days and collect it?" And he would drive throughout Southern England doing this. And so here I am with him on one of those trips, but Anthony on the phone accompanied him on many more of those trips. So this picture, Oliver is the second from the left, the most distinguished of the people in the picture, standing with Anthony Norden and Terry Feast, his two co-authors on that 1994 description of Dent disease. Thomas Jentzsch in the middle, who described the CLC family of chloride transporters, and Raj Thacker, who has made so many contributions to our molecular understanding of Dent disease. Then there's this interloper here. His other deep interest in retirement, and really his first publications on this didn't come out until 7 or 8 years into retirement, were on the band 3 anion exchanger and Southeast Asian ovalocytosis. Now remember that he had spent his military service in Southeast Asia. And the band 3 protein is the most abundant of the proteins in the red cell membrane. It is a structural protein in the red cell membrane, and is important in connecting the cell membrane with the cytoskeleton. And it was well known that mutations in the band 3 protein were responsible for what was first called elliptocytosis and is now called ovalocytosis in Southeast Asia, and well recognized to be endemic in Southeast Asia because, like the thalassemias and sickle cell disease, it conveys a resistance to the falciparum malaria parasite. But Oliver, with his colleagues in this paper from 1987, were the first to demonstrate that a mutation in that gene is responsible for distal renal tubular acidosis. This was in patients in England. They were not from Southeast Asia. They did not have ovalocytosis, but it was the same gene. And so pursuing that gene, they subsequently demonstrated that a recessive form of renal tubular acidosis occurred in Southeast Asia in patients with ovalocytosis. So he wrote, with colleagues, he wrote a number of patients on this over the last 15 years of his life. The cells, you can see the typical ovalocytes of the disease. He defined specific mutations and the geography of those mutations. And so the BAN3 protein is the chloride bicarbonate anion exchanger that exists on the basolateral surface of the alpha intercalated cells of the collecting duct, so that the bicarbonate produced by the activity of carbonic anhydrase can be excreted from the cells. Impairment here will obviously impair the ability of carbonic anhydrase to continue generating acid to be secreted. This is from Oliver's, this diagram is from Oliver's paper. His work, this published in 2002, defined the association between those specific mutations in this gene and their geographic location throughout Southeast Asia. And then 10 years later, in a paper in the Quarterly Journal of Medicine, appropriately, when he expanded significantly on this, you can see that the experience has broadened geographically across Southeast Asia. This paper, from which that map comes, was his last paper. And he was working on that paper on his deathbed at University College Hospital in the ICU. So when I say that when I call this a life well lived, I think it's hard to argue with his scientific accomplishments. But I think absolutely critical to his ability to be as successful as he was his very happy marriage. This was a marriage between two people who could not have been more unlike each other. And yet, among marriages of unlike people, this was the most perfect union that I have ever witnessed. The story of how they met is legendary in the family. Several, I think there were 3 British soldiers on holiday in the Black Forest of southern Germany are cruising along in a car, and they see these 2 pretty young ladies hitchhiking. So the guys say, Oliver's driving and wants to stop, and his colleagues say, no, we don't have any room in this car. Where are we going to put them? And Marilda is hitchhiking, and her friend says, no, we can't get in a car. It's 3 guys. We don't know who they are. It's not safe. But Oliver and Marilda had their way. How they both ended up, she was Italian, he was English. They both happened to be in the Black Forest. She happens to be hitchhiking at the right moment, and they both insisted on making this hitchhiking happen. And 2 years later, they're married. Here, outside the registrar's office in Italy. Marilda Masaccio was a school teacher and was vivacious, brilliant, and outgoing, and the exact opposite of Oliver. You think that Englishmen are typically reserved. He was more reserved than that, and he had, according by his family's own description, difficulty expressing emotion, but they were a perfect pair. He was an accomplished pianist. He loved Bach, and he was a passionate gardener. She was very passionate about art. She was a docent at the Wallace Collection, which they could walk to, having moved into central London. It was delightful to be in their presence when Marilda would correct Oliver in the most cheerful and funny way, and he would just acknowledge that she was right. And to quote their daughter Marilda, theirs was a passionate, utterly loyal union. And just to give you an idea of her, because she was really impish. Here we are at this meeting, Oliver standing beside her, and everybody says, Marilda, come join the photo. So there she is. And I think you can see that gleam in her eye. Here they are on holiday. It was a perfect union. And so I'll end with two quotes from one of the obituaries, one of the many obituaries when Oliver died. He was a clinical scientist of broad intellectual scope whose accomplishments reflected his great energy, devotion to his patients, talent for close observation, and deep understanding of electrolyte, mineral, and acid-base physiology. Professor Rong left an indelible stamp on generations of physicians in and beyond nephrology. He was a physician whose connection with patients was deep and mutual, and an insatiably inquisitive investigator who blended close observation with the broad view. So thank you very much. And I will invite, get rid of the slides, and I'll invite either Marilda.
Michela.
Wish Marilda were on the screen. Invite either Michela or Anthony to correct anything I've said. You can get rid of the slides.
I figured out. Stop share, I think.
Shall I say something?
Please.
Yes.
Okay. No, that's really lovely. Thank you so much, Steve. That's a fantastic tribute. And I do hope you can send me the recording and I'll send it around the various family members who couldn't be on this. It's a lovely tribute to my dad. He would be absolutely delighted. Can you not see me or hear me?
We're trying to figure out how to get the Zoom back up.
Where's Dr.
Any people in the audience want to?
Well, never mind. I can hear you. It seems you can hear me.
Yeah, we can hear you.
Okay, so thank you very much. And yes, the tubes, the plastic tubing was very important. I remember watching my dad. He used to wash them down with beer, very methodically.
There back behind it.
Here we are.
There we go.
And the other thing that my dad went in for, when we were growing up is he would stop the car. We would often be driving through Scotland in that stint where we were in Scotland, and he would stop the car and then go out into a field to pick up a cow pat. Because obviously they were fascinating, and he had to pick up the cow pat and put it in the back of the car so he could go and study it later. So that was the kind of house I grew up in. The other nice story which I remember him talking about is when he went to Papua New Guinea in the latter part of his career. He went there with my mum, and it was all part of the studies towards the end that he was doing on renal tubular acidosis. And they knew they wanted to bring blood back, but they hadn't got the permission to do it. And my dad tended to cut corners, which is why he used families and colleagues so often as his trials. So they put the blood samples in a thermos, and just kept asking the hostesses, the stewardesses on the flight back from Papua New Guinea to just bring them ice. And I think they thought that they were drinking a lot of gin and tonics, but in fact it was to keep the thermos of blood samples cold. And then when they landed, I think it was Raj Thakur who met them at at the airport and took the blood samples away. So all of that, I'm sure, broke every every rule in the book, no doubt. I'm sure all sorts of international laws were broken, but it got it done. So thank you. That's a really lovely tribute. He was on his deathbed, dictating to me final changes to his paper. his last paper. And I did think it was a good distraction for him, actually, because I think he knew he was dying, and he knew he was dying in his old hospital. And several colleagues, Ben Walsh and various other colleagues that he very much loved, came and said goodbye to him, and also took instructions on the last changes to that paper, which was all he wanted to do. He wanted to get that last paper done. And after he died, I'm a journalist, and getting into print is an important part of my career, but I was mortified because he had more papers published after his death than I did that year. And I thought, right, thanks, Dad, showing me up again. So thank you very much. It's lovely to see Jennifer on this call, because I think, what she's been doing with the foundation is something that my dad would have so appreciated. And he'd been fascinated by it, and he'd have wanted to get involved. And it's just such a shame that he's not around to do that. But your tribute has been beautiful. He would have loved to hear it. You've been so generous with your recognition and your praise. And it's lovely also to see Anthony on this call, because, I know they were sort of fellow conspirators and they were always talking about work right to the end. So thank you very much for including me in this. I really appreciate it.
Thank you, Michelle. And for those who may not know, what Michaela was referring to with Jennifer is Entities Foundation, and the foundation has established a research award which is named in Oliver's honor and has been given out twice now, and so it will be longstanding. Thanks.
Thank you. That's excellent.
Okay, excited to be moderating the Aligning Patients, Caregivers, and Researchers in Common Vision. First off is Dr. Kevin Koo, and we're really excited about this session. We're partnering with biotech to get these things done, just like the registries and all of this information. Patient advocacy groups really can be instrumental in helping pass things, get research funded by the NIDDK, NIH, Office of Rare Disorders. So it's really important that we partner with each other. Kevin, I'll turn it to you.
Well, thank you so much, Kim, and thanks for being here and sharing your expertise and your important perspective and the work that you all do at OHF with all of us. And I think I think this session will be exciting because it brings together the voices of across the research spectrum, and I think that's when we really are the strongest. I'm pleased to kick off this session by providing perhaps a bit of a surgical perspective. I'm a urologist. My surgical practice is focused on kidney stone disease, both common and rare stones. And I'm pleased to share a little bit about what's going on in our space with all of you, and as well as talk about how we can leverage surgical capacity to drive and accelerate new discoveries that many of you are leading. I don't have any relevant financial disclosures today. So as we think about advancing kidney stone science and advancing where we can go next with all of the many ways, the many facets of research today, we like to think about surgical challenges and how we can continue to push the boundaries of what we do in the operating room to advance what we're doing on the benchtop and in translational work. So how do we overcome the traditional technical limitations of kidney stone surgery to make stone surgery safer, more efficient, but also to yield better insights? How can we do it faster and less invasively? Can we pick the right approach for the right patient and use predictive technologies to help us to advance those outcomes? And then, of course, importantly for our discussion today, can we integrate surgery and surgical perspectives with a research platform? And how can we integrate those perspectives with the dialogues that we're having today? Let me use the trick we've all now learned to hide the floating meeting controls. See one, do one, teach one. So one of the traditional technical limitations of endoscopic surgery for kidney stones has been truly that, the technology that we use. And so you'll see here that the optics of traditional fiber optic endoscopes and telescopes, as well as the fragile nature of this equipment, has led to yielded limitations on both optics and durability, which has certainly impacted the surgical outcomes as well. Now, as we move toward newer technologies and better endoscopes, we can see now that our ability not only to see into the kidney, into the renal collecting system has improved, but we can now get the type of resolution that makes possible modeling and artificial intelligence-driven discovery about the renal papilla, about what the differences might visually look like between stone formers and non-stone formers, and stone-forming calyces and non-stone-forming calyces within patients themselves. We can't quite get to the resolution of seeing those inflammatory pathways happening here, but perhaps very soon we'll be getting there as well. So you see here with the new digital technologies that just our endoscopic resolution has really improved and allowed us not only to be more accurate and safer during surgery, but permitted new research discoveries based on these findings using our visualization during surgery. Now, here we have newer advancements within the past 5 to 10 years called single-use ureteroscopes. So one of the advances now has been that we have these these ureteroscopes that are used, they're disposable, they're used for a single procedure in the operating room, and they can be especially useful in cases where patients have complex stone disease, where traditional instruments might sustain damage, and we might be more hesitant to use certain types of equipment. These newer ureteroscopes have really allowed us to perform procedures in ways that previously were not possible. And you'll see, importantly, that the miniaturization of these endoscopes and this endoscopic technology has really facilitated a new ability to look into places we might not have been able to look into and treat patients with complex anatomy that were previously technically very challenging. Now, there have also been important advances in lithotripsy technology. Lithotripsy, of course, being the procedures we do to break down the stone, decompose the stone into fragments that we can remove or otherwise evacuate. So the advancement and now widespread dissemination of high-power lasers has helped to reduce time to completion of procedures, thus shortening procedural time, increasing patient safety and satisfaction, as well as empowering efficient dusting lithotripsy. So unlike the conventional form of lithotripsy, where we break down a large stone into smaller pieces that we can then physically remove, which is still, of course, feasible, but now we're able to use a dusting technique whereby we pulverize stones into sand or small granules that can then be flushed out in the urine, which again is a way to improve our efficiency and gives us better options for different types of kidney stones. So here's an example now of what dusting lithotripsy looks like. The laser fibers coming in on the right side of the screen, and where traditionally we might have broken this large stone down into pieces that we then use baskets to remove. Here we're applying a dusting a lithotripsy type approach in order to pulverize that stone into pieces. So all the small granules you don't even see floating around after the laser touches the surface of the stone, because those stone pieces are now dissipating into the surrounding irrigation fluid and being flushed out of the kidney system. Those that remain at the end of the procedure can stay there, and the patient simply passes those pieces safely and efficiently. So it gives us additional options so how we can treat these large kidney stones, and especially in patients with these large complex type stones, such as the patients we're talking about today. It really provides this opportunity to create, as you can see, this sort of this field of small dusted granules that can pass easily and safely. We also have advancements creating next generation lithotrites, which are technologies that allow us to not only break down large dense stones quickly and efficiently, but also evacuate those pieces. And so now with our next generation lithotrites, we can currently use both ultrasonic and ballistic energy to really make percutaneous nephrolithotomy, the procedure that we reserve for large complex kidney stones, even more efficient and even safer for patients. So here's an example now of our lithotrite coming in at that 12 o'clock position, where you have a large staghorn stone in the patient, and this is a very dense calcium oxalate monohydrate stone that is efficiently being broken down. You can see they're in large chunks by the lithotrite, and then sucked out, directly evacuated, so that there's very little, if anything, left in the kidney at the end of the procedure. And so again, enhancements in our endoscopes and our ability to see into the kidney, as well as advancements in our technologies to break down the stone, evacuate the pieces in a single procedure, rendering the patient stone-free and able to recover as safely and quickly as possible. All these advancements have allowed us to really continue to advance what's possible for urological surgery. Now, our research and our, ability to predict how patients will respond to research has also concurrently enhanced our ability to choose the right patient, choose the right procedure, and combine the two for the safest approach. So an important question that still remains here is, how do we predict the stone response to lithotripsy? Now that we have more than one approach, now that we have more than one technology, how do we couple what's right for the stone and what's right with the patient, for the patient, based on that technology? So we've been pleased to have a very active and productive collaboration with our colleagues in radiology and our CT Innovation Center and Dr. Andrea Ferrero's group, and you'll hear from him later, about predicting stone response to lithotripsy. And so what you see here are 3 different stones with three different characteristics of stones where we were looking at the predicted comminution time, the time it would take to effectively break that stone down in surgery, and the observed comminution time. So building this complex model based on CT and imaging guidance, most stones follow along this general model, predictive model, about how they're going to respond, where the observed time and the predicted time is about the same. However, there are interesting discrepancies here. So for instance, there may be stones with very different volumes. So you'd expect that larger stones take longer to break down, and smaller stones take less time. They may have very different volumes, but they may have similar comminution time. So what is it about the density and the composition and the structural integrity of those stones that leads to this discrepancy? And how do we leverage that to use the right technology for the right stone? In contrast, there are stones with similar volumes here, as you see in the blue dots, but very different comminution time. So clearly, volume itself is not the only predictor and not the only way to accurately prognosticate how a stone is going to respond. So how do we choose those stones as well? And how do we empower CT technology and innovation and modeling to predict which ones are going to respond which way, and then make those projections preoperatively so we have the best outcome postoperatively? Finally, I wanted to spend a few minutes just talking about how we integrate surgery and surgeons into our research programs and integrating surgery with research. I think for a long time, we may have thought about these two seemingly disparate aspects as related, but perhaps marching along on their own continuum. So patients sometimes need procedures. They have an acute stone event, or their stone recurrences get to a point where surgery is reasonable. And then we come in and we remove those stones. And then we hope that they continue to go on to their surveillance. In parallel, the research community and the work that all of you are doing has proceeded along, and they've been involved in studies and helping to advance our science, but perhaps once again in independent fashion. I would suggest that an integration here in our research platforms is not only necessary, but vital to allowing us to continue to work together and collaborate for the best research outcomes for our patients, the best patient-oriented, patient-driven research. And so I think by integrating this, by allowing surgeons to be part of the research platform, and by then feeding those insights back to us and allowing us to improve patient outcomes, we can come up with this continuous cycle that really works in partnership with each other, from intra-op to the benchtop, and of course then delivered to the bedside so patients can benefit. So in summary, I think that enhanced technical capability of our surgical technologies has allowed us for faster, more efficient lithotripsy and stone removal procedures. We are excited about predictive models for improved surgical planning and intraoperative decision-making using clinical data, using genetic testing, using imaging innovation. And finally, I encourage all of us to think about how surgical innovation can be used to empower translational and clinical research and multidisciplinary care. And I think this session and this day really highlights the value of doing so and the promise of what can happen when we all work together. Thank you so much for having us, and we're excited to be here. Thank you.
Moving right along, Dr. Christina Peniston.
So thank you for having me here today. This is a really well-designed program, and I'm really pleased with how the day is going. My name is Chris Peniston. I'm a scientist and also a registered dietitian at the University of Wisconsin-Madison. I'm lucky enough to have a small clinical practice. I practice with my colleague, Dr. Alan J. Grew, who's a nephrologist, who I believe was trained by Dr. Simon. So he talks about you all the time. Anyway, so, but then I'm here today, I'm not really representing my research per se, but I am representing a new initiative that the NIH has put together called CARIBU, which stands for Collaborating for the Advancement of Interdisciplinary Research in Benign Urology. This talk is going to be different than all the rest that you've heard today and that you will hear later today. I'm going to step back and talk a little bit more broadly about urologic diseases, not just stone disease, but this will give you a little bit of a taste of where CARIBU fits into research. When we talk about non-cancerous urologic conditions, we're trying to get away from that word benign, because really these are not so benign diseases, right? We all know that. There's hundreds of millions of people that are affected by these kinds of diseases, whether it involves the bladder or the urethra or the prostate or male reproductive or kidneys or ureter. So we know that a lot of people are affected by these diseases, and it's actually growing because as the world population ages, these diseases affect more people. They're associated with very high cost, and there's a hidden burden. And that's really an important feature of these diseases, is that there's oftentimes a stigma associated with some of these diseases. So you got incontinence or overactive bladder. You might not be as willing as some patients with, say, prostate cancer to sit around and talk about, your disease, because it might not seem as important, or it might seem like you should be able to deal with it better or something. But it's really a huge burden on people, and it affects them for decades of their life. And so we think that the prevalence of some of these diseases are underreported as a result of that. And then the problems are complex. I've just shown a little diagram here that shows you how all of these problems, kidney stones included, and your program well represents that, involves neurological and immunological and genetic and all kinds of other influences. So these are complex diseases. And if you haven't read this paper, I believe it was in Gold Journal a couple of years ago, by Brady et al., you should take a look at it. There is some aspect of stones in the paper, but it's really talking about these diseases as a whole and about how there's a public health priority, hidden burden rather. These are complex problems. I think you've all seen this notion, as you can see on the right, where if you give 5 different people pieces of a box, right? But you don't tell them that it's a box, and you just give them each one piece. They'll study that one piece. The field 1 guy here is going to study that purple thing, and he's going to say it's purple and it's shaped like this. And the field 2 is going to look at the thing and say it's yellow. But only when you get people together, as Dr. Koo was just talking about, do you get the sense that it's a box, and really that it's much more than it would seem. So as our problems become more complex, as the research questions are larger and more intricate, the more we know, the more there is to know. And especially now with new fields of inquiry, like all the omics and big data and AI technology and so forth, these things require specific mastery. And we can't just, be the solo investigator in a lab that does everything. So this is just a diagram to show kind of a problem that I'm going to be hinting towards. There's a rising world impact of some of these diseases, like I said before, but fewer providers. So this is actually about BPH, benign prostatic hyperplasia. And the solid green line is men over 65 globally who are expected, it represents the rising prevalence of that disease among those men. And in the yellow dotted line at the very bottom is the US projected urologist workforce. So it's not keeping up, right? The purple and blue are Canada and the UK, so they're different from us. But anyway, we're definitely not keeping up. This is not a new thing. The AUA has facts out about how the urology workforce is aging. Median urologist age is 55. And what is the impact of these fewer providers on stones? Well, David Bain is a urologist at UCSF. He did a fellowship, he did a K-12 scholarship rather, where he studied some of the determinants of cost to stone disease. And one thing he found was that lower regional urologist density was an independent predictor of higher surgical cost. And the reasons why are multifactorial, but people come with delayed presentation of their stones. They're sicker, they're more infected, the stones are larger. And so maybe they have uncontrolled diabetes, maybe there's economic issues. So there's going to be an impact of fewer providers on stones. And also, what about scientists? There's also a shortage of scientists who are working on these questions. This is supposed to be the pipeline that everybody's talking about, where on the left, you see maybe there's a lot of students who say, yeah, I'm interested in that. And then you get to the postdoc and fellow stage, and then the academic staff. We lose a lot of people to industry. And then you get to the bottom where you're at scientists and urologists. So we need more of these people as we go through this. And this isn't a perfect situation. Some people believe the pipeline has more to it than just leakiness, that there's problems getting into the pipeline in the first place. So we have this problem working from the right to the left of we don't know the true disease burden. We don't really know characteristics of the disease. We have a shortage of biomedical workers, clinicians, as well as scientists. And then we have a problem with the status of our science as a result. Other diseases have higher priority, more money. Fewer non-cancer applications are being submitted, so we don't have a study section. There's slower progress in making discovery, so we can't solve these conditions as we might want to. And then our messaging isn't reaching out far enough because we don't have the kind of patient advocacy in many areas of these benign urologic diseases that we should. And so for all these reasons and more, the NIDDK decided to create a community of investigators who would work together and share knowledge and network with each other to do mainly 4 things: to support the next generation of urologic researchers with very thorough mentoring and education, to enhance the knowledge of mechanisms that are associated with normal lower urinary tract development as well as disease, to cultivate new research tools and ideas by expanding the collaborative network within and outside of traditional urologic research. There's a lot that people are learning from people who never heard about urology, right? And so there's techniques and tools that we can pull in. And then finally, of course, to reduce the burden of these diseases on people. So the NIDDK created a U24 mechanism, which is what I got, which is what I have, called the Caribou Interactions Core. So it's like a coordinating center, but they're calling it an interactions core, because my main goals are to create these kind of transdisciplinary connections by creating infrastructure that promotes these collaborations, and then to encourage and embrace new people into the field. So, you know, obviously we all know that we need to work together, but here's just a little example of how when a person is researching on their own, we call that intradisciplinary. Multidisciplinary models are abound. Many of you are in these kinds of models. And then there's cross-disciplinary models where you kind of work together, but you're yet still in your separate domains. Interdisciplinary, and then where we're all supposedly trying to end up is at transdisciplinary, where we have that sort of bullseye sort of thing. We're all working on the same thing and bringing different tools to it. So who is Caribou? So on the right, you see some logos from some of the institutions that are currently part of the Caribou community and who were previously part of the Caribou community. We are, our main sort of flagship organizations are the Urology O'Brien Centers. These are U54 mechanisms that are funded for 5 years at a time. They get quite a lot of money. Biggest aims are to train and produce resources that the broader community can use. We have P20 centers like the one here at Mayo, and then we have 4 K-12 programs. They are 2 different kinds. One kind is an epidemiologic urology research focus, and the other trains early stage investigators in multidisciplinary research, basic science, and so forth. So the Caribou community sort of, people come in and out of it, because you can imagine where your grant cycle might end and you maybe didn't get a renewal, but you're still part of the Caribou community. So as we go forward in time, we're only going to grow because everybody that is Caribou and so forth. So we also have a lot of trainees. As you can imagine, there's a lot of grad students and postdocs that are part of all these labs. So as I said, it's a collaborative-minded, organized community, a conscious force within the broader urologic research community. And what the Caribou Interactions Core does is create platforms for the exchange of knowledge, recognizing that exchange of knowledge is the first step in developing meaningful collaborations, to develop that infrastructure for community building and networking and learning and training, to expand the visibility of research. We are active on social media and all kinds of other things, because one of our goals is to promote our investigators, to promote their science, to promote and advance their knowledge. So we're tweeting about this meeting right here today. I forgot Mariana's gonna kill me because I forgot what our tweet or Twitter thing is, but I think it's @Caribou1. Anyway, it is? Okay. And then we also importantly want to catalyze interactions that are productive, that produce real research findings that would have been impossible had that collaboration not occurred. And then importantly, the NIDDK wants me to measure the effects and the impact of our work. And this is kind of new. There isn't like a playbook for this. I partner with some people who are familiar with the the science of team science. That gives me some tools. There's other tools that we can draw upon, but we're kind of paving a path here. And so some of the events and the forums that my interactions core organizes to create that infrastructure for knowledge sharing is all these virtual events that we do. How many of you have been on any sort of a virtual event that Caribou has sponsored? I know a lot of you have. Erin has spoken at a number of them. Some of you will be asked to speak at some in the future. But we have research interest groups, like-minded people. Our most active one is on the urobiome. We've got over 100 people that meet off and on from like 20 different disciplines, veterinarians, infectious disease, urologists, gynecologists. And then we've got all these series of events that are designed to exchange information and, bring us all up to date on what the latest knowledge is. So we also really try to promote personal relationships, recognizing that also is what we do to build trust, and that collaborative efforts really are nothing without trust. And so we put a lot of stock in really knowing people and in really putting people together in various ways. And we do those through the research interest groups, as I said, but also at our annual Caribou meeting. And you're all welcome to come to that. I'll show you the slide that gives you the details in just a second. In summary, think of the Caribou community as a bridge. We want to connect investigators on both sides of the bridge who are capable of addressing these critical knowledge gaps that we have. We want to exchange knowledge, develop tools that make collaboration easier. Many of you are in collaborations, have done it for years, and are really good at it, but it's not a thing that's taught, right? And so we all need some tools that help us collaborate better and easier. And then we want to increase our visibility, which will increase our funding, which will advance science. And then, of course, we have some other goals, which are to increase the diversity and the numbers within the pipeline. So please take a look at the Caribou website. It's caribou.urology.wisc.edu. We have an annual meeting. This year it's in Kansas City, and it's November 28th through December 1, Wednesday, Thursday, Friday. We're accepting abstracts from trainees and ESIs, anything to do with urology, through October 20. And all these centers that I talked about are going to be presenting their science there in posters and in presentations. And it's going to mix people together who are studying prostate with all kinds of bladder and all kinds of other things. And then we have some really cool networking and interaction sessions that we always do at these meetings. So I'll end it there, and I hope to be communicating with many of you in the future. We're working on some patient alliance and patient advocacy things. Right now, we have one going on with a UTI group, but we hope to expand that. And we want to provide resources to you. I have some money in the form of grants that I can provide from time to time. So as a community, stay in touch and look at our website, go there often, and we'll talk to you later.
Thank you very much, Dr. Peniston, and for all the work that you do with Caribou. And we're proud to be part of the Caribou network alongside many of our colleagues across the country. Questions for Dr. Peniston?
So what is it that UK and Canada are doing that are promoting urologists?
Does any of the urologists here understand that better than me? Here. Just different healthcare systems, I would guess, right?
Yeah, that certainly is part of it, I think. And these waves of interest do tend to be cyclical sometimes, but we do know that we predicted 10 years ago a major workforce shortage problem in urology. It's no longer prediction. We're here. We're seeing the shortage. It is active. And because the training pipeline is so long, it's hard to just turn on a switch and produce more urologists. And the same is true for any of us in this room, right? The training pipeline is long. So it's going to take a lot of creative strategies to try to make that up. Yeah, but Caribou is one of them. Thank you very much. So next, we will have Dr. Neera Dahl from our department, or our section of nephrology. And Dr. Dahl is going to be speaking on developing a kidney stone registry and centers of excellence. Dr. Dahl.
Thank you all for having me. And I think what I'm going to talk about is actually, I'm going to say not my day job. This is work that I did before I came to Mayo when I was at Yale. And it led to some interesting collaborations, talking about nephrology and urology collaborations in particular. And so here we go. And this is really like 15-minute overview of two very different but important topics. So the first is when I was at Yale, we developed a multidisciplinary kidney stone program. And so what this was initially purely a clinical patient-facing program where the patients at one time and in one space would meet with me, a nephrologist, with the urologist. In this case, it was Peruz Montemedinia and Dinesh Singh, and then meet with Laura Wilson, who was our dietitian. And so they'd get 3 separate interactions in terms of management of stone disease. And actually, the patients loved it. From a scheduling point of view, it was kind of a nightmare, but the patients really, really enjoyed this process. And the knowledge, sometimes it was repeated, right? Like drink water, but sometimes it was individual to the doctor or to the provider. And so I think that's also part of what made it very successful. And we had been doing that for a while. And we decided we needed an academic arm of that. And so we invited actually David Goldfarb to come and to talk to us. And actually some of what we developed was after that conversation. So we got urology to fund for us a postgraduate research associate, someone out of college who they know all the computer stuff. And so that made it very, very good. And we built a kidney stone registry in REDCap, and this allowed for both prospective and retrospective recruitment of patient information, and information coming primarily from EPIC, but also coming from Litholink. We contacted John Asplin and said, hey, give us all the Litholink data that's come to Yale for the last 10 or 15 years, and he did. And then some of it we would then go back and look at manually to really confirm that we had the data we thought we had. And what we did is we would meet with the postgrads once a week, once every 2 weeks, and see what they were up to and sort of guide them. And so this was really research that was done from a clinical perspective first. So this is the first part of what some of that data looks like. So this is looking at a correlation we saw between kidney stone factors and chronic kidney disease, which I'll show you in a minute. And so here we started with our pool of 10,000 patients. Of those, the urologists, I love them because they always get stone analyses when they extract stones, which is really, really fantastic. And so of those 10,000, we had about 4,000 with kidney stone pathology. And then we broke them up into quintiles based on GFR. This is by CKD epi. So over normal, so GFR greater than 104, all the way down to this 5th quintile, which is GFR that is less than 64. And what you can see is maybe what you would expect, which is as you go through these quintiles and as GFR declines, you see an increase in uric acid stones and an increase in uric acid stones, that's the green, and a decrease in calcium phosphate stones. And then this is looking at that data where you can look at GFR and then look at the mean GFR for each different stone type. And you can see there's clear separation here between calcium phosphate, calcium oxalate, and uric acid. And then this is looking at the 24-hour parameters and how they correlate to stone type. And what you can see is as kidney function declines, so the way this is set up is each 10 ml drop in GFR is associated with this decrease in pH, in decrease in normalized citrate and calcium, but not an oxalate. I know this was a little bit of a surprise to us because there's this idea that as CKD progresses, there are higher plasma oxalate levels, and that this may be important in progression of chronic kidney disease. But this is not what we saw in this data set. And so just pointing out, since we just heard about Oliver Wrong's very nice work, that we also saw that decrease in urinary ammonium. So this is what this data looks like. And what it sort of led me to in terms of my thinking is, I feel like I'm missing a slide, but that's okay. That as we think about patients, so as we clinically think about how to treat patients, we don't always think about small changes in GFR determining what stone type they're likely to have. But maybe that makes a difference in who you're selecting a thiazide for versus who you're selecting citrate for. And so maybe you're more focused on managing calcium in those people that have higher GFRs and more focused on managing acid-base status or citrate levels in people who have lower GFRs. Okay. And then this is the part where this is actually looking now at protein intake and uric acid stones. So again, looking at those 4,000 patients, and of those, a little over 700 had uric acid stones. And we divided those stone formers into people who had pure uric acid stones, here defined as a stone analysis greater than 50% uric acid, versus impure uric acid stones with at least one stone analysis less than 50% uric acid. So these were further defined into 3 groups, but then you'll see some of the analysis is done with these 3 combined together as the pure stone uric stone acid formers, and these 2 combined together as the impure. And this I am just going to show you briefly that this data exists because I want to kind of keep moving along and go to our punchline data. So here what you can see is that as the purity of uric acid stones increases, the GFR declines. So that's part of what I showed you before with the change in GFR and more uric acid stones forming from the prior data. This is looking with different modeling projections at those urinary lithogenic risk factors for uric acid formation. And I will cut to the chase and show you this final slide. So this is looking at specifically at those things relative to protein intake and uric acid stones. So if we adjusted the odds of being a pure uric acid stone former for age, sex, race, ethnicity, BMI, and GFR, the things that were statistically significant were citrate, sulfur, urine urea, and PCR. So these are from the LithoLink data. So they calculate PCR, which is protein catabolic rate for you. And then if we further adjust these models for serum uric acid, urine uric acid, and urine calcium, you can see now that urine urea and PCR still are significant, as is pH. So this is really just an example of data mining what's already in EPIC. That's basically what we did. And I think that was why John wanted me to present this work. I'm moving on now to something that is slightly different and not kidney stone related. And that is talking about the Centers of Excellence program. So the PKD Foundation realized they had a problem in terms of patient care, in that a lot of patients, so PKD is a rare disease. Christian talked about it earlier, 1 in 500 to 1 in 1,000, and probably fewer numbers actually diagnosed. So not at a large center like Mayo, but at a smaller, say, private nephrology practice, patients with PKD would come in and they would feel that the doctor didn't know the disease as well as they did, that they had complications that the doctor dismissed or didn't understand. And so the PKD Foundation said, we need to address this in a larger way. And we need to develop places where we can say, these are centers of excellence. This is where people will truly understand you, right? If you're a PKD patient, they can take care of you. And so what the PKD Foundation did, and this is similar to what the Cystic Fibrosis Foundation has done, what other rare disease foundations have done, is they created a tiered program of either centers of excellence like Mayo or like Yale, and then also partner clinics. So people who were interested in PKD had people who knew the field well, but maybe didn't have all of the resources to offer. And this is, it's really, we're just past the 1 year mark, actually just coming up to the 1 year mark for this program. And what you can see are the centers of excellence in white and the partner clinics in the darker dots. And the idea is that eventually this map spans the entire country, that you know, wherever you are, we will be able to connect and provide PKD care that we think meets certain standards of quality care. There's a few different parts of this program. So the big part of it is mentoring. Within the COE program, there are quarterly conferences that happen for the providers, for the nurses who are part of the program. There are outreach to the different PKD Foundation groups that are nationally around. So this is part of this job, is to connect people and to connect networks for people who are interested in PKD, for patients who have it, for researchers who are interested, for people looking for clinical trials, just to connect the pathways. And so some of the ways we are doing this, there's an annual PKD Centers Conference. This last one was in June, and it was really fantastic because now we could meet as a group and say, this is how I take care of this problem. How do you take care of this problem? And so people could start to talk about this in meaningful ways. And it ran nicely at the same time that a PKD conference for patients ran and a PKD research conference ran. So all of a sudden, patients, providers, PKD Foundation, all in the same place over 3 days, which made it very successful, I think, as well. There's a lot of education that goes on with this program, a lot of sharing of best practices, topics that we talk about. Pain, for example, is a big problem in PKD. How do you manage that? The KDIGO guidelines are coming out any minute, perhaps. And so the PKD Foundation, as part of that, has been invited to write a commentary on those outcoming KDIGO guidelines. And so this is kind of the work that happens within the Centers of Excellence. And then also reaching out to local healthcare providers, right? So we don't diagnose PKD. That's done at the level of the primary care doctor or the ER doc who orders that for CT scan. And so how do we connect with those guys and get those patients referred to us in a timely way? And also the DEI goal, right? So how do we reach out to programs that maybe don't have the same, you know, we at Mayo don't have the same diversity as we have in some other programs. And so how do we start to reach out and solve that problem a little bit? So part of this, we have a patient navigator. So a nurse or nurse practitioner that's highly specialized in PKD. This is a person who is the first point of care for patients. We have a patient advisory panel. We just met with our patient advisory panel recently, and they had some great ideas. So we thought here that we offered really good care, and they said, you know, what would make us feel even more connected is if you gave us at that first meeting a set of resources of, this is how I can connect with the foundation. This is how I can talk to other people who have PKD. And actually all of those resources exist. It just didn't occur to us to say, hey, this is the Twin Cities chapter. This is how you connect to the Twin Cities chapter. So that was actually really, really helpful to have that patient advisory panel meeting. And that is it. That is, so this is, if you actually make it outside, this is the Peace Fountain in the Mayo Peace Plaza. And it's actually a really interesting sculpture. It's this Native American and sort of interesting sculpture, but starts at the beginning. Thank you.
Our next speaker is Dr. Hatem Amer, who is with our Department of Nephrology and will be speaking to us about building a nurse-driven registry and treatment protocol. Thank you very much.
So thank you everyone for the invitation to present today and for seeing some folks that we've met before and new faces. So the disclosure I will start off with is that I'm actually a transplant nephrologist, so I'm 100% in the transplant practice, so you might get a transplant skew to this. And when I was asked about the registry that was built here for the I'd like to just give a little background because I thought we might have some of our newer fellows with us, so some of this information you might already know and are experts on. But this is an old study from 2011, which looked at essentially the incidence of bariatric procedures for weight loss over time. And as you can see, there's been a significant increase in the amount of procedures that are performed. And from a very long time ago, we knew that essentially some of these malabsorptive procedures results in kidney injury and kidney damage. And you can see one from the 1970s there, and on the right from 1995. So as you can see, even defined as a creatinine going greater than 2, by about 14 years post-procedure, there's at least about 40% of patients would meet that endpoint. So it was not unexpected that we'd start seeing some of those in our transplant practice when they come in for renal replacement therapy. So the question was, of course, why were they losing kidney function? Where were they more likely to have kidney failure? And, you know, malabsorption, you think about dehydration and so forth. And then Dr. Lieske and Dr. Kumar and several others from here showed that essentially the individuals who undergo a Roux-en-Y gastric bypass procedure will have an increasing amount of oxalate post-procedure. And that was shown nicely in this figure here from their paper. And essentially at 6 months and 12 months, if you look at urine calcium oxalate supersaturation, it goes up and it continues to be high. And if you look at the actual oxalate load that was seen in the urine following an oxalate administration, you can see that it goes up. And essentially what they end up with is essentially an increased amount of oxalate absorption and secretion in the urine. And this certainly may contribute to the kidney failure. And this is from Samih Nasr, who's one of our nephropathologists at this time. And you can look at the calcium oxalate depositing within the kidney and causing some inflammation. So why did the transplant nephrologist get involved in all of this? And I'll show you 2 cases representative of what we ended up with and how we sort of tried to solve the problem. So this is a gentleman actually who is, at the time of this happening, was about 65 years of age, very low immunological risk. He didn't actually have a Roux-en-Y gastric bypass, but he had Crohn's disease with a short bowel. And he gets a living donor kidney transplant, and this is within the first year. As you can see, there was a rapid decline in early function. And it just progressed, and every measure was taken to try and improve this. And what we thought of was, well, it's probably the hyperoxaluria and the deposition, and we did see calcium oxalate deposits within the kidney. And many of you probably are aware that the transplanted kidney by default is an acute tubular injury once we take it from a donor into the recipient, and they tend to be a little more sticky. They might be much more vulnerable to this than native kidneys. So he got stabilized with a regimen that we borrowed from the PH practice, and as well as a little modification from Dr. Lieske and Dr. Milner. So we're very fortunate that they are with us, and the experience from the primary hyperoxaluria has helped us develop the protocol. But this is one example. So this is post-transplant, losing a lot of function, needs to be managed, needs to be stabilized. And once you do that, they stabilize. And he continued to do well for many, many years afterwards with a GFR in the mid-30s. This is another example, but in the opposite spectrum. This is someone who came to us for a repeat kidney transplant. They had previously had their own Y-gastric bypass. And when they came in, we decided to check oxalate, which is actually currently right now one of our standard procedures for any transplant candidate who had a malabsorptive procedure in the past or is at risk for malabsorption. And what this person turned out to have was hyperoxidemia, hyperoxaluria, but there were no intervention for this. And we went ahead and applied our protocol, and guess what? Their kidney function improved, and they actually didn't require transplant for quite a long time. Several years out, they're still having their first kidney allograft functioning and no concerns for a repeat transplant. So interventions do work, but what are the interventions? So this is just the key highlights of this, which is we advised them to go on a low oxalate diet, a low-fat diet, and we use calcium as an oxalate binder. The targets that you are seeing here were essentially for the urine and plasma oxalate we borrowed from the primary hyperoxaluria practice. And the serum and urine calcium we put in as safety measures just to avoid that we don't cause any injury with hypocalcemia or hypocalciuria. Some people were concerned with the calcium supplementation that we're administering. What is that, unfortunately? So you have to give these medicines, you have to titrate them. And as I mentioned, I started off with, I'm a transplant nephrologist, right? So most of these patients that come to us for a transplant eval, we are not managing them. They're being managed by a nephrologist in the community all over. 3/4 of our patients are more than 3 hours away. So we're not actually following those. So the end result is we make these recommendations, but then how are they going to be implemented? And if we are talking to a dialysis provider somewhere, they're worried that, oh, you're going to give them hypercalcemia, you're going to give us calciphylaxis, you're going to cause problems, and you're going to cause worsening function. So this became a very time-intensive thing to do. And if we look at the transplant practice, we have nurse coordinators, we have pre and post. The pre are busy trying to make sure that they are up to date on their CPRAs, up to date on their screening, their cardiac workup. To add this to the mix became a problem. For the physicians to follow, it was very time consuming and easy to miss, because if you're busy doing something else, you don't remember to go back and look for the 2-week follow-up and so forth. And then it turns out that there was also a need on the CKD practice, because these patients were also showing up in the CKD practice that needed to be seen and followed. So how do we fix this? We said, okay, if we have a clinical protocol that says we're going to follow and administer, patients will visit with a dietitian, then we're going to use calcium supplementation, not as a supplement, but as a oxalate binder, then we have a regimen that can be followed. Is it really the best thing to do to have multiple people see these patients when they only have one in the practice, for example, or 2 in the practice? It's better to try and consolidate and have a regimen that can be followed. So what we decided to do is write the clinical protocol and then involve our nurse hypertension specialists who see a lot of our hypertension patients long-term and develop a way of having them manage these patients. And that was done. But then it turned out that you had to you can't just be a clinical protocol where you write it and say, this is what you do. With the nursing protocols, and when you implement it into a computer system, it had to be a little more structured. So that we essentially spent some time working through this, and I don't know if we can connect. So it created this sort of, it had to, this is the PDF version of it, but built into the system, you do these checks and it will take you down the tree to what to do. And as long as the patients are, meeting these criteria, the nurses can do it automatically. So we don't actually have, it minimizes the amount of physician input, and it actually works a lot smoother than when you start looking through the chart and trying to fix things. So that was built. But then the question is, well, okay, we have this for the practice. We should probably keep track of these patients, right? So it can't be that we're doing this and we can't use the data in the future. So the first attempt to get this into a database was going again back to REDCap. And this was sort of my amateur due to this, which is essentially we created a demographic page and an encounter page. And I think the nurses really are saints, because they were willing to go ahead and spend the time to do the clinical documentation and then fill out the REDCap database. And they would do the demographics the first time they see the patient, and then with each encounter, they're going to fill out one of these forms. So they finish the clinical note that's required by, administrative purposes, and then they will do this as an add-on so we can go back and track what the patients were doing and what was happening. And they managed about 48 patients with this modality. But then in 2018, we had Epic join us here, and then by 2019, we sort of got comfortable using it. And I owe the next step to this is to really one of the nurses with us, Ted Mueller, who's one of the hypertension nurse specialists. He said, well, Epic exists, and Epic now has all these tools. Have we heard about these registries that they're building? And can we use one of those registry systems to actually do this? And that way it will all be in one system. So we spent, this is the REDCap as it looks, as you can see here. Quite complicated and a little unwieldy to do on a day-to-day basis. So we spent the COVID year 2020 with our EHR folks, and they essentially we said, okay, what is the issue that we're dealing with? We have patients that we follow, we have a strict clinical protocol that we're using, and we have essentially information that we need to gather and make decisions on, document, and send the patient's recommendations. So they built us a tool within Epic that essentially we can manage these patients. We have a report that gives us all the patients that we're managing. This is cut and paste from it, but essentially, as you can see, the patients are up here. It will tell you who's managing them. I took the patients and MRN numbers out. And then it would say when the dietitian saw them, when the labs are due, are they voiding or not, because that affects how the protocol works. Are they on dialysis or not? What's their status with the transplant? Are they pre or post-transplant? And if you look at the flow sheet that gets developed, it actually gives you over time, and this is just a snapshot from it, but below it, you'll find other parameters regarding their medications, what was happening, and so forth. And it plots it over time. So that became a much easier way of following these patients. We were collecting data through it. We do a quarterly review just to go over, make sure everyone is doing fine. It used to be quite a hassle to do. With this, either myself or Dr. Lieske actually spend maybe like an hour to go over the 52, 53. Currently, what's in our current cohort, we have 132 patients entered. As you can see there, their average age, more predominantly female. And as you can see, how many are waiting for the transplants and how many are not actually in the transplant phase. The tool, once it was developed, it actually rolls out to all the Mayo sites, but each site is geographically limited. So that data is just for the Rochester cohort. And essentially, it has been a huge improvement. And we're learning from it more. We're trying to figure out, actually, is the protocol right now the best way of doing it, or can we modify it to make it easier, or can we do less work? But it really has consolidated the work. If you ask the nurses, they now have one person assigned for a day of the week that they go over all the patients, do the communications. It cut down on the number of misses. It cut down on the number of hours that the physicians are using. And I think it has worked very, very well for us. And this is the calcium carbonate squad that helps us with it. So I'll be happy to take any questions related to this.
Sam.
Why not use calcium citrate? That was a thought. The concern was calcium carbonate is cheaper. It's available. Sometimes they can chew it, and we're using quite high doses. And one of the concerns was if you give the citrate, it might be more absorbed, so you might end up with more hypercalcemia. So we made the standard the carbonate. There are some of the patients are on citrate for a variety of reasons, but because of the nursing protocol has to be really precise and they cannot deviate, so we decided to use the most commonly used and the cheapest formulation.
Great talk. How long did it take you to get that Epic protocol?
We were lucky in a way, but also unlucky. So it was in the COVID year. It took us essentially the full year. I think we started in about the end of February, and by the time it went live, it was December. But it's an ongoing process as well, because once it launches, you sometimes find things that you want to improve, so you put in for enhancements. And that takes a little cycle. It gets prioritized and then it comes back. And the actual clinical protocol is also just the question marks. For example, if you have someone who has GFR of less than 20, right? they're making urine. Do you really need to do a supersaturation at that point, or could you just go by the plasma? So it took a lot of tweaks. Electronically, it gets prioritized, cycles through. The clinical one, as we make changes, it has to go through its approval processes again so that nurses can actually do it and it's within their scope. And then it gets implemented in that algorithm that goes through the system. If any of you are in transplant, so BK, for example, is another one that we could do this. And we use this one as a jump step to it. And we said, okay, we did it with this, let's do it for BK. Lama.
This is great. We're struggling to build something to get data on our EH patients. Do the patients, they have to sign a consent when they first start? No. Yeah, the IRB for us did not allow longitudinal collection.
This is clinical.
Yeah, it's all clinical. But you put it in effort, that's why.
Well, no, so this is actually our clinical practice. If I want to pull the data to do research with it, I would submit to IRB. And we have an IRB protocol in place, essentially with an exemption that as long as it's clinically obtained, we can use it. But this is a clinical tool. It's super effective clinically.
Basically just giving you like a cohort of just being able to see within Epic that pulls together all of your data points. And then if you actually wanted to like export it to REDCap to do a paper or something like that, then you'd have to do IRB. Call me, we'll get it done.
Yeah.
We'll get it done.
So are you successful in reducing plasma oxalate levels?
Yes.
In patients who have really low GFR or on dialysis?
Yeah, so because for us right now, because of the issues that we've had with post-transplant mishaps, we don't let anyone go to transplant by protocol except if they're less than 30. The 30, I think, is too tight. I think you could probably get away with a little higher. The ones we've run into trouble with are the ones that are on PD and refuse to go to hemo. So some of those have lingered around 45 and they won't drop below that. There was a case that they were running out of vascular access and we allowed them to go through, and they were actually much higher. They were at like 80, they dropped and then it stabilized. But the ones, everyone who's on hemo or not on dialysis and is following, we've been able to get them to transplant with that less than 30. If they're on PD, it's been problematic in some.
You haven't made anyone reverse their Roux-en-Y?
Not since we implemented this. I've done it before, years back, and we've actually ran into issues where when they reversed it, they didn't actually have any benefit because they ended up with short bowel. And one unfortunate person actually ended up in their hospital for like a 9-month stay because of severe complications following the reversal. So we've not forced anyone to reverse at this point. And we've not run into issues post-transplant as long as they're being followed.
Once again, fascinating insights from the intersection of our different disciplines and the way we can leverage each other's discoveries to enhance patient care. All right, our next presentation will be from our colleagues virtually. This is Drs. Mira Keddis and Ryan Tatton speaking to us on building a multi-center EH registry. Welcome, thank you for joining us.
Thank you for the invitation. Really excited to be here. Thank you, Dr. Lieske, for the invitation, and really excited to bring in Ryan Tatton to this amazing group, and hopefully we can recruit him to be one of our young investigators of the future. So Ryan will be actually taking the lead in the presentation, so I'll just give you a little bit of background. We have I love, by the way, the presentation about the difference between multidisciplinary, transdisciplinary, and cross-disciplinary. So I'm going to call ourselves cross-disciplinary clinic at Mayo Clinic in Arizona, where we do have a dietitian, urologist. I'm the nephrology representative in the team, and we take care of large volumes of patients on a weekly basis. And we've seen a number of enteric hyperoxaluric cases that have come through. And in an effort to try to better serve our patients, we thought through how can we learn more, and reached out to Dr. Lieske and was a thoughtful input. The thought was maybe we should think about a registry because there's an absolute need for that. So this is what we'll talk to you all about, very initial phases of this project, and some motivation behind it, some of the challenges we've run into, and some early preliminary findings that we'll be presenting at a poster at ESN next month. So the main objectives that will be covered: understanding why are we doing this? Why are we pursuing the idea of developing an enteric hyperoxaluria patient registry, our approach to it, current or challenges and how we've mitigated them, preliminary results and future directions. And we welcome your feedback and ideas on how we can make this sustainable and productive. We have no disclosures, and I am going to pass this on to Ryan Tatum, who is a fourth-year medical student. He shined during the nephrology course. And not only that, he was my very first student TA for the nephrology course, and he's really set an amazing precedence. And a lot of people wanted to follow his footsteps as a TA, and he's interested in nephrology and currently applying for internal medicine. So really excited to have him join this meeting as well.
Thank you so much, Dr. Keddis. Before I go on, just making sure that everybody can hear me okay. All right, great. If you could move on to the next slide. Really honored to be talking to this audience today, and grateful for the remarks of Dr. Amer already giving a brief introduction into enteric hyperoxaluria. I recognize I'm speaking to many experts in the field, so I won't belabor this too much, but to give a brief background for enteric hyperoxaluria. Hyperoxaluria, of course, is an important pathogenic risk factor for nephrolithiasis, nephrocalcinosis, and also, as has been mentioned, kidney disease, chronic kidney disease. And while traditionally this has been thought to be primarily attributable to fat malabsorption and subsequent chelation of calcium and bioavailability of oxalate, as was mentioned by presenters earlier this morning, there's ongoing research into the enteric permeability and the role that plays in enteric hyperoxaluria as well as the gut microbiome and how that affects the enteric permeability and excretion of oxalate. You could move to the next slide. This is a disease that affects a large number of people. Dr. Amer already mentioned the role that Roux-en-Y gastric bypass surgery plays in the development of enteric hyperoxaluria. Of course, inflammatory bowel disease plays a large role as well. Exocrine pancreatic dysfunction, chronic biliary dysfunction, really a broad spectrum of enteric pathologies. And while the exact prevalence of enteric hyperoxaluria isn't well defined in the literature, it seems that somewhere between 5 and 24% of those with malabsorptive disease are affected by enteric hyperoxaluria. And to put more specific numbers on that, in the United States, it's estimated that about 250,000 patients have malabsorptive disease and renal stones or renal failure, and approximately 40% of those have concomitant hyperoxaluria. So this affects a large number of patients. Next slide. But despite the large number of patients affected, the treatments that we have for enteric hyperoxaluria remain very limited. There's no specific FDA-approved therapy that we have for these patients. Dr. Amer already mentioned the typical approach, treating malabsorptive disease, which of course isn't always possible for these patients, limiting oxalate absorption, oftentimes through dietary modifications, though these can be difficult to maintain. And then reduction of crystallization, though hydration and citrate therapy can be somewhat challenging depending on what the enteric pathology is that's contributing to EH for patients. Even for this traditional protocol for treatment of enteric hyperoxaluria, there's limited literature regarding the efficacy of these treatments in addition to the other therapies that are being worked on. And one of the main bottlenecks that we have in the development of these therapies is that there's simply not a lot of information that we have regarding the appropriate endpoints that we should be using for this literature. I have on the screen here a study that was published just earlier this year by Langman and colleagues in CJSN about this point, the endpoints in enteric hyperoxaluria. And they went through a number of potential endpoints. Of course, patient-oriented outcomes like renal stone events would be ideal. However, there's limited data regarding the frequency of these stone events, and it can be a difficult outcome to be focusing on, particularly with limited effect size for these therapies. When it comes to other therapies, as they mentioned in this paper, really the resounding theme is we just don't have a lot of data yet. Specifically, we don't have a lot of data to be able to say what effect on the surrogate outcome would lead to a subsequent benefit in stone burden for these patients. If you could go to the next slide. A quote at the very end of their paper was, although we've identified a number of promising endpoints, unable to provide definitive recommendations because serves as some of the motivation for us forming this patient registry is we want to be able to help support, provide some of this data so that we can provide endpoints and support research into creating better therapies for these patients. If you could go to the next slide. So we sought to create a patient registry with both retrospective and prospective components that was focused specifically on enteric hyperoxaluria. And the goals that we had for this registry were, first of all, to be able to additionally characterize the natural history of enteric hyperoxaluria and its associated complications. Second, we wanted to be able to evaluate the relationship between hyperoxaluria and symptomatic stone events. And then finally, we wanted to look at how treatments on these biomarkers, on the urine metabolic profile, affects subsequent symptomatic stone events to try to help with addressing those questions I spoke about on the previous slide. Moving forward, talking about some of the methods, and some of this will sound similar to what was mentioned earlier on the other stone registry that was mentioned today, if you could move forward. But we've gone through the EMR for all 3 of the Mayo sites looking for patients who have a history of recurrent calcium oxalate nephrolithiasis. As was mentioned by Dr. Keddis, we have a cross-disciplinary stone clinic that allows us to have good access to these patients within our system. Of those patients, we're looking at those who have a 24-hour urine oxalate measurement greater than 40, and we're looking for associated enteric diagnoses that are known to be associated with EH. And for all of these patients, we've then manually gone in and screened to ensure that any hyperoxaluria and stone events are reasonably attributed to this enteric pathology so that we can appropriately term these people as having enteric hyperoxaluria. Next slide. In terms of the data that we're actually gathering for each of these patients, we're getting data on some of the baseline characteristics. This includes their history of stone events and their history of stone imaging, so we can look at that as a potential biomarker. We're also looking at their enteric disease and the status of that enteric disease at the time of diagnosis, along with some of their baseline urine and serum studies. And then at each of their subsequent follow-up visits, we're getting more information about interval events and interval imaging, updated urine and serum studies, and then information regarding the treatments that these patients are undergoing so we can correlate that with some of the changes in outcomes. And then finally, we're also including data on endpoints, including ESRD, dialysis, transplant, and death for these patients. Next slide. There are a number of challenges that we've, of course, come into on this journey, nothing straightforward. One of those is that a lot of the data that we're dealing with is unstructured data. A lot of this is entered free form into the chart. And this, of course, leads to issues with efficiency, but also leads to issues with standardization, as we have a number of contributors who are working on abstracting this information. So this has required a lot of communication amongst us, frequent updating of a common protocol. There's also some incomplete data. Not all stone events are captured cleanly within the notes of their managing provider. So we've had to look at other specialties as well. Sometimes stone events are mentioned specifically with their primary care provider or with gastroenterology. So searching the chart broadly has helped with some of that incomplete data. And then, as I mentioned, that we have multiple collaborators, and there are these issues of trying to standardize our approach to gathering this information. Of course, there's also a need for a review of the quality of that information. So trying to incorporate ongoing quality checks and planning so that we can do those quality checks appropriately has been another challenge that we faced. So talking about some of these preliminary findings, which, as Dr. Keddis noted, will be presenting it in poster format at ASN this year. Next slide. This is just a sampling of some of the baseline characteristics of these patients. It's pretty well split between male and female, but as you can see, it is a predominantly white cohort with a relatively preserved GFR on average, and a urine oxalate with median of 53.7. Next slide. In terms of the specific enteric etiology contributing to the EH for these patients, about 50% of these patients are have bariatric surgery as the primary etiology, with the vast majority of these being Roux-en-Y gastric bypass surgery. However, inflammatory bowel disease constitutes about 25% of the patients, 50% of those remaining after bariatric surgery, and pancreatic insufficiency, another good portion of these patients. Next slide. This is comparing some of the characteristics of those with and without bariatric surgery as their primary etiology for EH. And if you can advance one, Dr. Keddis. A few of these have statistically significant differences. Of course, unsurprisingly, BMI is different between those with and without a history of bariatric surgery. Those with bariatric surgery more likely to be female. And then in terms of the history of stones prior to their diagnosis of enteric hyperoxaluria, those with bariatric surgery were less likely to have had overt symptomatic stone events, more likely to have had stones detected by imaging, which isn't entirely surprising given that these patients may have had more imaging studies being performed. Moving on, oh, sorry, staying on this slide one more moment, I wanted to point out that while not statistically significant, looking at some of the baseline urine studies, the urine oxalate doesn't differ significantly between these two etiologies. However, there is an interesting trend toward statistical difference in the calcium and citrate, both of which were higher, though not statistically significantly so, in those with bariatric surgery. Next slide. This is looking at the time to first event following diagnosis with enteric hyperoxaluria for all of the patients that we have so far. I didn't mention that that's a cohort of about 74 patients overall, and little more than 60 of whom have long follow-up with which we can look at these numbers. So as you can see, there's, oh, sorry, staying on that last slide, there's a pretty wide variability in terms of the frequency of stone events following diagnosis. So by about 5 years, 50% of patients have had a stone event. And moving on to the next slide, there is not a statistical difference in the those with bariatric surgery or without in terms of their time to first event. So in conclusion, some of the future directions that we're excited to look at, next slide, we've only looked at fewer than half of the identified patients with their data fully abstracted. We look forward to fully abstracting some of that data, of course, continuing to clean and accumulate controls for these patients that we've already looked at. And we look forward to including additional sites into this database. And then finally, in terms of additional investigations that we hope to perform on this data, we look forward to looking at some of the differences in the natural history of EH by etiology, looking at the effects of treatments on various endpoints, and correlating some of these changes in biomarkers, especially urine oxalate, with patient-oriented outcomes like stone occurrence. So finally, just to acknowledge the team that we've been working with, Dr. Lieske and Dr. Keddis have been very helpful. I'm very grateful for their mentorship. And then we have a number of other contributors, and I'm grateful for their support as well.
Thank you very much, Dr. Keddis and Dr. Tatton. Thanks for giving us your perspective on how you too have leveraged your data there to build a registry and to study it and tell us more about natural history. So we look forward to some of those future directions that you mentioned. So thanks again. All right, next we welcome my colleague from Department of Urology, Dr. Kevin Weimer, who is an accomplished health services researcher and is going to be sharing with us about the costs of surgical stone management. Dr. Weimer.
Thank you all for the chance to participate today. As Dr. Koo mentioned, my name is Kevin Weimer. I'm the newest urologist here on staff, having only been here on staff for the past 2 or 3 months. And today I'll be talking about the costs of surgical stone management. We'll go over a few topics. Obviously, this is a large thing to discuss. We'll kind of start by looking at societal costs and sort of the large overview of how these costs impact us as a whole, and then focus more on patient level costs and how this leads to difficulty with access to care, and ultimately how we can use some of these data to improve patient care. So we all know stones are highly prevalent. The prevalence is increasing. And importantly, this is a recurrent chronic disease. It's not a one-time insult or one-time occurrence. So it's not surprising that with that, there are high costs, with estimates over $5 billion per year in the US, 3 million lost workdays per year in the US, and per patient incremental costs being similar to those of other high resource chronic diseases like diabetes and asthma. That $5 billion figure came from this paper published out of UCLA back in 2004. They looked at a medical expenditure database and a workplace database to estimate yearly direct and indirect medical costs associated with stone disease, and found that on average, a diagnosis of nephrolithiasis resulted in double the direct medical costs per year, with an absolute difference of about $3,500. Indirectly, it led to 19 additional hours of lost work per year. Thanks, Dr. As far as the cost of surgical intervention, so this was a study that our group published a few years back, looking at intermediate sized stones, 1 to 2 centimeters in size. And we did a cost effectiveness comparison looking at different surgical interventions for these patients. It included ureteroscopy, PCNL, both standard and mini approach, and shockwave lithotripsy. And I'll try not to get too much into the weeds on cost-effective analyses. I know that it's late in the day, but simplistically, these are models that are kind of structured here on the left, where you start on the left side of that tree with the blue square, and you have a decision between multiple management options or multiple treatments. And then a theoretical group of patients moves to the right through that model. And each of those treatment strategies are associated with health states, which are then accompanied by specific costs and effectiveness values. For example, for our model, the health states were things like stone-free, recurrent stone, postoperative complications, and the likelihood of each of those was driven by probabilities that we got from meta-analyses published in the literature. And again, you have costs, which can be Medicare, private payer, or patient-based. In our model, it was Medicare costs, and effectiveness, which is measured using quality of life, and the standard metric for that is QALYs, which is sort of a weighted average for quality of life. These were the results at 3 years for our model. So on the left, you see average 3-year costs. This includes the index procedure, follow-up stone procedures, cost of complications. Second column is the effectiveness, again, measured in those QALYs, with a maximum being 3.0, and then some comparative values for how to compare the cost effectiveness between these. And overall takeaway here was that on aggregate, ureteroscopy was the best balance of a relatively low index procedure cost with a relatively high stone-free rate, and resulted in significant improvement in regards to cost effectiveness compared to the other interventions. For all these models, we run a bunch of sensitivity analyses where you basically vary a bunch of the factors going into the model to see if anything changes the outcome. Main takeaway from these graphs is that particularly for lower pole stones, if you were willing to pay more kind of as a society, PCNL and specifically mini PCNL may become cost effective, basically representing that it led to a higher stone-free rate, slightly higher quality of life, but at an additional index procedure cost. The same was not true for non-lower pole stones, where stone-free rates between those approaches were not as pronounced. So those are kind of the model-based costs, again, looking at Medicare data and literature outcomes. We know that that is not, quote unquote, real world. And so this was an attempt to get a better sense of these real world costs. This is using IBM Watson, which is a large data set that includes both Medicare and private insurance costs. This graph represents Medicare patients, and this is still kind of preliminary, so I apologize. It's not as polished looking, but there's a lot of data here. But the main takeaways here is each of these bars represent a different index procedure. So we looked at these patients between 2014 and 2017, and if they underwent a stone surgery, that first stone surgery that they underwent is how they're categorized here. And then you have index and follow-up costs, and they were followed for 2 years. So the index costs are blue and red, follow-up costs are green and purple. And the separation is based on kidney stone specific costs, which are blue and green. So these were costs directly related to a kidney stone surgery. And then non-kidney stone costs at the index procedure, which are red, and during the 2-year follow-up, which are purple. And really, simplistically, the main thing that I want you guys to take away from this here is, for example, if you look at PCNO on the far right for large stones, the index procedure cost is about $14,000. It's not all that surprising. That's near the Medicare reimbursement rate, and that's what you'd expect. But if you look at the kidney stone specific follow-up costs in green there, it's over $30,000. So this is, again, just highlighting that within 2 years, these patients have multiple stone episodes, whether that's stones that weren't treated the first time around, recurrent stone events, but this is an expensive, recurring, chronic process that accrues a lot of costs. So trying to transition that from this broader picture to more of the patient level and how these costs impact patients, we start to get into the area of financial toxicity, and obviously that leads to issues with access to care. As some of you are probably aware, financial toxicity is a term that initially kind of started in the oncology realm, but refers to the objective and subjective impact of cost on patients' care and overall quality of life. It doesn't take long to see that medical debt and financial toxicity is hugely prevalent in the US, and staggeringly so. There have been a lot of studies on this, but recently two that came out, one in JAMA said 1 in 5 US adults surveyed had some level of medical debt. And then one that was a partnership between Kaiser and NPR actually found 40% of US adults report having some level of medical or dental debt. And of those, 1 in 8 have $10,000 or more. It's actually the leading cause of debt in the United States. This was a map based on that study. It shows that there are differences in level of debt throughout the country. But again, the main takeaway here is that this is an extremely prevalent problem, and whether or not we focus on it, it is going to be a huge issue for all of our patients, or at least a large majority of our patients.
Same map of kidney stone prevalence.
Yeah, and I mean, the South has higher rates of issues with medical debt, and this does have a concordance with stones as well. So not surprisingly, stone disease is not immune to these effects. Unfortunately, there's not been a large volume of research looking at financial burden for stone patients, but this was a nice paper that was published last year by the group out of Vanderbilt, and they looked at 1,000 patients within a registry and looked at the Wiscall survey as well as the cost survey, which is a standardized metric for financial toxicity, and found that 1 in 4 patients met criteria for financial toxicity, so sort of met this threshold for having financial toxicity that was disease specific, so directly related to treatment and management of kidney stones. These patients were more likely to be female, Medicaid patients with recent stone events, and those, the criteria for financial toxicity associated significantly with poor quality of life across all domains on the Wiscall. Our group did a project looking into this area as well. We used the All of Us Research Database, which is a NIH database targeting at recruiting a diverse patient population. We identified roughly 10,000 patients with a diagnosis of stone disease and matched these based on age, gender, race, and medical comorbidities to a control group. And our main outcome was looking at access to care based on these 4 domains. And these were based on patient survey responses. We ran analysis first looking to see if the diagnosis of stone versus no stone made a difference as far as association with these outcomes. And then within patients or among patients with stones, seeing how these demographic variables may or may not impact access to care. What we found that overall, the diagnosis of stone actually did not correlate significantly with a lot of these outcomes when you compared the stone versus non-stone group. But when you looked among patients with stones on multivariable analysis, we found significant associations between a lot of these demographic variables, including younger age, female sex, higher medical comorbidities, lower socioeconomic status, lower education, corresponding, not surprisingly, to multiple aspects of worse patient access to care. So overall, the main takeaways, number 1, stones are expensive. Number 2, that expense varies significantly based on how they're treated and what type of surgeries we perform. This would obviously expand to medical management as well. And number 3, patients are facing an increasing burden of costs. Insurance companies are transitioning more and more costs to patients. Most recently, estimates are 12% of total healthcare costs are now patient out-of-pocket costs, and this is only going up. And so for all of our patients, including stone patients, financial toxicity is common. It's impacting patient access to care, and this impact is not equal. It impacts disproportionately poorer patients, sicker patients, those without insurance, and female patients as well. So now that we identified all of this depressing information with cost, the real question is what do we do about it? And obviously this is a large, complex question that's not going to be easily addressed, but I think it's imperative that as clinicians and researchers, we are involved and knowledgeable when it comes to cost effectiveness, and how this impacts our patients. And by at least being able to be involved and knowledgeable with that conversation, we can help to guide some policy, whether it be reimbursements to identify cost-effective interventions and prioritize those, or truly understanding which patients need to be targeted to help improve access to care. But ultimately, as clinicians, it is our responsibility to have some knowledge in this area to help guide these decisions, because I think we are best suited to, from a clinical standpoint, with the hope being somewhat of an improvement in delivery of high efficacy treatments. Thanks.
Thank you very much, Dr. Weimer. Finally, we're going to close out this session on the voice of a patient with our colleague, Dr. David Sass from our Division of Pediatric Nephrology.
So I'm just going to be talking a little bit about the voice of the patient. So I'm a pediatric nephrologist here at Mayo, and I specialize in kids with kidney stone disease, and in particular, rare kidney stone diseases. So just, going over sort of the traditional model of research and how this is evolving over time. This is sort of the traditional, I'm a scientist, I have an area that interests me, I have a question I want to answer, so I'm going to study it. So that's basic science research. We heard a lot of that this morning, right? And then enters the physician. So, the physician or the clinician is faced with patients that say, gosh, I have a lot of patients with this particular problem, and we have a gap in our knowledge. We should study that. And so they can do clinical research. And then together, sometimes basic scientists and clinical researchers would get together, and we would call that, bench to bedside or translational research. And that's the way it's been for a long, long time. But now in more recent years, we've started to think more about getting patients and families and other stakeholders involved, asking the questions, what actually matters to them? Right. So we think we know what our patients need, and the basic scientists have an idea about it too. But it turns out somebody smart said, hey, why don't we actually ask the people we're taking care of? And so with that, we are able to understand the patient's priorities. And where those three triangle or circles meet is where we've landed today with patient-centered research. So there are a few different names about that we refer to patient-centered outcomes. There's CER, which is comparative effective research, which incorporates the voice of the patient. So this is where research is certainly moving. And so there is this organization called PCORI. So it's a nonprofit research organization that was authorized by Congress in 2010 and signed into law in 2019. And so what this organization does is it funds patient engagement and comparative effectiveness research, or CER. And so they provide grants for people and organizations to first prove that they can engage with patients, and then move forward with trying to create patient-centered research plans. So we're currently writing up a grant that we're getting ready to submit entitled Engaging Stakeholders to Develop Patient-Centered Research in Monogenic Kidney Stone Disease. So you can see in this table here, this is a table of 5 different rare stone diseases that we specialize in and have registries for. Let me move this out of the way in case anyone wants to know what year the Cystinuria Foundation was. Gail's here; she could just tell us, because that's what I did. I emailed Gail and said, "What do I put? How do I fill in this table?" So, so these are different groups that we've been working with, and so these patient advocacy groups are connected with all of the patients, or at least a lot of the patients with these conditions. And our plan is to engage these patients and their families affected by genetic kidney stone disease, and specifically, we're going to talk about genetics, right? So we do a lot of genetic testing, and there are all of these emerging gene-based therapies. And we just assume, okay, well, gosh, if my kid had had a bad genetic kidney stone disease, sure, if somebody developed a gene-based therapy, I would give it to them. But that's not necessarily true. Turns out there's a lot of distrust of the medical industry, and certainly when the word genetics or genes gets involved, people get skittish. So what we wanted to do is as we're so we help develop the clinical trials for some of the recent gene-based therapies, and there are a lot more coming. And so we said we should really incorporate the voice of the patient, make sure their concerns are being heard. So we'd like to, survey them, find out their attitudes about genetic testing. By the way, a lot of them have concerns about, where do the results of who owns the data? Is my privacy going to be safe? So we're going to do some surveying, learn about their attitudes, and then the intervention would be provide some education about it and address their concerns, and then do a post-interventional reassessment and hopefully see that they feel more comfortable, or at least that their needs are being met. And so this first one is sort of a preliminary thing, sort of a proof of concept, and hopefully would lead to more funding down the road to do more of this sort of thing. So just as I think a primary example of engaging with the patient. So this Voice of the Patient report came out October 5th, 2020. It was put out by the OHF. And so the reason why this was a critical report was that for years and years, patients with PH1, there's no FDA-approved treatment, right? So there was the standard of care, hyperhydration, potassium citrate, really that's what we had to offer. And when there were proposals for clinical trials for drugs, the FDA was really pushing back, saying, you've got to use traditional endpoints that we've traditionally used for other kidney diseases. Otherwise, that's the only thing we're going to allow. And it wasn't until the Voice of the Patient came out and said, dearest FDA, we are desperate. Our children, they're at high risk for death. They're at high risk for, needing a liver and a kidney transplant. And we understand the risk of departing from the traditional outcome measures. We understand all of that. We are willing to take that risk for ourselves and or our family members. And that's what led to urinary oxalate excretion being an acceptable endpoint for the FDA, which then led so first of all, led to this publication by two of our primary hyperoxaluria parents, but more importantly, led to the FDA approving initially lumasiran in 2020. And then this is, hot off the press is October 2nd, FDA approves nedosiran. So, and by the way, all the applause go to the patients and our research coordinators who make all that happen. So, these are the leaps and bounds we're looking for. These are the game-changing drugs that are improving the lives of our patients. And again, it only happened because of the Voice of the Patient. So that's what we're looking to do, is to continue to do that. So I would encourage everyone who's doing research to consider getting the Voice of the Patient involved early in when you're designing your clinical trials.
Well, I want to thank all of our speakers in this session, and certainly Kim, who had to step out a little early. But thank you for bringing these diverse perspectives, which I think have been a great complement to the perspectives at the more genetic and molecular level we heard this morning. Because really, it's only when we bring these perspectives together that we really can advance the science we're doing and help to inform each other and the work that we're doing. So thank you to all of you who participated in this session. Or welcome back, everybody.
Thanks.
And to moderate our last session of the day, Dr. David Goldfarb, your friend and colleague and mine, to lead us through our final speakers on AI and EPI tools for stone disease. Dr. Goldfarb.
Okay, amazing. Terrific. Kevin, thank you so much. And today, our first talk, Project One, is AI possibilities in clinical research, and Dr. Jaleh Zand is here to do that.
Well, hello, everyone. My name is Jaleh Zand, and I oversee AI efforts within Division of Nephrology and Hypertension at Mayo Clinic. And I'm going to talk about AI possibilities in clinical research. So why AI? Why would you want to consider, for example, AI compared to traditional statistical methods? One of the advantages of AI is that AI is much better in handling complex and nonlinear relationships. And this is very helpful for healthcare research, as a lot of time we have variables and features which have a very complex relationship to each other. Another advantage of AI compared to traditional methods is that it allows for automated feature learning. So if you're using an AI model, you don't need to upfront decide what are the features that you want to use. You can potentially allow the model and the algorithm to decide that for you. And again, this could be useful in healthcare, as a lot of time within our research, we're going to have high dimensional data. The next reason or advantage of using AI is that AI is much better in dealing with big data problems compared to statistical methods. It can extract information much better, and it can do a better pattern recognition. And again, this could be helpful because a lot of times in healthcare we have a big data problem. Finally, AI is much better in generalization compared to traditional methods. And this could be very helpful in healthcare, as a lot of times we have a diversity in our settings. So for all these reasons, it makes sense to consider AI for clinical research. Now, there's so many areas that we can use AI for in clinical research. I'm going to mention some of them in this slide. You could use AI for patient recruitment and eligibility for your research. You can also use it to design protocols. It's also helpful for data creation, data quality, and data management. It can be used for literature review and mining information from text. It can also help us with drug discovery, dosage optimization for patients, also optimizing any type of treatment. We can use AI models. Another advantage could be for resource allocation and improving efficiency. It also can be very useful for decision-making, prediction, and risk assessment. Finally, an area in AI which has been talked about a lot in the recent years has been generative AI. And generative AI could be helpful in research, especially if you're thinking of any text and imaging tasks. Now, in this talk, I'm going to mainly focus on the last two items on this slide. And I'm going to start with decision-making, prediction, and risk assessment with focus on kidney stone research. So how could you use AI, let's say, within kidney stone research? One way you can think about using it is for diagnostic prediction. You can think about sort of a risk assessment of kidney stone in general population. This could be individuals who, for example, have had kidney stone in the past, but it could also include individuals who have never experienced kidney stone. And you can use AI models to assess the risk for developing kidney stone in the future. Another way we can use AI in population that has had a kidney stone is to identify the risk factors which contributed to the individual developing the kidney stone. Now, as we all know, for many medical conditions, the causes could be multifactorial. It could be genetic, it could be environmental, it could be lifestyle, and AI models good at identifying for each individual which one of these factors contributed to their kidney stone. And this way, we could set up better preventative measures. Another we can use it is in patients with kidney stone, is that we can sort of categorize the type of kidney stone that a particular individual has had. This can be done using EHR and clinical notes. It can also be done using images of the kidney stones. So just to summarize, it could be beneficial to use AI for decision making. It can improve our understanding of individual risks, and that can in return sort of improve our treatment protocols and our preventative measures. And if implemented properly, it can reduce the burden on healthcare staff and also reduce costs. So if you're planning to use AI for decision-making in your research, there are a few points you want to consider. The first is for the good performance in any AI model, the model is going to be reliant on high quality data. So it's important that we have access to good quality data. Also, if you're planning to extend that research and put it in production, it's important to make sure that we have access to live real-time data. Also, if you're building an AI model for decision-making, it would be very helpful to include the explainability component. This will make us understand why did the model arrive to the decision that it did. It would also make us to be able to trust the model more. And it's also helpful with identifying biases and errors with the model. If you're trying to then use that model in long term, potentially in production, you would also want to make sure that you keep monitoring the model, make sure that it maintains accuracy and safety. And from time to time, you also want to probably improve the performance. Finally, it's important to make sure that the model as well as the data are going to be compliant with regulatory and privacy rules. And within healthcare, we want to make sure that it's compliant with HIPAA rules. So the next topic I'm going to cover is generative AI for text and image tasks. So what is generative AI? Generative AI models are a group of models that generate data analogous to what they were trained on. It's important to mention that generative AI actually has been around for quite some time. It has been talked about a lot in recent years because of the latest development in the area of generative AI. And these developments in terms of images has been diffusion models, conditional image generation models. And for text generation, it has been large language models or LLMs. Now, how could we use generative AI to help us with our research in healthcare? I think the most significant way that we could use generative AI would be to transform unstructured data to structured data. This could be very helpful in healthcare as approximately about 80% of the information that we have in healthcare is unstructured data. And by that, I mean things like clinical notes, it could be handwritten prescriptions, pathology reports, any social media which is relevant to healthcare. It could be voice recordings and the like. And if we can access to that information, this is the information that normally you cannot use for your models because it's very hard to extract the information from it. Using generative AI and put it in a structured form, we can access this data. And this is obviously going to then improve the quality of data, and it's going to really improve the performance of our models. Another way you can consider using generative AI is if you're doing research which includes images. you can use generative AI to improve image quality. This can be done by, for example, improving the resolution or, for example, denoising. Another way we can take advantage of generative AI is to generate synthetic data. This is particularly useful if for your research you don't have enough data. So for any type of a research that you lack data, generative AI can help us sort of generate synthetic data. And that can in return improve robustness and the generalization of the model. Finally, another way which could be very beneficial to use generative AI is for literature review. So in your area of research, you can look at any journal articles and generative AI can summarize that for you. You can do, for example, keyword extraction on those articles. You can also get access to journals that are not in English. That's because generative AI is able to translate and summarize for you in English. So this way you can get a much better sort of view of what's happening in your area of interest. So as I mentioned, these were the benefits, and it can be very helpful to use generative AI, but it's important to understand it's not the answer to everything. And I'm going to talk about some of the shortcomings of using generative AI if you're planning to use it. So there are, I would say, some concern about data security. And most generative AI models at the moment are not HIPAA compliant. Although we have the sort of a PubMed from Google, which is HIPAA compliant. So it's important if you're trying to use it to make sure that you're using the model which is compliant with HIPAA rules. Another issue is hallucinations. That means that from time to time, they can give you a wrong answer, or they can just make up things that are not facts. They also lack from transparency, can be inconsistent at times, and have a variable performance. There are also some questions around their adherence to copyright laws. And they could also suffer from citation inaccuracies. Also, because they train on a large amount of data, and this data can be anywhere. This could be the data, for example, from internet or any source. It's very hard to address fairness and bias when using generative AI models. Another shortcoming is explainability. So it's very hard to understand how the generative AI model arrived to the decision that they did. There's also limitations to logical reasoning. They don't understand logics in the same way that us humans understand logic. And also similar to logic, they also don't understand the causal reasoning. So for these reasons, it would be helpful from the outset to think what's the type of a task that you want to use generative AI for. If it's a low-risk task, then it's great. If it's a high-risk task, something like, for example, treating patients, then you don't want to use generative AI for. So overall, it would be good to have some sort of a risk-based approach where you can assess the benefits and the risks and then make a decision if it makes sense for that particular research to use generative AI or not. Now, some final thoughts. If you're planning to use AI in clinical research, I think it would be helpful from the outset to decide if you're using AI strictly for research or if this is something that you're thinking more long-term, and in the long term, you want to use that research in production and sort of applied AI. The reason for that is that there are different objectives. Research AI often focuses on exploring new frontiers, and there is a large focus on performance. If you're using AI for production, however, the focus is mainly on automation, infrastructure, scalability, speed, memory, and real-time data availability. Now, some other final thoughts for using AI for research. As I mentioned earlier, data quality is very important, and this is something you want to think about upfront. Also for healthcare, you often have very long development cycles for the research. So it's important to think of the timeline in advance, how we want to sort of incorporate the AI research within that timeline. Another thing to bear in mind is cost and resources, and this is not strictly human cost. Very often when you're using AI, you have to consider computational costs and infrastructure costs. And finally, for again, any model, any data that you're using, you want to make sure that we're compliant with ethical constraints and sort of any regulatory rules. Well, thank you for listening, and let me know if you have any questions.
Thanks. Have you all published a paper that is AI based on kidney stones or kidney disease? That's something that would serve as a model, something I'd like to look at.
That's a question for me.
Yeah.
Yeah, right. So in kidney stone, no. We currently have a very big project on AKI, and we are working on that. This is going to be more sort of on the applied side, and yes, we are planning to publish on that. On kidney stone, I personally haven't done any sort of a published research in this area.
Okay. Other questions? Can AI help me publish my paper?
That's a good question. Well,
I've always been hallucinating, and it's not right. That you know, we read so many stories. They're really anecdotes about people asking ChatGPT questions and then having things quotes attributed to people that have not really been said. Not real quotes, right? So that's sort of an example. Right. I haven't seen that occur, or no one's told me an anecdote about that occurring in the research realm.
Sure. Like as I mentioned, those are part of, I would say, downsides of using generative AI. There's definitely problem with citations. There are inaccuracies. Again, one problem is it's also very random. So you don't know when it's going to happen. You know, it could get it right 90% or 99% of the time, but then occasionally it has that one-off errors, which is very hard then to, if you don't know about that topic too well, it is very hard to understand that error. And so at times it could be misleading. That's why I think it's very important that you use it for something that is not high risk. And by that, I mean, if you make errors, the stakes are not going to be so high. Right. For such a task, it's great. It definitely can really make you efficient. Otherwise, yes, I agree that those are the shortcomings that we have to be very careful about.
It's like doing research with a fellow. Yeah. Okay, Lama.
For generative AI, I don't have a sense of how much data is needed. Like, do you need 500 patients, 1,000, 100,000 patients, or like data points? Because it's not very clear. And is it something that cannot be used unless you have very, very large data?
Right. So actually, no, you don't. Technically, you don't. I mean, the model itself has been trained on large amount of data, right? It has gone through all the internet, especially the kind of the most known ones, such as ChatGPT. These are like being trained on really huge amount of data. But so because you're trained, you actually don't need to, have a lot of data. I mean, it depends on your purposes, but you could, if it's just, you want to get a summary of, let's say, an article, you just put your article, you just ask it very easily, like, give me a summary. It's quite actually efficient. Like, you would be surprised. It just, summarizes for you. You can also, tell it, I want it specifically in, for example, 3 sentences, right? So you can put constraints and limitations around what you want it to achieve. And again, it depends on what you're trying to do, right? I think for this type of task is actually great, right? It can make you really efficient. Now, your question, for example, about writing papers, I don't think you can publish the paper using generative AI, but in fact, I think it could be helpful in sort of editing for you, right? It's actually great. It has a great power on editing, and it can do any style of conversation that you want to have. You can say, okay, make it friendly, or make it more formal, or make it for a research, and it can make all those adjustments to your text.
Great. Terrific. Thank you very much.
Thank you.
Our next talk, number 2 for today, is regarding clinical integration of quantitative stone image analysis by Dr. Timothy Klein.
That was perfect segue. So yeah, I'm going to talk about work that we've been doing, bringing AI tool into the clinical practice in radiology. I'm an AI scientist in the department, essentially tasked with trying to bring AI tools. So we partnered with the CIC, Dr. Ferrero, who's in the back and talking next, who had developed the quantitative stone analysis software, and essentially hooked it up with some of the automated tools that we had to make an automated report. So I'm going to talk a little bit about that development and what we've been doing to monitor the algorithm as it's been put into the practice. So a quick overview, we call the tool QSAS AI, so Quantitative Stone Analysis Software. It provides a fully automated segmentation of kidneys and renal stones. The algorithm generates a report capturing an overview of a patient's stone burden as well as metrics for the individual stones. And this was deployed into the clinical practice back in July of this year. Currently, it's supporting the clinical practice in Rochester, Arizona, Florida, and the health system, so essentially enterprise-wide. And it's applicable to both single and dual energy CT exams. And really the benefits of this are an improved workflow, also helping with a detailed and standardized reporting in the radiology practice, and ultimately improve patient care and monitoring. So this is a really high-level overview of what the algorithm looks like, but essentially, as soon as a patient is scanned, if it's a relevant exam, it's automatically routed to the algorithm, and the algorithm performs detection and segmentation of the kidneys, does some basic post-processing of that mass to do things like include the renal pelvis, and then is sent to the QSAS software tool, which then segments the stones and allows us to automate a reporting of the stone burden. So the example on the right is the automated report. This is something that can be pulled up by the radiologist through Visage. And what they see when they first pull this up is this kind of summary view where we have a 3D rendering of the kidneys. We show the detected stones, and then provide a high-level summary of the stone burden. So in this, we're showing the number of stones, breakdown of stones in each of the kidneys, as well as volumes, and then kind of a histogram breakdown of the stone sizes. So in this case, the patient had 2 stones smaller than 2 millimeters in the left kidney, and so on. Then also within this DICOM series that's generated, we can scroll through and then actually get details of the individual stones as well. So here's just showing a result of the individual stone report. So here showing the detected stone, as well as metrics like volume, max diameter, information about the CT numbers, and then we can also do composition. Right now doing composition for uric acid, non-uric acid, as well as indeterminate stones. And then we do a color coding of the stone similar to other commercial softwares that are showing the composition overlay. So there was a lot of work to get to where we are today. So I kind of wanted to highlight a bit of the background in terms of what needed to take place from sort of the idea into post-production monitoring now of the algorithm. So we obviously had to develop the kidney segmentation model, and there we looked at performance, so largely trying to make sure that we get an algorithm that can perform as well as a human reader. So need expert segmentations by our radiologist colleagues for that. We also assess things like bias and fairness of the algorithm. I think that piggybacks well off of what Dr. Zand was highlighting, essentially wanting to make sure that there's no factors like, based on patient sex, BMI, maybe scanner variables that could make the algorithm not perform as well. We also had to do comparisons with the existing research tool that's been developed by the CIC, and compare what we're doing with the AI to the existing workflow that's being done in the radiology practice in Rochester by our 3D lab, where they're essentially providing information about overall stone volume burden. So we're trying to provide a much more granular report for both the radiologists as well as the caring physicians as well. And once we had everything sort of ready to go, we went into a reader validation study. And I'll talk a little bit more about that to get us to the place July where we were able to roll this out. So in terms of the kidney segmentation model, we have a lot of experience developing these type of AI tools. Just wanted to highlight what we were looking at. A lot of times people ask about data set sizes, you know, what's required. So here we trained our model on 300 cases and had a holdout test set of 100 cases. Again, really with the goal of getting the AI algorithm to perform just as well as human readers. On the bottom left, you can see the inner observer variability for this task is typically 0.92 to 0.98. This is a metric called DICE, which essentially compares the similarity of the segmentations or tracings. And we saw that our AI was performing just as well as the human readers. One important thing to assess with these AI models, obviously, is whether there's any biasing factors that might be occurring. So here we just assessed in our test set, looking at factors like sex, BMI, age, race, really trying to understand if there, some of these lower performing cases, if they had any significant effects based on these demographic variables, and really not seeing anything. Fairly similar performance for patients, male, female, different BMI, et cetera. So then we moved into our reader study. So this was essentially a prospective study where we were running the algorithm sort of in a shadow mode where it wasn't being available to clinicians or patients, for instance, but running everything to make sure the workflow was solid, making sure that the radiologists were happy and would accept the output of the algorithm. And really we saw that in general, the scores showed a good deal of acceptance of the reporting that was occurring through the QSAS AI tool. So then we built a dashboard to monitor the algorithm. So this was taking place during that shadow mode, again, to understand basically how many cases we'd be hitting, looking at, metrics like the number of stones that were detected, and other important things for someone like myself to see if there's any changes in processing time of the algorithm, if the algorithm's actually failing. These are the things we want to be able to monitor and catch. So one thing that was interesting during this limited rollout was we weren't actually seeing any errors with the sort of algorithm itself. We were actually seeing errors that were being generated when we tried to automatically make these sort of secondary capture views. And so we had errors where the kidneys were at the edge of the field of view, as well as handling cases with only a single kidney. So this was something that we were able to update then before we went live with the algorithm. So that moved us into our second reader study. We made a more refined survey for the radiologists to complete as they reviewed cases, but really trying to get an assessment before we went live that there would be a high level of satisfaction, not only impacting the workflow, but also the quality of what we were reporting. And again, with our analysis here, we saw a very strong agreement between the radiologists in terms of their acceptance, in terms of what was being captured within the reports. There was some variability in one particular case that had microcalcifications. This is something we've continued to see as we've gone live, that sometimes we're not capturing something that maybe the radiologist would like to report on. This is just some more details there. I might just move on with the interest of time. And then also assessing the quality of the renderings that were being generated. Here, just highlighting that one case that was identified, where here you can see in the report, the punctate calculi in the lower pole of the right kidney, which was too small to be characterized with the imaging. So we've continued to expand our monitoring, and obviously now we've gone live at the majority of the sites, Florida, Rochester, Arizona, and the health system. You can see that we've processed over 1,700 unique patients. and also automatically measured over 8,500 stones as well. So I think pretty exciting. And then this is just a breakdown looking at the algorithms that we have currently in the practice. So these are AI tools that have been developed in-house that we're running live in production. Here I'm just highlighting QSAS. You can see this is the look at 1 week. So essentially we had 85 cases that were run successfully, didn't have any failures. And then you can see the number of acceptance and rejections from the Rocket tool. So this just means when it's accepted, now it actually flows through and lands in the patient's report. Just quickly, some reasons for the rejections that we've seen. It's obviously going to struggle with severe metal artifacts. This isn't surprising. Just showing what it looked like on the left with the QSAS tool, just getting false positives based on those streaking artifacts. And then on the right, you can see a commercial tool that essentially completely fails for this case as well. And then finally, a quick timeline, what it looked like. So this was brought forward to my team back in March of 2021. We essentially prioritized it later that year, and then all the work I just showed took place, and now we're running in full rollout, automatically generating a PowerScribe template for the radiologist to include within the reports, and hopefully satisfactory for the nephrologist colleagues that are looking at these cases. So with that, I wanted to acknowledge the team. I worked really closely with Dr. Ferrero on the QSAS tool. On the radiology side, worked very closely with Dr. Petretsky and Dr. Fremming. Got a lot of good input from our nephrology colleagues. And then there's a lot more people that could be acknowledged. I work for the team called FAST. We have IT support, project management, and data science support. So with that, thank you, and I welcome any questions.
Congratulations, nice work. How much of this would be applicable in outside of your places where dual energy is not the standard of care?
Yeah, so that's a great question. So we are actually running it on single energy. And in single energy, we're doing all of the same sort of quantification other than composition. So we really need the dual energy to do any of the composition work. But actually for the health system, we're mainly just running it with single energy exams. Yeah.
Cool. Okay. I know that in the past, we heard stuff about trying to separate calcium phosphate from calcium oxalate. Is that something you've been working on?
So Dr. Ferrero could probably comment a little bit more. We're considering additional composition measurements, but I think right now we'll probably stick with the uric versus not.
Yeah,
it's a difficult problem.
Right. Yeah. Understood. Great. Andrew.
How does it can it be used on images that have contrast enhancement?
No, we're not using it with. It's basically non-contrast.
That might change.
That could change.
Hold on 10 minutes.
Do you have a particular application though that you're thinking? Or?
I'll be presenting.
Okay.
So I apologize, I may have missed it, but as far as next steps for this, I know Dr. alluded to it, but do you guys have anything that works as far as, so we're getting a volume, but can we predict how long it'll take the laser or angles to access these stones as far as intervention and being able to predict intervention based on the stone?
I mean, I think that would be something for next steps, but nothing we're targeting currently. I think some of the things that are earlier on the horizon would be automatically kind of the longitudinal report. So, getting the information from prior exams and incorporating that automatically, but any of the kind of interventional planning, that's probably a longer roadmap. Yeah. That's cool. What's that?
Let's talk.
Yeah. All right. Sounds good.
Great. Thank you. Our next talk is nephrocalcinosis versus stones by imaging. Dr. Ferraro.
Thank you for inviting me to talk to this. As Dr. Klein was alluding to, we've been working on QSAS for the quantitative cell analysis software for close to a decade, basically since I came here in 2015. It was already a project undergoing at the time. And our ultimate goal was always to rolling it out on for for our patients, and we thought that we were done the moment like that the AI team like took it over and actually was rolled out in the clinical practice. Turned out not to be the case, and Dr. Lieske and Miller, they had some other ideas on how to use it, and we were very excited about this. But I just wanted to segue to what Tim was just showing, but showing you like the actual impact for our patient. This patient over here was maybe like a month or so ago. This is the kind of report that currently goes into the clinical practice and into the medical records. So clearly detailed description. These are like fairly small stones altogether, clearly classified. The same patient was scanned like a year before, and you can see the difference in the report. Again, we have like the best radiologists in the world, but when you have like 10 or so stones, you're not going and do measurements in multiple directions, like for all of them. And, we can argue how clinically important it is, but as we're tracking them longitudinally, it does matter because all these stones, they add up, right? And so again, here, this on the right, you have the report from a year ago before QSAS rollout on same patient. On the left, you have the report from the most recent scanning, and hopefully going forward, they will all look like this. And to Dr. Goldfarb point, hopefully this will translate also outside of Mayo Clinic. There is no intrinsic limitation that makes it unique to Mayo. So again, as I was mentioning, we thought we were done, and then Dr. Lieske asked us, like, well, can we apply this to like nephrocalcinosis? And he said, and at the time I didn't know much about nephrocalcinosis. So the first thing that I did was I asked him for a few cases to just get an idea of like how nephrocalcinosis presents at CT. And I quickly realized that the presentation can be very, very different. There's like mild cases where it just looks like, like a contrast enhanced exam, right? You see that, like mildly uptake in like on your kidney. It looks slightly brighter, but it still will not confound any software in terms of whether or not you have a kidney stone. Then you have some cases where they look a little bit more moderate and they come together with kidney stones. And then finally you have the more severe cases where it almost looks like the whole kidney is almost entirely calcified. And of course at that point patients might probably be required to have a transplant. So very wide presentation at CT, and this can change very rapidly. This was within like a 3-year span for these patients. Again, like really large increase in the stone burden, but also which again QSAS can quantify, but also an increase in diffuse nephrocalcinosis for this patient. And currently there is no reliable clinical method to quantify this increased mineralization in the renal parenchyma. So really the challenge that our colleagues in nephrology had for us was whether or not we could use QSAS for this different task. And so we started looking at that. So just a quick overview, more into the details compared to what Tim was showing about how QSAS detects and analyzes the stones, the key workflow steps. The first one is either user or AI-based segmentation of kidneys, and Tim was talking about it. And then we have a CT number threshold for an initial first pass kidney stone detection. It goes through like the whole region that has been identified and looks at anything that looks bright and say like, this might be a stone. And then we have a specific stone-based histogram approach that we use to determine like the optimal threshold for each stone, and so that we can actually like properly estimate the volume depending on the stone type and not just have a fixed threshold for every stone. And this was also required for mixed stones composition, so that you're properly capturing stones that have a mixed composition. Now, when you try to feed the algorithm with a case with like severe nephrocalcinosis, unfortunately, the whole process doesn't work, because the first step, your automatic like CT number threshold for initial kidney stone detection basically picks the whole kidney, right? So that is a problem. So when you then proceed to the next steps, now the whole kidney has been considered as like one giant stone. So you have two issues here. You're not properly characterizing kidney stone burden very well, and of course you're not characterizing calcification either. And so that was the first step, and it was clear that using QSAS as it was designed for at least for the severe case of nephrocalcinosis was not going to work. And so we started looking at that task. As I mentioned, basically, if you have a mild presentation of nephrocalcinosis, you can run QSAS and any other CT number-based algorithms, and they won't be affected. If you have severe cases, large portions of the kidney might be classified as a single massive stone, which is incorrect. in addition to that, is not uncommon for the more severe cases to also have transplant kidneys. So now any AI algorithm, now it has to identify like 4 kidneys instead of 2. And, again, yes, they're flexible, but, like if that data set was not in their training set, they won't know what to do, right? And so we believe that like the QSAS workflow, although some changes are required, it offers some opportunities to quantify and track the longitudinal evolution of nephrocalcinosis. So key principles for quantification of nephrocalcinosis was that, first of all, stones are still present. So we still want to be able to quantify number and volume of kidney stones. So that's there to stay. But we also wanted to add on top of it the quantification of the degree of mineralization in the renal parenchyma. So, and we started to hypothesize, okay, we should like quantify the percent of tissue, of the renal tissue that is calcified. We should try to estimate the degree or severity of calcification. It can change over time. And ideally, we would also like to be able to separate cortical and medullary portions of the kidneys. And that is challenging. And unfortunately, that last part, we're not quite there yet, but we're working on it. So how did we modify our algorithms? So we developed like what we call like QSAS plus, because you don't have to be Disney or ESPN to add a plus to your to your name. And so we just had like QSAS plus, because it's doing kidney stone plus nephrocalcinosis. And all this data that I'm showing today is probably like 48 hours old. So, again, and it's not related to the fact that we had to give a presentation just today. Actually started working on it really aggressively over the past couple of weeks. So again, we start with like the user or AI-based segmentation of the kidneys. But then what we do before doing the city number threshold is that we just compute what is like, like the 80 percentile of for the kidney value. And so we figured like even when you have like a lot of like kidney burden or kidney stone burden, they won't take typically more than like 20% of your total kidney volume. And so like, okay, 80 percentile is a reasonable number for still being in the renal parenchyma. And then we just subtract that value, so that if the patient doesn't have any nephrocalcinosis, then it won't subtract any value, basically, because CT number for normal kidney is hovering around 0. But if you have severe nephrocalcinosis, you might have 200 or 300 HUs, CT numbers, and then you just subtract that value. And so your kidney map now looks like something like this. Now it's much easier for the algorithm now to go and detect a stone with the existing method once you have subtracted the surrounding tissue. And so we're doing that. And when we do that, we see that the algorithm actually goes and segments the stone just like it's supposed to do if you didn't have any nephrocalcinosis. And so we do that. We can use the same approach without any changes on computing like the stone volume. So identifying the stones, segmenting them, and computing the stone volume. We didn't have to change that pipeline, which took us years to like validate with the Phantom test. And then the next step is, okay, now that we have classify the kidney stones, we need to go back to our renal tissue and try to classify. And so how do we do that? Well, first we add back whichever value we subtracted on the first step, and then we remove from that mask of the kidney, we remove all the stones that we found. We don't want to like have those drive our metrics. Everything that is left is tissue that we want to characterize. When we do that, and we figure like this is something that has been done before, coronary calcifications and things like that. You compute like the Agassiz score. It's just like a metric that basically tells you how much calcium you have in your vessels. And so we figured that we could borrow that and develop like a pseudo Agassiz score for the whole volume of the kidney. Basically, the more severe, the higher the number, the more severe your calcifications in the kidneys. And so we added an extra class that the traditional Agassiz score doesn't have. And that's because we realized that there were a lot of cases with minor calcification or minor degree of nephrocalcinosis where the actual CT number wasn't that high, and we still wanted to capture those. And so we added an extra class with a scaling factor of 0.5 compared to the traditional Agassiz score classes that are based on these CT number thresholds that are shown here. And then we took a bunch of cases that Lieske shared with us, and we went and we tested it and see how it performed. And so the easiest way to show you how well it performed, I figured, was to just put together a short video, like all the 30 cases we made as you're going through. So you can see that it's properly characterizing all the stones as it's supposed to. You will see that there are different degrees of nephrocalcinosis on each of these patients. And yet, without error, pretty much is going and is classifying all the stones and not classifying the renal parenchyma. So we're very happy with this because that was not the case when we first fed this data set into QSAS. So our algorithm would seem to be working well into classifying the kidney stones. And again, we processed quite a few cases, as many as we could over the last few weeks, and this seems to be working well. So very robust. So again, we confirmed the ability to automatically detect the kidney stones, even in the presence of the more severe nephrocalcinosis cases. Then we went on to try to see how accurately we could estimate the degree of nephrocalcinosis. So we have this patient over here with no diagnosis of nephrocalcinosis at this time point in 2008, and 3 years later, like diffuse nephrocalcinosis. And so we compute like a nephrocalcinosis score, and we can see that this number increased, by close to like 5 times within this short span. This is not severe nephrocalcinosis, but it's still significant. And so we can see that we have the sensitivity to capture this change into the tissue and actually put the numbers to it, right? So we still providing accurate numbers about like the stone volume. So again, it's big increase in total stone burden in this case between these 2 time points. But also now we can add, this is what Santilla QSAS could already do. Now we're adding on top of that the nephrocalcinosis score that again seems to be sensitive enough to appreciate these changes in the renal parenchyma. So again, we can independently quantify both the longitudinal increase in both stone and nephrocalcinosis burden. And then we tested it on the more extreme examples where you have like really diffuse, very severe nephrocalcinosis. And again, the score is significantly higher, like over like 10 times higher compared to the previous case. We also compute in the volume of stone that is affected by nephrocalcinosis. So again, 73% of the stone is affected, of the kidney volume is affected by nephrocalcinosis. And we do that for the left and right. And again, we combine it with the total stone volume that is was already included. And so now we really have a more comprehensive quantitative assessment of this patient disease that we can track over time. So maybe the stone burden might not increase for certain patients, but the degree of nephrocalcinosis might. And with this tool, now you're capturing that. We can do more than that because, again, this is we're doing for research and we're still exploring this, and that we can actually break down the nephrocalcinosis score into the 5 different classes that I was showing you earlier. So, again, like the 0.5 would be like the more diffuse, not very enhancing deposits of nephrocalcinosis, whereas on 4, you would have the really like, basically like that particular location in the kidney is almost completely calcified, almost like a stone at that point. So we can separate those, and so we can better evaluate, longitudinally on patients, like how quickly does a patient evolve from like a 0.5 to like a 4 class, for a specific segment on the kidney. So we can really get granular with this. And again, we're just like, like dipping our feet in the water at this point because we're just starting to get this data, and then we'll hand it over to Dr. Lieske's team and have them figure out what to do with it. But because that's what radiology does. The next step that we wanted to do is that is there a way? I mean, these are like giving them a bunch of numbers is always useful, right? But like is there like a more intuitive way to provide not only the provider but possibly the patient as well about a visual rendition of your nephrocalcinosis. And so we started looking at like, can we actually like make some like color maps of these classes that we have? And so here we have a case again with like, like visually this patient was not diagnosed with any nephrocalcinosis. And so you can see some like scattered distribution of like the lowest possible class, but probably this patient would not be of concern for nephrocalcinosis. And yet the same patient 3 years later, again, diffuse nephrocalcinosis, you can see the distribution. So here, again, like even if, like the number doesn't say much to you, the visual rendition can really do that. And again, you can see that like we remove the kidney stone so that they don't factor into those calculations. They don't like distract you from what you're really trying to assess over here. And here you have an extreme case where really like you really have a different color map. And so visually you can really look at the colors and say like here, like really this patient, it might look similar, but really like the classification here is much more severe. So again, trying to like figure out an intuitive visual feel for the severity of the calcification and not just having like a single number. that might not mean much in without the proper context. So some future direction we need to do. I mean, we did a proof of feasibility with this data here. We need to do a comprehensive validation and some fine tuning of these metrics that we're proposing, but definitely they do look promising. The lack of a ground truth is oftentimes a challenge. For kidney stones, our surgeons, they go, they remove them. We can measure these stones oftentimes, so the stone might pass. We might have some other ground truth for composition. Here is much more challenging thing to do without going ex vivo. There might be a potential for like a reader study working in partnership, with our radiologists to try to see whether our metrics correlate with their diagnosis for nephrocalcinosis. What is the lowest limit for diagnosis, and then start tracking our patient. We provide this data with the work with the Dr. Klein does. If we include it, we can track this patient, longitudinally ideally, and see like, hey, like this tool was predicting maybe some mild nephrocalcinosis. Was it actually true? Well, like sometimes you don't just like wait and see, right? And so, like if we see that, reliably the nephrocalcinosis is getting worse and worse, then we might be able to determine like a cutoff for when this is actually of concern. And that's what, again, we partner with like the clinical practice. There are some dependency on the CT technique factors that we need to comprehensively assess. And there are some novel like dual energy based material decomposition techniques that we might be able to leverage in this field as well, just like we do for stone composition, to better quantify the calcium concentration. And finally, we also need to start looking into incorporating native and transplant kidneys into the AI-assisted workflows. So with that, I wanted to acknowledge the people that we're working with on a day-to-day basis, especially James Anton. He's the image analyst that was behind all the coding that produced like the pretty images that I was showing today. And of course, we're like on the basic science, but we're working with a lot of outstanding collaborators in a variety of disciplines, and we're very blessed from having all of these feedback constantly that helps us not to waste time and really focus on what matters for our patients. So thank you. And thank you all for your attention.
Terrific. I was thinking about Jim Lingaman once said that the way to tell the difference between a kidney stone and nephrocalcinosis was for the urologist to go up the ureter and say that there aren't any stones there. And I wonder whether that is a way of kind of your validation. In a sense, to really say there is no stone there after a ureteroscopy. Because like here, just like as an example, I mean, this may be obvious, but I've seen a few patients that look like this, and you're calling those kidney stones. That could be papillary calcification, sure. And nephrocalcinosis, and not necessarily a stone. Kevin, you agree with that?
Yeah, one of the things that immediately came to mind was looking at these pictures is there's very, very few things are as frustrating for us when we go after a stone or we tell patients, so this is the source of your pain, and then we get into the kidney, right? We're looking at beautiful images of the collecting, the papilla, and there's nothing there to take care of. And if I had only known, or how could I have squinted more and seen whether the stone was under the urothelium or not? And I think tools like this get us that much closer to being able not only to predict that, but then to analyze those in-between cases where maybe doing a papillotomy or treating the papilla itself might be clinically useful. So again, more information that can help us with decision-making. Very, very.
I mean, this could be like huge, right? And help you with stone-free, because you do these ureteroscopies and you get an ultrasound and you tell the patient there's going to be stone, it doesn't really make a difference. So it's impossible to tell if they're stone-free, what's intraluminal, and I mean, all of these modalities have such an application from interventions. This is really exciting, and it could help with counseling, success markers, and things. It'd be awesome.
Have you done any non-PH patients yet, or is this all primary hyperoxaluria?
I think so far those are the only ones we've done. We tested on a few just regular kidney stone patients that we had in our records just to make sure it was still working properly, and just like sort of validation. But we will get like very small numbers for nephrocalcinosis.
The kind of nephrocalcinosis you see there is quite different, probably, than any other patient with a sponge.
Sure.
It's more corticomedullary.
Yeah. Right. I mean, a question about cystinuria and exactly what that does, and whether it leads to a kind of calcinosis. It's not calcium. Excuse the word nephrocalcinosis, but with cystine as the composition. right? And the patients with medullary sponge kidney that we're putting in, right, as John said to me, how do you know those patients have medullary sponge kidney? And we don't. I think a lot of them have nephrocalcinosis. The nephrologist or the urologist or the radiologist says consistent with medullary sponge kidney, but the likelihood, they haven't had a contrast study, and we don't really know if they really have medullary sponge kidney or not. That would be an interesting group as well.
The truth is going to be hard here. Testing.
Yeah. What? Visual inspection is probably the best bet.
Genetics,
yeah. You could isolate the patients
that would be the gold standard of a sort, right?
Right.
They got to be set a cohort. Yeah.
Beautiful. Thank you so much.
Thank you.
Great. Andrew Ruhl, can AI help us predict symptomatic stone events in persons with asymptomatic kidney stones?
I'm going to kind of talk about, brainstorm an idea of how we could potentially use some of these exciting tools that Dr. Ferrero and Dr. Klein showed us to do an epidemiological study and see if we can answer the question, can we predict symptomatic stone events in persons with asymptomatic radiographic stones? So here's my outline. We'll first look at risk factors for symptomatic stones, talk about radiographic stones versus symptomatic stones. I'm going to give you an example study that I just got funded looking at using abdominal CT AI models for epidemiology. Then we'll talk about what we might be able to do. So let's first review risk factors of symptomatic stones. We know composition is important, and certain compositions are predictive of recurrence, like uric acid, struvite, brushite, of course, cystine's even more predictive. What's also very predictive is has the patient had a symptomatic stone in the past. So if they've had a stone, it's their first episode, the risk of another stone episode is lower at 10 years than if it's been their second, third, or fourth episode. So this needs to be accounted for in our modeling because there are other factors that predict recurrence. And we've studied this fairly extensively, and things that are predictive clinically of symptomatic stones recurring or occurring as younger age, being more obese, having a family history. Men are more at risk than women. If they've had prior incidental asymptomatic stones on imaging, they've had suspected stones where they didn't have no stone was ever seen, but was thought to be the cause of their symptoms. They were pregnant at their last episode. Composition, as we mentioned. And then some radiographic things. Lower pole stones seem to be more predictive. Urovesicular junction stones, less predictive of recurrence. The more stones, higher risk of recurrence. Intermediate size, if patients have bigger stones, they tend to get removed surgically. So that might be why they're less likely to be predictive of recurrence. Number of episodes, as I mentioned, and also how recent they had their last stone episode. But even with all that, we only get a C statistic corrected for optimism of 0.68, which is not ideal for a predictive tool. And so we have made this into a nomogram that can be used. But, one of the things I just want to highlight is that things I think we just don't think about is how recently a patient had a stone. If they recently had one, they're at highest risk. The more time period a patient goes, more years without having an episode, the risk proportionally goes down. And that's been shown in these coefficients. The lower the coefficient, the higher the risk of recurrence. So the higher the coefficient, the lower risk. So with more years since your last episode, the lower risk of recurrence. So radiographic versus symptomatic stone. So we did a prospective study. We look at stone formers with a baseline asymptomatic stone, and we found they were more likely to form a new stone than those without. Not the stone that they already had, but they were just inherently a higher risk group. And if they had, and that stone that they had at baseline at 5 years, about 43% of the time it grew, and about 47% of the time it passed. And a smaller percentage, it didn't change. So about half the time, if you have a stone that's left behind after your first episode, you'll pass it. But if you pass it, there's only about a 50% chance that you'll report symptoms, either by survey or by clinical care. So this is important. So 25% of stones do not lead to a symptomatic episode in 5 years when they're seen radiographically. And this may influence our ability to predict recurrence, because some patients will pass stones and we just don't realize it because they don't report symptoms. And then even if they don't pass their stone, they can still have symptomatic recurrence from another stone. So it wouldn't necessarily be the same stone that they had. And if they don't have a baseline stone, then they can still have a similar rate. So they have this baseline rate just because they're a stone former, and then there's that radiographic stone that's been retained. And so that has to be considered in the analysis. And the point I want to make here, and I think this is one of the key points, is that we can't predict symptomatic recurrence well if we limit it to just things like clinical care for stones, or even clinical care or self-reported stones. There we get a C statistic of our model, our prediction ROC score of about 0.692. But if we include just stone passage on CT, that actually does a pretty good job of recurrence. And that's because you have all these stone passage episodes that don't get reported by the patient. And if you put all these things together, you get, your C statistic is, 0.8. But you've gone up from a 5-year event rate of 30% if it's based on symptomatic recurrence alone, to 67% if you account for things like new stone formation, stone passage, or stone growth. But we have to understand what's the relationship between new stone and stone growth with subsequent symptomatic episodes. And we have to figure out what do we do about stone passage. But a model can only do as well as the outcome is defined. And if the outcome is something you can't detect, like stone passage without symptoms, that's going to limit how well the model performs. Now, if we look at what the characteristic of the stone burden that predicts symptomatic recurrence, and we looked at this a number of years ago, we looked at the total stone volume, the number of stones, the diameter of the largest stones, bilateral stones. When we put this all in a multivariable model, the only thing that was an independent predictor was the total stone volume. And so I think, that's one way to reduce the complexity of the data into what seems to be the most prognostic predictor of symptomatic episodes is it's actually the total volume of all the stones added together. Now, if we look at that, these are, of course, patients who are seen in stone clinics, so they're more high risk, so they have more recurrence events, but it occurs kind of on a logarithmic scale. So you have some risk for a very tiny stone. It increases when you go to kind of a 79 to 1,000 millimeters cube stone, and then increases a lot more with a greater than what would be a 1 centimeter cube stone. So the risk is not linear. It's more of on a logarithmic scale. Okay, so next I'm going to talk about epidemiological studies with abdominal CT-AI models. So one of the things we can do here that's somewhat unique is we can study this well-defined population that's fairly rural. It's very well defined in Ulster County as well as the near counties, but also pretty well defined in the far counties, where we can capture nearly all medical care. And Ulster County goes back to the time of the Mayo brothers, but these other counties go back, now 15 years, sometimes 20 years. And so they can be used also to quadruple our sample size from what we would get with Ulster County alone, as well as to increase some racial and ethnic diversity in our sample. So if you look at people in the general population, this is a representative sample of the general population, what percent get an abdominal CT scan between 2010 and 2020? It's actually quite high. 35% of adults overall will get an abdominal CT scan. Now, you can look at that different ways. That's too much, that's too little. As a scientist, I'm like, great, it's more data. And I'm not going to worry about that. And in fact, that was the argument I made in my grant, as I said, look, we do a lot of CTs. We can decide whether or not they're needed, but they allow us to get a snapshot of what's going on in a patient. And there's a lot of data there that we can try and tap into. And it doesn't vary much by gender, race. Older people get more than younger people, not surprising. Now, the other thing we can do is, of course, you're going to say, well, people who get abdominal CTs, they have more comorbidities. And that is true. If you look at, almost 200,000 individuals who had a CT scan in this time frame, and then you compare that to the general population, they had more hypertension, they had more congestive heart failure, coronary disease. These are all the DHHS chronic conditions. And in every single one, they were higher in people who get an abdominal CT than the general population overall. But we can use statistics to fix, we can do, here what we did is we subsampled to make a representative cohort that had the similar frequency of these chronic conditions. What we plan to do in order to use all our data is do something known as inverse probability weighting, where we can weight our sample to make it more representative of the general population. We can do it more than just on chronic conditions. We can do it on laboratory tests. We can do it on medications, and we can do a lot of the enriched data in the rep to try and make a more representative sample that gets, and what we're trying to deal with is this whole selection bias. People who get CTs are just inherently higher risk individuals. And then what we did in this smaller sample of about 5,000 CTs is we applied some of these deep learning models. We did one that looked at identifies the liver, the kidneys, the aorta, identifies aortic calcification, identifies the spleen, which lets us normalize for contrast intensity in the liver. And what we mostly have done with so far is actually body composition. And what you do with this model is you identify 4 different tissues, organs, which you kind of throw out for this, and then you do muscle, skeletal muscle, which you can measure here in pink, and then you do visceral fat, which is shown in yellow, and then there's subcutaneous fat, which is shown in green, and then you have muscle, which is shown in red. And so you can quantify the volume and, if appropriate, the density of each of these structures. And for this study, we were interested in aging. We want to kind of estimate tissue age. And so for each of these individuals in the population, we plotted their chronological age on the y-axis and their various body composition measures on the x-axis, and then we fit regression models. And visceral fat increases with age, muscle area goes down with age. The thing that really changes with age is your muscle density. You get kind of a fatty infiltrate into your muscle. This is actually the strongest thing. And then bone area goes up with osteophytes, and then you get some decrease in bone density. And if you put this all together, you get a model that can estimate somebody's tissue age. And then you can divide people into those who are kind of along the line of regression, where their chronological age predicts their tissue age pretty well. But those who are in the upper quartile, where they have older appearing tissue age, and those who are in the bottom quartile have younger appearing tissue age. And so now we can ask this question about tissue age and what does that mean for patient outcomes. And so just to show you some representative images, what we did is we tried to identify an example CT scan of what was the typical characteristic presentation of a 25-year-old woman, 55-year-old woman, an 85-year-old woman. And we did the same thing for men. Men tend to have more visceral fat, as you can see. But the other thing you can see, I think, is the increase in visceral fat with age, and then the loss of muscle mass, the atrophy, as well as the hypodensity of the muscle that you see. You see more, less intensity, decreased CT number in the muscle as people age. And then what does this mean? So we've looked at it, a number of things, but just to simplify it in terms of predicting mortality, if you have younger appearing tissue age or average tissue age, not a big change, but if you have older appearing tissue age, it was a strong predictor of mortality. And this is independent of, diagnoses, chronic conditions and stuff that also predicts mortality. So this is the kind of thing you can do where you take an AI model, you quantify some characteristic, then you use that to try and predict some clinical outcome in a population-based setting. So can we do something similar with kidney stone epidemiology? And I have not done the study yet. I want to kind of brainstorm here with you guys, show you what my idea would be, and get your feedback. So I welcome your input here. So, one of the questions is, what's the risk of a first symptomatic stone episode in a person who presents with an incidental asymptomatic stone on CT? This is a common question. People get a CT scan, they see a stone, they don't know what to make of it, they're worried about it. What is that person's risk? Well, here's the challenges. We don't know when that stone formed. We don't necessarily know how it grows because we don't get repeat CT scans on a lot of those individuals. Some of them will get a preemptive surgery that will remove it. Some of them might pass it, and then they never, never get seen again. They don't, we don't know when it passed because there's no way to know about a stone that somebody self-manages. Maybe they have pain and they just don't report it, or it just passes without much symptoms. And you need years and years of follow-up. These, stones in this kind of a setting, we're on the scale of decades sometimes. We're not on kind of the tip of the iceberg that urologists and stone clinic nephrologists see. And then we need to be able to detect all the clinical encounters for when they do have symptoms so we can understand that relationship. And that's where I think the things like the Rochester Epidemiology Project, which ensures that all the care these patients get, we can capture because it's contained in our database. So I always like to ask, when you design a study, what's the ideal study design? Maybe not the realistic one, you couldn't do this, but could you take, if there wasn't a radiation risk and you wanted to answer the question, or use an animal model or whatnot, you would do serial CT scans over time, and then you would have these individuals where a stone might show up in their kidney on a CT, and then maybe somewhere along the line, you'll see that stone, maybe it'll grow, maybe it won't. And then at some point, maybe they present for care for a symptomatic episode. And then maybe there'll be somebody who gets worried about their stone and they get it surgically removed. Maybe there's somebody whose stone just stays there and it never goes anywhere and it never passes. And then maybe there's somebody who presents with a symptomatic episode. There was no radiographic stone beforehand. And then they have intermittent stone seen and symptoms. This is kind of your typical more aggressive stone clinic patient. And then there might be individuals who just pass a stone and we never know about it. Now, this isn't the data we're going to get, but if we have that data, we could really answer the question. What we get is probably something more like this. And this is the challenge in epidemiology. We're going to get little bits of information on each individual. We might get no CT, so we don't really know what happened. We might get a CT here, which showed us the rate. This is the same patient. I'm just removing some of the CTs and what the information we would lose. And so for each individual, we get these little snapshots at time. But what we have thousands and thousands of individuals. And this is kind of what statistical modeling is for. It's like you take a little bit of information from each person, and then you try and model the sum is greater than the individual parts. You try and get some population level estimate of when do radiographic stones form, and it would be kind of population average type estimates, but it might teach us something about what is the relationship between radiograph stone formation and symptomatic stone. So if we could, for every CT out there, and serially get this kind of information, and we get all the symptomatic episodes, we will have very incomplete data. But if we have enough individuals and we model it correctly, do the right censoring to try and minimize biases, we might learn something about stone disease. So some of the strategies, I think Dr. Ferrero mentioned that there might be a way to do contrast enhanced studies. So in case you weren't aware, most abdominal CTs are with contrast, and that's just the reality. So if we restrict it to non-contrast CTs, we get more bias, more selection bias, and we throw out a lot of data. So if we can get stones seen on all abdominal CTs, 35% of adults, we get a pretty good representation. We would then apply this model, like the QX, what is it called? QSAS, okay. You apply a QSAS or some variation of it to all the CTs, and generate this data, validate the symptomatic stone episodes, which we've done before. We've got a system for to do that, and then try and create some sort of a statistical model that incorporates these rocks, these other characteristics, the number of episodes, other things that predict things like composition, and then try and understand the relationship between radiographic stone formation and symptoms. So I will end with the question: could this work? And stop here and see what you guys think.
Vote. People think this will work. Don't think it'll work. Nobody.
I leave the room and then you can re-vote.
What's the second bullet? So you would apply. So the AI model would create the CTs for the 200,000 adults.
The AI model would take 600,000 CTs within. So there's about six. Some people have more. There's on average 3 CTs per person.
I see. Okay.
Some people have more CTs. Some people have fewer. There's a lot of people have a lot.
I get it.
But we would apply to all of them, and then from that model we identify all the radiographic stones in the kidneys.
So of course I'd want to know the answer to, I would love to have a predictive model, but from a practical standpoint, I keep asking myself, would this change anything I do in clinic? And I don't know if I think the answer is no, because, you could say, well, what is the chance that this stone will pass? And to the patient, the answer is either 0% or 100%, right?
Yeah.
And so my counseling is not going to be any different if the model tells me it's 20% or 50% or 80%. Either way, I'm going to counsel them what to do. What are the signs that the stone is passing, and what should you do about it? And when is it a real emergency? And you better get to the ED quickly. So that's the only question from a practical standpoint. Does it change the way we counsel? I don't know if a patient feels better if it's quote unquote only a 20% chance of passing.
I would say from our standpoint, it probably would, because you have a lot of these patients where they're debating, do you want to have a procedure or an intervention? If I can tell them your likelihood of this causing symptomatic event is 85%, I think those patients would be much more likely to choose to have a sort of prophylactic ureteroscopy than if it was 25%. And then selfishly, sort of a sub-analysis that maybe that more defined is just look at surgical patients. So they had a CT, then they had a surgery, predicted value of success of surgery with stone-free rates, kind of surgery, those types of things. Then you'd have to define info with the CT, and all of them would have a defined colon event. Less applicable to the broader population. Sure. Those also more limited. But it's a good point. I think it would help in our thing. I don't know what you think. I think it's. But because we have a lot of these patients where you go back and forth, like, do you want a ureteroscopy? We just watched this. It may pass, it may not. It may cause symptoms, it may not. And our ability to predict that is important.
I think that the information is actionable because it predicts, or at least it gives us our patients information about not only if but when, right, stones may become symptomatic. That could be potentially useful for patients. Now, and I think an entirely separate issue here is how our patients even understand the concept of risk and probability, which, as we know for many of our patients, especially in our EP area, is very, very poor. But that notwithstanding, I think this could be, I think it could be useful in counseling and at least from the intervention point of view, deciding or helping patients decide whether something prophylactic or preemptive might be in their favor.
Well, the classic use also for epidemiology is to define event rates or to identify your population that you want to do a clinical trial in. So if you don't know who's going to have symptomatic episodes, it's hard to study a treatment. So that might be one use, is that you can define populations that are high enough risk that we want to do prophylactic surgery and randomize them and see if that makes a difference in some sort of a pragmatic trial. Yes?
How would you control or understand the role of prevention or medical management?
Yeah. So it's interesting in all our models when we put in what's least documented in terms of diet and
alkali.
Yeah. It doesn't work. I'll be honest with you. There's a problem here. That the more the patient recurs, the more it gets documented that they're being told to do it.
That's true. Yeah.
And so I think it's kind of one of these things where it kind of, the more your problems you're having, the more likely you're getting told, and so it makes it look like it doesn't work. So.
Yeah. That New England Journal of Medicine article was right.
Well, you mean the thiazide paper?
Yeah.
Right. Yeah. Well, and the Cochrane review says that there's no really convincing evidence that water drinking actually helps.
Yeah, and Gary Curran, I think, did a study where they looked at the diet, and you're probably familiar with this study, and like a year later, I mean, almost nobody was following it. People don't change their diets very well. It's very challenging. You might disagree with that, but that's my experience.
We haven't met Chris Benison yet, but
yeah, but I made that point before about Sorensen's paper, where randomization of patients showed that taking out small asymptomatic stones was effective, right? And that we made that about your thing, right? And so yeah, right. Yeah, exactly. Right. Lingaman was on there. Right. And so there is that argument to make that, whatever the analysis is, those patients should have their small asymptomatic stones, particularly if they've had a previous episode. And that goes to your thing about the more episodes you've had, the more likely it is.
There's probably a subset of patients that drive that benefit that you could potentially identify.
Right.
I guess I'd argue, I don't think you can show treatment effect this way very well, like you said, but it'd probably be in your AI model that they were given a 5, right? I think the risk prediction is going to be very helpful because, people want to know if I've got an 80% chance of having a stone in the next year or whatever. They may be more likely to want a medicine that's 10% over 10 years, maybe not. But I think the risk thing, I don't.
Well, and then like next level stuff could be like, well, what if we could correlate that prediction with all the millions of other data points that we have in the EMR, like urine osmolality, like if we wanted, as a proxy for water intake, right? So there are all these other data points that we get that could correlate with, show the effectiveness of treatment.
I mean, to me, the most what I'm interested in is let's just understand what is the natural history of the kids.
Right.
Yeah. I mean, and maybe that's just an academic, intellectual physiology question, but I still think if we learn that and we understand what is the distribution of stones and how they progress and when they become symptomatic, because our clinical care is just a snapshot here and a snapshot there, and we don't know the full picture. And until we know the full picture, it's hard to know how to design the right studies that would intervene.
Yeah, I agree. I think the most important part would be because a lot of treatments have been done for years, and there is every now and then people like now thiazide should not be used, like citrate, this and this. And the clinical trials are very difficult because you have the outcome of kidney stones that is like, how long do you wait for? 2 years, 3 years, 5 years, 10 years. So if you can identify the subset of the patients that are the highest risk, although they will not represent all the population, but at least you start there to show an effect and you'll be motivated because we don't have so many newer treatments for diabetes.
The previous talk based on the abdominal CT, the muscle degradation with the aging kind of thing, can you predict that biological age of that individual?
Yeah, we have a paper in press at the Mayo Clinic Proceedings where we provide us an Excel spreadsheet where you put in 7 biomarkers from the CT and it calculates what your tissue age is.
Well, I think that really the day has run the entire spectrum of the broad and diverse community of work that that we do. I'm so glad that we've been able to bring together all of these voices in the same room. And I think it's really highlighted that although we are analyzing and studying many different things in many different ways, that our commitment to advancing kidney stone science and kidney stone care is what unites us all. So we're so proud to have hosted this event today, and we're so glad you were able to join us, whether online or here in person, and we hope that you'll continue to work with us and partner with us as we advance kidney stone care across our whole community. For the final remarks, Dr. Lieske.
Thanks. It was great. I think we maybe were a little bit over ambitious with the agenda, but I think partly that's we haven't had a meeting in so many years and there was just a lot to do. And so I thank everybody for their talks. I think we can be adjourned. Thanks.